यार काम यही है ना, माँ कसम, जरा ये निकाल के तेरे
हो गया हेलो
तेरे हर काम यही है।
चला
चला
चला क्या रेड यही चला था। हेलो गाइस, नाउ
Hey guys, what's up? How are you?
सरप्राइज देखा।
Yeah, surprise.
Ma'am, please, human health and disease. Our health is not fine, Komal.
Yeah.
See, our health is not fine.
Mm-hmm.
So how are you all?
They're good. What are you? माइक लगाएगी।
मुझे क्या पता था ये इतना यूजलेस है।
You know, whenever you see problem in life, that means Ambika would be around.
अब तो मैं भी मान गई हूँ यार।
ऐसे नहीं लगाना होता है।
बस ठीक है।
हाँ बस ऊपर रख थोड़ा।
ऐसे। Hi everyone, how are you? All good? Sir, some noise is there. अरे sir is a noise. Okay, done. Now I believe it's fine.
Yeah, it's fine now.
Now I believe it's fine. Thank you, Rabia. चलो अब आप जाओ. You are a problem solver. Who? He? सच बताओ.
और क्या पढ़ा रही है आज? हाँ? पता नहीं है.
Principal.
She does not know what she's going to teach today. She has
That's
no
true.
idea.
You know, my studio is not working fine, so I'm here at his place. Okay, so I was just trying to be over smart, but ma'am, our session complete होगा ना? Komal.
Someone is saying crystal clear, ma'am. Already the topic is crystal clear. She's not started yet, huh?
He's my student. He knows that.
Okay, nice, good.
So
Good.
So everybody just hit the like because Wasim sir is here.
No.
Matlab somebody is writing Wasim sir is your husband. Seriously? Matlab I don't want to spoil my life.
Nor do I.
Chalo, so let's start this. Now you can leave. Everything is working fine. Thought ma'am was crying. I'm crying. है ना? हाँ, ये suffocating है. इसको बंद करो.
बंद करूँ?
हाँ, नहीं बंद करो.
कम कर.
अरे नहीं, मेरे लिए मैं आपकी तरह सॉफ्ट सी नहीं हूँ. I'll manage.
You'll manage?
हाँ, इसीलिए आप बीमार हो रहे हो. वैसे तो मैं भी कौन सा ठीक हूँ, बट ठीक है. चलो. हाँ, मतलब the moment I'm saying that he's not well, he's doing yeah.
तो Ambika कैसी है?
मैं सही
में चलो guys, you guys carry on, ठीक है?
करूँगी आज.
चलो, good luck.
मैं सही में क्या ही करूँगी आज? पता है, this room is reminding me something that is not good. So, you know, Wasim sir's studio is so clean, so nice, so good. I believe you all can see these patches on the screen as well, right? If you'll focus, you all can see that.
Wait.
See, गीला कपड़ा नहीं मारते स्क्रीन पे.
हाँ, ये मैंने थोड़ी मारा है.
चलो कोई बात नहीं, अभी जाओ. अरे नहीं, बस ठीक है. I'm just kidding.
Yeah.
His studio is really dirty.
चलो.
I'll record it from his studio. So now today, let's start this chapter. Basically, my studio is not functional, so that's why I'm at his place, right?
She's also not functional.
I am functional. So basically, my studio is not functional. That's why I'm at his place. So today, I'll be able to complete the chapter, right? Because he has all that electricity arrangement and all. But you tell me the screen is clearly visible to you or not? Like if there is any problem, if screen is not, I think everything is clearly visible, right? Just let me arrange it a bit. This man is going to kill me. Okay, I think now it's fine. Just a minute. Zoom is not working. Just a minute, guys. Okay, fine. Now I think it's fine, right? So the chapter that we are going to start today is very important. It is the principles of inheritance and variation. So you tell me after this particular chapter, what do you want me to start? You want me to start molecular basis of inheritance or the evolution? You tell me. And I'll use his studio and I'll record the human health and disease chapter as well. Okay? Okay? So I'll record his, human health and disease as well. As I said, I'll post the recording. I will definitely post the recording. It's just that studio is not working. Okay? Studio is not working. So let's start this chapter. So you tell me which chapter you want me to start next as well. Okay? Tell me which chapter you want me to start next as well. Molecular or evolution? So later on, you guys can comment that in the comment section. So now let's start the session. So the chapter name is Principles of Inheritance and Variation. Okay. So what is the name of this chapter? The name of the chapter is Principles of Inheritance and Variation. So let's start everything one by one. So we will start with the first word, that is genetics. Right, we are going to start with the first term, that is genetics. So students, when we talk about the genetics, what exactly is the genetics? Of course, it is the branch of biology that deals with, the study of, yes, that deals with the study of heredity and variation. This is what genetics is. So in simple words, if I have to write it, so what will I say? That genetics means, yes students, genetics means heredity plus variation. This is what genetics is. Are you getting my point? This is what genetics is. And this is what we are going to study today. Is that clear? Right? Now, when we are talking about this word heredity, what exactly is the meaning of heredity? Heredity means transmission. Transmission of characters from one generation to another, or simply you can say from parents to offsprings. Yes or no? What exactly is the heredity? Heredity is basically the transmission. The characters, they will pass from one generation to next. And we know that that is from the parents to the offsprings. See, we know it very well. If you talk about a dog, right? A dog is going to give birth to dog only. An elephant is going to give birth to elephant only. A human is going to give birth to human only. What is the reason behind? Obviously, everything is hidden there in the genetics. Basically, we talk about the genetic material here. We talk about the processes by which that genetic material passes from one generation to another. We discuss the process, and here in this chapter, we will discuss that process in detail. Is that clear? Is that clear, everybody? So that's what we have to study, right? That's what we have to study. So as I said, heredity. So what exactly is the heredity? Transmission of characters from parents to offsprings. That is what the heredity is. And now, what about variations? What is the meaning of variations? So students, when you talk about the variations, see, it itself is explaining everything, right? What exactly is the meaning of variations? When we use the word variations, so variations means to vary, right? Means it is a degree of difference, right? It is actually the degree of difference by which the progeny differs from the parents, right? Like see, parents, they'll give birth to obviously parents, they'll pass their genetic information that will be there in their progeny, isn't it? But we are not ditto same like our parents. We are not their replica. We are not their carbon copy, right? We are not just their photocopy that exactly we are like them. No, we vary from them, isn't it? So the degree by which progeny differs from parents, the degree by which progeny differs from parents is called variations. What's that, student? That is actually the variations. And for variations, two things are responsible: mutations, which we call as sudden changes, and recombination, which is also known as crossing over. These two things are responsible for the variations. Okay, so we started with the genetics, branch of biology that deals with the study of the heredity and the variations. When we talk about the heredity, it means transmission of characters from parents to offsprings. It means the transmission of characters from parents to offsprings. And when you talk about the variations, it is the degree by which progeny differs from their parents. And how these variations occur? So these variations occur due to the mutations and the recombination. And these variations, they are very important. They are very important to adapt in the changing environment. Is that clear, everybody? Up to this part, all clear? Yes. So now there is one more word that we have to understand. And what is it? It is the inheritance. I have seen that sometimes students think that inheritance and heredity is same, right? But actually that is not the case. When you talk about the inheritance, inheritance is the process. Okay, like heredity is the transmission of characters from parents to offspring. Inheritance is the process by which the characters pass from parents to offsprings, right? By which the characters pass from parents to offspring. So it is the process behind that transmission. And students, this inheritance is actually the basis of heredity. Am I clear? This inheritance is actually the basis of heredity. Am I clear? Yes, everybody. So we'll start with the basics first. So this is the meaning of inheritance. Okay. So now you know principles of inheritance. Principles means we are going to talk about the rules, right? That will tell us that how this transmission is taking place and what are the reasons behind these variations as well. So this is something that we are discussing. Okay. So this is something that we are discussing. So I believe up to this part everything is clear to you. Okay, so what's the next point that we have to discuss? So next thing that we have to discuss is, as I said, we'll start with the basics first. See, all you know, and overall, if I have to see, here I'm writing it, term genetics. Term genetics is given by William Whitson. He was the one who gave the term genetics. Who? William Whitson. Now, if you talk about the father of genetics, I believe you all are aware of it. If you all are aware, do let me know in the chat section. Father of genetics. Who's the father of genetics? Who's the father of genetics? So father of genetics is Gregor John Mendel. Gregor John Mendel. Then next is father of experimental genetics. So when it comes to the father of genetics, that is Gregor John Mendel. When it is the father of experimental genetics, then you will talk about the Thomas Hunt Morgan. Right? Who's the father of genetics? Experimental genetics rather. Thomas Hunt Morgan. And then father of modern genetics. So when you talk about the father of modern genetics, then again same, William Whitson. Right? That is again going to be William Whitson. Is that clear? Is that clear? So of course here in today's lecture we'll firstly talk about some general terms, then we'll talk about the Mendel, right? And then after that post-Mendelian inheritance, then chromosomal theory. So these are the topics, these are the things that we have to discuss. You can see the same from, guys, is it visible from this side also? Just a minute. Let me check. Okay. It's visible. Okay, so this is what you have to remember. Okay, right, this is what you have to remember. Okay, so we have to start some general terms, right, that will help you to understand the genetics in a better way. But overall, just a minute. Wasim sir is going to kill me because I'm disturbing his studio settings, but it's okay, I don't care. So this is what we need to know. So we'll start with the Mendel. You know that Mendel, if I start about a little bit about the Mendel. So Mendel belonged to, the family to which Mendel belonged was not financially very sound, but since his childhood he was quite interested in the plants and all because of his father, of course. So he was there in the Augustinian monastery as well, right? You know these basic things regarding Mendel. There he studied along with natural sciences, he also studied the mathematics, and because he had that keen interest in the plants as well, right? He observed many things. So basically whatever laws that we discuss, like the basic laws of inheritance, it is all because of Mendel, isn't it? So all that things, From where they came? Obviously from the observation. He started observing the things. He started noticing the things. Then, of course, he had that keen interest in the natural sciences plus in the mathematics as well. He applied that sciences and then he provided us some results. Because of that, we know about these basic laws of inheritance. So this is what we have to discuss. So when you talk about the Mendel, he was not the one who used the term genes. He used the term factors. Okay. Now, at this point of time, we all know what genes are. Basically, gene is the basic unit of inheritance, right? We know that we have the DNA, which is the genetic material, and that DNA is having the gene, which is the basic unit of inheritance. But the term gene is not used by Mendel. Mendel used the term factor. Later on, it was renamed by Jonasson. Jonasson called it genes, but initially, Mendel called it what? Mendel called it factors. Okay, so this is what we have to discuss. So before starting these general terms, right, I'll start from the basics. Okay, I'll start from the basics. So if you talk about the Mendel, at that time, like when Mendel conducted all his experiments, at that time, this concept of chromosomes, the concept of meiosis, it was not there. Okay, it was not there. But now, because we know, so I'll relate some things here initially. We'll discuss the chromosomal theory of inheritance later, but we'll discuss few general terms. See, you all know we are diploid individuals, right? Our 2n is, right, 46 chromosomes. Basically, we have 23 pairs, isn't it? We have 23 pairs of chromosomes, or I would say we have 46 chromosomes. So basically, our chromosomes, they are in pair, isn't it? They are in pair. And you know that this pair of chromosomes, what do we call it? We call it as homologous pair of chromosome. Right, homologous pair of chromosome. So what exactly is the homologous pair of chromosome, students? When you are talking about the homologous pair of chromosome, homo means same, right? The chromosomes having same shape, same size, same centromere position. The chromosomes having same shape, same size, same centromere position and same genes as well, but yeah, alleles can vary. I'll explain this to you, but alleles can vary. So these are the homologous pair of chromosomes. So homologous pair of chromosomes, one is the maternal chromosome, another is the paternal chromosome. So homologous chromosomes are the one which are having the same shape, same size, same centromere position, even the genes are also same, but the allele can vary. I'll come to this line, I'll explain it to you. But allele can vary. Is that clear? So genes are also same, but allele can vary. See, it's not like that. Many students, they say that this is a chromatid, this is not a chromosome. Actually, it is also a chromosome, but it is having the single arm. It is monad, right? And when you talk about this chromosome, it is having two arms, it is having two sister chromatids, right? So here you use the word dyad, right? Here you use the word dyad. Ultimately, students, chromosome is made up of DNA and the proteins, okay? And obviously that DNA is having the genes. And we have already studied it, right? That during cell division, you know that during interphase, there is a phase known as S phase, in which you see that if initial amount of DNA is 2C, it becomes 4C, but chromosome number remains same. Do you remember this concept? Yes, but chromosome number remain same. We all are aware of this concept. We know that before a cell divides, right, it undergoes an interphase in which there is a S phase, and in that S phase, the DNA amount, it gets doubled, right, but chromosome number remains same. So before this, right, doubling of DNA or before the application of DNA, the chromosome is like this, single arm, monad. Okay, obviously it is not condensed in this particular sub-stage, but just to show you, I'm telling you, right? Obviously we see such type of chromosomes when the cell divide, but just to show you, right, so that you guys can understand this part. So I'm telling you that imagine this is the chromosome, right? So this chromosome, it is having single arm, means its initial amount is 2C. In S phase, its amount will get doubled, DNA amount will get doubled, so it will become 4C, right? So initially, if let's suppose it is 50 picogram DNA, it will become 100 after S phase, but number of chromosome remains same. So it is monad, it is dyad. So this is also a chromosome. You cannot call it as a chromatid. But when you look at this particular chromosome where, you know, these two arms, they are attached to one centromere, then what do you call it? You call it as sister chromatids. So that is the difference that I'm telling you right now. Now, when obviously during the division part, what is going to happen? The chromosomes, they'll show the pairing, right? They'll do what? They'll show the pairing. This is what we have already studied. And I'll tell you that why are we discussing it here in the genetics. Just allow me 5 more minutes. You'll be able to understand it in a better way. Okay, so this is the first thing, right? That you know that when there is a cell division, even our chromosome, right, they just get paired. And this is what we see. See, they just get paired. So one chromosome will be the maternal, another chromosome will be the paternal one. So this is actually the homologous pair of chromosome. This is actually the homologous pair of chromosome. Are you aware of it, everybody? Are you aware of it? Now, are you all aware of it? Yes or no? Right? So this is what we already know, that this is the homologous pair of chromosome. Now students, when you look at these chromosomes, maternal or paternal one, let's suppose if here you have, right, you have a gene, of course, because this is the duplicated arm, right? Right? Or see it in this way. Let's suppose in this chromosome, here you have a gene, right? So the position of that gene on a chromosome, we use the word locus for it. What is the term that we use for it? Locus. What is the term that we use for it? Yes, it is the locus, right? So locus means position of gene on that chromosome. Like it's just like house of that gene on chromosome. We call it as locus, right? So singular is locus, plural will be loci, L-O-C-I. So this is the locus. Let's suppose here we have a gene. Let's call it as A. Right? Right now understand this. Now if this is the chromosome, in the S phase, its arms will get duplicated. So if here you have A, here also you will have A now. Like let's suppose if I will get duplicated, right, you'll get exact replica of me, right? Right, you'll get my exact replica. Same is the case here. This is the chromosome. Let's suppose at this position, so we have a gene A. So if this chromosome will get, like if its DNA will get doubled, so obviously this arm, because it is just the duplication of this only, so here you will also have the A. This part clear? This part clear? Now, the another thing here that you need to understand is, right, see, let's suppose here, as I said, A. Now, if it is a homologous pair of chromosomes, so the another chromosome, right, the paternal chromosome, right, this is a maternal, this is a paternal chromosome. Here also, either you will have capital A or you will have small a, right? Like you will have the same gene, but allele can vary. You will have the same gene, but allele can vary. Like it can be like this also. Or if it is a homologous pair, so anything is possible. But all I'm trying to tell you is that if it is a homologous pair of chromosome, right, then at this locus, if you have gene A, or you can say that gene for any X character, so here also you will have, at the same locus here also you will have the gene for that X character only. This is what I'm trying to tell you. The allele can vary, but the gene should be same. So this is the meaning of homologous pair. But now, ma'am, why are we talking about the homologous pair here? Why are we talking about the chromosomes here? Because see, no doubt we have to start with the gene part. But the point is, why do we say that gene occurs in pair? Right? Why do we say that gene, they are in pair? Why do we say that gene is a pair of alleles? Right? Why? Because overall you see, we are also the diploid organisms. We are having our chromosome in a paired form. Right? We have the maternal and the paternal chromosome. Right? One chromosome is from mother's side, another chromosome is from father's side. So obviously if we will get one gene from our mother, the another gene from the father as well for the same character. So this is what I'm trying to tell you. So that is why I started with this basic introduction here. Okay, that is why I started with this basic introduction here. Okay, so got that? So here you are talking about the gene. So gene, whenever we say gene, we always say it is the unit of inheritance. Right, it is the unit of inheritance and it is in the paired form. It is actually the pair of alleles. That's what you have to remember. So it is actually what? It is actually the pair of alleles. Got it? Got it? I'll explain the alleles as well. Wait. So Because Mendel, he gave the term factors. So let me write it here. So what are factors? Just like gene, obviously this is also the unit of inheritance. What is it? It is also the unit of inheritance. Is that clear, everybody? Yes, please. Is that clear? Sure? Sure? So it is also what? It is also the unit of inheritance factor, right? Or unit of heredity, I would say. Unit of heredity. Okay, unit of heredity. And inheritance is considered as what? Inheritance is considered as the basis of heredity. So do not get confused here. So it is the unit of heredity which is responsible for a particular character, right? Which is responsible for a particular character. So it is the unit of heredity which is responsible for inheritance and appearance of a character. Inheritance and appearance of a character. This is what the meaning of factors is. So Johnson named it as gene. Is that clear? So Johnson named it as gene. Is that clear? Is that clear? Yes? Huh. Exactly, Pooja. It's same. So that's what I said, that Johnson named it as gene. Now I'll explain you what. So gene or the factor, what are they? So it's a pair of alleles. Now you know why do we call it as a pair. So allele or allelomorphs, that's what you people can say, right? Allele or allelomorphs. So pair of alleles or allelomorphs. Is that clear? Pair of alleles or allelomorphs. So what exactly is the allele? So now when you talk about a gene, right? So students, with the time in that gene, mutation will also occur, no? Mutation will also occur. Yes or no? In this gene, mutation will also occur. So because of mutation, its alternative forms will also form. Okay? Because of mutation, its alternative forms, they will also form. So because its alternative forms, they will also form, right? So that alternative forms of a gene, what do we call it? We call it as the allele. Now I'll explain it, right? I'll explain it in detail. Okay, but firstly tell me, factor word clear? Factor word clear? Everybody. So factors were referred as genes by Johnson. Is that clear? Factors were referred as genes by Johnson. Done? Right? And what are alleles? Alleles are basically the alternative form of the genes that forms due to the mutation. And you know that it is the mutation, it is the recombination that results in variation. So mutation is a very important force, right, which causes the variation. So because of these mutation, with time what happened? These genes, right, they get changed, they get modified. So that alternative forms, right, that alternative forms are actually the alleles. Is that clear? So before going forward or before writing that part, so you understand this thing. So factors clear. So if there is a factor, which is dominant. So for dominant factor or dominant allele, right, it is represented by, how will it be represented? It is represented by capital letter, right? Whenever a factor is dominant, it is represented by capital letter like A, T, etc. Right? And there are recessive factors as well. I'll tell you what are they. Recessive factors as well. They are represented by small letters. How are they represented? They are represented by small letters. It can be capital A, small t, etc. So let me explain this to you, right? Let me explain this to you. I'll give you the example of the, I'll give you the example here and then we'll discuss it, okay? I'll give you the example here. So students, let's suppose, I just read that genes or the factors, they are responsible for inheritance and appearance of a character. Like this is the thing that I have mentioned, that is character, right? Character. What exactly is a character? Yes, anybody? Anybody? What exactly is a character? Anybody? What do you understand with the word character? Anybody? Yes, I really want to see some. Answers. Yes, please. What do you understand with the word character here? Try to elaborate it. Try to give the example. They say, I just said that gene is responsible for the inheritance and the appearance of a character. So my question here is that what exactly is a character? What exactly is a character? Students, character is I would say an expression, right? Character is what? Character is a kind of feature, a heritable feature or kind of an expression. For an example, like you compare two individuals, right? We comment now that that person is tall, that person is comparatively short, or that person is having black colored eyes or blue colored eyes or green colored eyes, hair curly, straight. What are these things? They are the characters basically. That this is actually that expression. This is actually that heritable feature, or I would say these are the general features that we see. This is a character, right? This is a character, isn't it? This is a character. So a gene is responsible for that character, number one. A gene is responsible for that character, number one. Firstly, tell me this point clear. A gene is responsible for a character. So what exactly is a character? Character is a heritable form or it is a general feature. So I have given you the example, like we say somebody is tall. You are talking about the height there. So height is a character. Height is a character. You can say eye color is a character, right? Hair color, hair type is a character. This is what I'm talking. But now when you people talk about the height, let's take the example of a plant here. You talk, this is what we say, that a plant is tall or dwarf. Let's say, right, that the plant is tall or the dwarf. This is what we say now, that plant is either it is tall or dwarf. So this tallness and dwarfness, what exactly are they? This tallness and dwarfness, they are basically what? So here actually this tallness and dwarfness is basically the character only. We are talking about the character, but here we are talking about the alternative or I would say contrasting form of the character. Right? Let me simplify this. So a gene is responsible for controlling a character. Which character I have picked? I have picked up the height. Right? That let's talk about the height. So we know that height is also having the contrasting forms, isn't it? Height is also having the contrasting forms. Like tallness is also a form of height. Dwarfness is also a form of height. So character, you know what is it, what it is, and then there is a word called trait. Right? So trait is the contrasting form of a character. Like let's suppose I'm talking about the eye color, blue or green. Okay. So eye color, it is the character. Blue colored eye, trait. Green colored eye, trait. This is what I'm trying to tell you. Okay. So gene is responsible for controlling a character. So we know that character is also having the alternative form. So how is it possible? Because gene is also having the alternative forms now. Gene is also having the alternative forms, and how that alternative forms, they form due to mutation. So let's suppose height is a character. So tallness is a dominant trait, or dwarfness is a recessive. Now what those who are studying it for the first time, so let me tell you, but I believe majority of students, they know it already. When we talk about the dominant, so dominant which expresses itself, right? Like let's suppose you are observing a population. In that population, you are saying out of 100 people, 90 people are tall, right? Only 10 people are dwarf. So you are saying that 90 people, they are tall people. So tallness is a dominant trait because it is expressing itself more, right? So that dwarfness will be the recessive. So dominant trait is the trait which expresses or which we notice more in the population in comparison to the another one. So the one which is less will be the recessive, right? The one which is less, it is going to be the recessive. So this part clear, everybody? This part clear? Sure. So the one which will express less in that population, that will be the recessive. So I told you that height is a character, or obviously character is controlled by a gene. So the alternative forms of this height, right? So the alternative forms of this height, like tallness and dwarfness, we are using the word trait for it. What is the word that we are using for it, students? We are using the word trait for it. So, because gene is also having the alternative forms that we are calling as alleles or allelomorphs, right? So, obviously, they are the one which are responsible for the different traits. Is that clear? So, like gene, right? So, in simple words, gene is a pair of alleles. Gene is a pair of alleles. Okay, so I can explain it in this way, like height is a character. It is having two alternative forms, tallness and dwarfness. So height is controlled by a gene. This tallness, for this tallness there is an allele, and for dwarfness is also there is another allele from this. Is that clear? So these basics clear? Yes, please. Tell me, these basics clear? If these basics are clear to you, then I'll write all the points here. So now you got it that why do we say that dominant factor or the dominant allele, it is represented by capital letter. General rules there in class 10th also you study, and you have the recessive factor after this. Right, what you have after this, students? You have the recessive factors after this. Is that clear? So now write about the alleles or the allelomorphs. So these are the alternative forms of a gene. Yes, please. It is the alternative form of a gene which are located on same position, which are located on same position. Yes, please. Which are located on same position on the homologous chromosome, right? On the homologous chromosome. Am I clear? So it is the alternative form of a gene which are located on the same chromosome on the homologous chromosome. So that is why initially I specified what is the homologous chromosome here. Is that clear? Is that clear? So next part is each gene. Yes. So what exactly is the locus? So each gene occupies a specific position on chromosome called locus. Am I clear? And the plural will be loci. Understood? The plural is going to be loci. So each gene occupies a specific position. Okay. So now the another terminology that you have to understand here. So let's suppose now if an individual is homozygous. So what is the meaning of homozygous here? So when you are saying homozygous, homo means same, right? Like when same alleles in a gene, right? Like for an example, if it is, see, both are capital. So basically both are capital means they are dominant now. So it is both are capital, like both the alleles are same. So word will be homo. So homozygous dominant. That's what we have to write. Or small t. What's that? It is going to be homozygous recessive. What is it? It is going to be homozygous recessive. And what's the meaning of heterozygous? So heterozygous means when a zygote is having two different alleles. Okay. When two different alleles are present. When two different alleles are present. Right? Or two different unit factors are present. You can call it allele, you can call it a unit factor as well. So let's suppose capital C, small t. So then you will call it as heterozygous dominant. Right? Heterozygous dominant. And always remember, heterozygous is always going to be dominant. Why? Like as per the for the Mendel part, right? We'll discuss the post-Mendelian inheritance as well. So capital T or small t, that's the word that we have. Capital T and the small t. So capital T means dominant allele. So dominant allele or the dominant unit factor is something that will express itself, right? And recessive one will only express itself in the presence of another recessive allele. It cannot it cannot express itself in the presence of dominant. So this is what you have to remember. So any doubt? Any doubt? Right? So homozygous means a zygote which is formed by the fusion of beta two same type of, right? Two same type of alleles, or I would say two same type of identical factors for a gene. Is that clear? Is that clear? Right? Got that? One student is asking now what exactly is the meaning of this homozygous. So homozygous means beta a zygote which is formed by the fusion of the identical unit factors, right, for a particular gene. This is the meaning of that homozygous. Okay, the individual that will form from that homozygous will be homozygote. Understood? So any doubt here? So heterozygous will always be dominant because here, right, when two different alleles are present. So heterozygous means the zygote which is formed by the fusion of two different types of alleles. Okay, so I'm not writing it. I hope this part is clear. So heterozygous is dominant here because if only one dominant allele is present, it is definitely going to give its expression. It doesn't matter the another one is dominant or recessive, but if one dominant allele is present, it will definitely express that trait. Understood? It will definitely express that trait. Is that clear? Is that clear? So next thing that you have to write is, you will write that what exactly is a character or what exactly is a trait. Is that clear? Is that clear? Yes? Okay. So now when you talk about a character, so as I said, character is a general feature. It's a general feature or heritable form, right? Right. So it is what? It's a general feature, right? It's a general feature or heritable feature or heritable form, yeah, attribute of an organism. Of an organism. So for an example, like you can consider height, flower color, pod shape. So this is the character. What's that? It is the character. So it is the general feature, right? It is the general feature or heritable attribute of an organism, right? Right? Is that clear? So trait is what? So trait is a specific expression of a character or in simple words, I can say that it is the variant form of a character. It's a variant form of a character. Just for an example, people, right? You see, height is a character. So if it is tall or dwarf, so they are what? They are the traits. One will be dominant, another one will be recessive. So flower color is a character. So whether it will be violet or it is going to be white, that is a trait. Pod shape, it can be inflated or constricted like this or constricted like this. That's going to be a trait. Is that clear? So the difference clear? So the difference clear? Sure? Done? So this is the meaning of a character or a trait. Any doubt? So it is a variant form of a character, right? Sure? Okay, so the next thing that we have to discuss is phenotype and the genotype. Okay. Next thing that we have to discuss is phenotype and genotype. So do you know anything about it? Yes, please. Do you know anything about it, that what exactly is the phenotype or what exactly is the genotype? So when we use the word phenotype, right, when we trait is simple, height is a character. So alternative form of a character like tallness, what exactly it is? You are talking about the height only. Dwarfness, what exactly it is? It is again the form of height only, right? So that alternative forms of a character, they are known as trait. Just flower color, if you are talking about the color, you are talking about a character. Like you are talking about white color or the violet color or the blue color, you are talking about the trait then. Okay, done? Okay, so now the phenotype and the genotype. So PH, phenotype, from that what you have to pick, you are talking about the physical appearance, right? What exactly you are saying? You are talking about the physical appearance. For an example, there is a, let's suppose there is a plant, okay? You are looking at that plant, you can just simply say that this plant is tall, isn't it? You are talking about its phenotype. You are looking at a plant, by looking at it, you are simply saying that it is tall. So means you are talking about what? Its phenotype, physical appearance. external appearance. But now, when I'm saying that what sort of tall it is it homozygous tall? Is it homozygous tall in which both the alleles they are same, or is it heterozygous tall? Then you are talking about the genotype. Are you getting my point? Then you are talking about the genotype. Is that clear? Is that clear? So it is the external and morphological, right? So here you can see it is what? It is external and morphological. It is external and, right, morphological appearance of an organism for a particular character. For a particular character. This is what phenotype is. And when you talk about the genetic composition, so genetic composition of an organism for a particular character is called genotype. Is that clear? So that's the meaning of phenotype and the genotype as well. Am I clear? So phenotype is the external appearance, genotype is the, right, genetic composition, and obviously phenotype is the, so what exactly is the phenotype? See, so phenotype is the result of genotype plus environmental factors. So we cannot simply say that it is just the genotype only, it is just the genes only. Nope, it's not just the genes. Along with that genes, environment also influence the phenotype. Is that clear? Right? Is that clear? So it is the result of genotype plus environmental factors or environment. So it is the result of interaction in these two. Okay? So in the case of phenotype, we'll see genotype plus environment. This environment. So these two, their interaction will tell me about the phenotype. Am I clear? Their interactions will tell me about the phenotype. So you cannot say, see, if I'm tall, so obviously that is because of my genes, right? That is because of my genetic composition, my genetic constitution, my genetic composition. So obviously phenotype will always be the result of genotype, right? Phenotype will always be the result of genotype, but you cannot say that it is simply the genotype, right? Along with the genotype, you will also discuss, right, along with the genotype, we will also discuss that there will be the effect of environment as well. Environment plays the role. Okay, environment plays the role. Is that clear? So this is all. Okay, so these two, their interaction will decide the phenotype. Okay, their interaction will decide the phenotype. Understood? Yes, beta, we will discuss the questions also. But firstly, let's focus on the basics. If your basics are done, then everything will be clear. Okay? Okay, so that's what you have to remember. So what we discussed so far, we started with the basics here. We talked about the genetics. Term is given by William Whitson. So genetics means heredity plus variation. Heredity means transmission of characters from parents to progeny. Variation means what is the variation? The degree by which parents they differ from their progeny. And then inheritance, it is the process. Is that clear? None? So inheritance is the basis of heredity. Again, NCERT. Nothing else. This is all NCERT. So basics of this chromosome and all, factors we have covered, dominant, recessive, phenotype, genotype, and you know there is one more thing which is known as phenocopy. Do you know that? There is one more term which is known as phenocopy. So phenocopy is exactly what? Like phenocopy. Obviously, I'm somewhere talking about the phenotype. I'm somewhere talking about the external appearance. Sometimes what happens, like when we see an expression for a particular character, we notice that it differs or it resembles. Like let's suppose there is a particular expression, but that resembles, right, with some other expression, and it feels like it occurs due to mutation. but actually it is not. I'll give you the example, then you'll be able to understand in a better way. Just if I take the example of Himalayan, Himalayan rabbits, in their case, what's going to happen? Just let's suppose there is a rabbit, okay? So that is having a brown fur color. But if you are, right, brown fur color, and when you will, when this rabbit, when it will develop in a very low temperature, right? Let's suppose if, right? Then in that case, the fur color will become white. So temperature has played the role there. It is not the genetic mutation that has occurred, no. Right, we are not discussing about any of the genetic mutation here. There is no such thing. There is no genetic mutation. But exactly what is there? What is there? It is the role of environment. Am I clear? It is the role of environment. Is that clear? So it's a non-genetic, right? It's a non-genetic, but it is something which is produced by the environment. Okay? It is not because of the mutation here. Done? Okay. Any doubt? Any doubt? Right? So it's a so if I have to define it, so it's a non-heritable phenotype produced due to environmental factors but mimics a phenotype that appears to be a genetic mutation. So it feels like that there is a genetic mutation, but actually it is not. It is just the environment. Am I clear? It is just the environment. Understood? It is just the environment. So it mimics a genetically determined trait, right? Okay? Genetically determined trait. Okay? Done? Any doubt? So it mimics a genetically determined trait without any mutation. So there is no mutation here that has changed the phenotype. It is just the environmental factor. So these are the basic terms, right, students? These are the basic terms. Okay. So now let's talk about some points of the Mendel as well. Okay. So let's discuss that. So I think up to this part everything is clear, है ना? I believe up to this part everything is clear. So if there is any doubt, do let me know. So basics about the Mendel I told you already, right? He joined a monastery, Augustinian monastery. Then there, right, he studied natural sciences, as well as mathematics as well. But overall, you know that basically, see, if you want to one should have the keen interest, the observation ability, then obviously you can figure out such things, right? Mendel was such type of person. He wanted to be, or he tried to be a teacher as well, right? So when it comes to the Mendel, so he is believed to be he is actually the pioneer of the classical genetics, right? So basically in October 1843, right? In October 1843, he was admitted to the Augustinian monastery, right? And there he got the name Grieger as well. Right, earlier it was not Grieger John Mendel. There he got the name Grieger as well. So I'll just pick up the important points here only, right? Because this history part is not much required for us. So Mendel began experimental crossing of pea variety in which year? 1856. Is it important? Yeah, it is given in our book. And one more thing I would like to specify here. See, this is the historical experiment of the Mendel, right? And he conducted that experiment on pea plant varieties. It is actually what? It is actually the garden pea, right? It is actually what? It is actually the garden pea. And what is the scientific name of this? So it is the Pisum sativum. What's the scientific name, students? It is the Pisum sativum. And here 2n is equals to 14, or I would say 7 pairs of chromosome. So what am I trying to tell you? So no doubt it was the historical experiment of Mendel, but it's not like that that he just experimented on pea plants. No, he conducted experiments on other plants as well, but this one is believed to be that historical one. Is that clear? Is that clear? Understood? Yes, right? So I am not talking about that history. As I said, he studied natural sciences as well as the mathematics, and that helped him a lot. That is what we are going to see now. But overall, I have just picked up the important points here only. Okay? I have just picked up the important points. I'll just pick up the important points here only. So Mendel began experimental crossing of pea variety in 1856, and his experiment continued till 1863. So once there was a question, right, it is a PYQ that Mendel experimented on pea plant for seven years. For how many years? For 7 years. That's a PYQ, and yeah, you should know. So it is 1856 to 1863. You have to remember this. Okay. Then his research, he published the paper, right, Experiments in Plant Hybridization. So it was published in 1865. Yeah, in some books it is 1866 is also given, right? Some sources say 1865, some sources say 1866. So this is what you can note down. But he conducted the experiment till 1863, given in NCERT. You have to go for it. So as I said, some sources say 1865, some sources say 1866. So he proposed that several basic genetic principles in that paper, but you know that his work remained unattended and unrecognized for quite a long time, or I would say till 1900. His work was unrecognized. Yeah, there are reasons behind, right? There are reasons behind as well. Okay, are you getting my point? So this is what you have to remember. The year, as I said, the year, right, the work published in, as per some sources, 1865 or 1866, and then his work remained unattended or unappreciated, unrecognized for quite a long time, and then 3 scientists, they rediscover his Mendel's work. So basically he got that fame. Now we know him as the father of genetics. So he got that fame after his death. So after his death, so what is the term that we use for it? We call it as posthumously, right? Posthumously he got the recognition, means after his death he got the recognition. So 3 scientists, they rediscovered his work, right? 3 scientists, they rediscovered his work. So here you can see, right, the 3 scientists are this Karl Correns, Karl Correns, Eric von Schemach, and Hugo de Vries. Hugo de Vries we also study in evolution when we talk about the mutations. Okay, so yes, the name of the scientist, again, it is very important. So 3 scientists, they rediscovered the Mendel's work. So imagine they like obviously they were also studying the genetics, and whenever we conduct any research, or so obviously we have to collect all the data, like if in any part of the world, if anybody has done anything in that particular field, or what is that, like we have to collect all the information, right? So they all were experimenting independently, and they found his papers, and they were amazed to see that. So after, I would say, after 16 years of his death, right, after 16 years of his death, his work, right, it came in that notice. So this is what you have to understand. So if you keep on doing the, keep up the good work, right? This is what I want to suggest you from Mendel's story as well. Just keep up the good work. Not today, but someday it will be appreciated, and don't just do it for appreciation only. Yeah, we are humans, we need appreciation, but just keep up the good work. Okay, so sooner or later you will definitely get benefit out of that. Definitely get benefit out of that. But you should not have that intention that I'll do something and I'll get appreciation. No, just do your work. If it is good, definitely you'll get the appreciation. Okay, so Karl Correns, he experimented on maize. Hugo de Vries experimented on evening primrose, which is also known as, scientific name is Oenothera lamarckiana. Okay, Oenothera lamarckiana. What is happening to my spellings now? Lamarckiana, evening primrose. So why? So this part I am definitely specifying because this is again a previous year question. But yeah, we discuss it in evolution part. We'll discuss this there. So Aaron von Schemach. So they rediscovered Mendel's work, right? They rediscovered Mendel's work. Am I clear? They rediscovered Mendel's work. Understood? Understood. So now we will discuss 2 parts. We'll discuss the reasons for the Mendel success as well as the reasons for his failure as well. Then after that we'll start his experiment. But yeah, firstly, initially itself, we will discuss the reasons for his failure as well as for his success. Okay. So actually, what happened is, firstly, I'll start with the failure. Why? Because initially his work didn't get the recognition. So that's why I'll start with the failures. But later on, after his death, posthumously, he got the recognition. So what were the reasons that his work got that success? So let's discuss this. So firstly, I'll start with the reasons that why his work was not recognized until 1900. The number one. At that time, the communication was not that easy. Right now, so we have Instagram, internet, the easy access of internet, everything. But at that time, it was not there. So his work, right, it was not widely publicized. That was the issue. His work was not at all widely publicized. Listen to this, okay? Number one. Number two is, he used natural science, he used biology and mathematics. Nobody was able to imagine that. Nobody was able to understand that. He experimented on pea plant, right? He experimented on pea plant. He applied that statistical knowledge. He applied his mathematical approach. Then he got some results. And see, that was the reason of his success later. But initially, because he was way ahead of his time, people were not able to understand that how somebody can, right? How somebody can mix biology and the mathematics. So they were not ready to accept it. They were not able to accept it. Are you getting my point? They were not able to accept it, that how this can happen. Okay, so that was the reason for his failure as well. Plus, he gave the concept of factors, right? He gave the concept of factors. Yes, he gave the concept of factors. So factors, he said factors are stable, discrete units. Factors are something that are responsible for the characters. So this was a new concept. People were not ready to accept. His contemporaries were not accepting it. Plus, he was not able to provide any physical proof for that factors. Okay, so that's why his work, it didn't get the recognition. So here you can see communication is issue. Communication was not easy. Work could not be widely publicized. Then the concept of factors. He proposed that factors are stable and discrete units, and they are the one that are controlling the traits. Right, they are the one that are controlling the traits. So he also proposed the idea of this pair of alleles or pair of unit factors, and he said that they do not blend with each other. He proposed that ideas, right? So this contradicted the widely accepted blending theory. Right, that time blending theory was there, but he said that factors, right, factors are there, factors are responsible for these characters or the traits, right, and factors are basically in the pair, and they did not blend, right? Right, so it was not acceptable. Are you getting my point? Then his contemporaries did not accept his this explanation for the continuous variation that we see in the nature. In the nature, variation is there, but they were not ready to accept it. Plus, factors are also not showing the blending. Then how is it possible? Right, so again, that was a problem. Then, as I said, use of mathematics. This is like my favorite. Right, later on, this thing, right, later on after his death, because of this mathematics only, he got that phase, because his data was authentic, sampling size was good. But initially, he didn't get the success. Like, if many years back, when people used to say that online we can buy something or we can just do some shopping, nobody was ready to believe that, right? But now you know that it is very easy. Like, now we cannot survive without online shopping. If I talk about myself, right, obviously even I cannot. But like many years back, can you just imagine that? No, of course not. We are studying online, okay? We are studying online. Nobody has ever imagined it. So even Mendel was way ahead his time, right? Even he was way ahead his time. So that is why people, by then, they were not able to understand it. Then no physical proof of factors, as I said. So he studied that factors were discrete units, but he couldn't provide any physical proof for their existence. Moreover, he couldn't explain what they were made of. Is that clear? So that is why his work was not acceptable by them. But what are the reasons for his success? Actually, that mathematics is reason for his success. First of all, unlike his predecessors, okay, he worked on the inheritance of one character at a time. He was not somebody, I'll study all the characters at a time, right? That I'll study all the flower color, flower shape in one plant only and just, no, he was not the one. right? What he did, he studied the inheritance of one character at a time, and due to which he was able to observe the inheritance of that particular character, right? Due to which he was able to observe the inheritance of that particular character, for generations, right? And his sampling size was quite large, right? He applied the mathematical approach, right? He applied that statistical approach, so he was able to get the result. So these are the reasons for his success. He applied the for these qualitative characters, he applied the quantitative approach. He selected the characters, but his large sampling size, large number of plants he studied, or large number of population he noticed, he observed. That's why he got the success. Plus the selection of material also, right? That is also the reason. Are you getting my point? That is also the reason. Jaise during election we see this that this poll and all, that in this particular region, in this particular area, the chances of this party may win, that party may win as per the polls. So how do they calculate it? Obviously it is all the statistical approach. It is all they collect the data, they apply the formula, they apply the statistical approach, and they got the result. So that's how it works. Okay, so when it comes to the reasons for the Mendel success, so as I said, he studied the limited characters at a time. Like, unlike his predecessors, this is the word that I'm using, right? Unlike his predecessors, right? His predecessors, they did what? They studied many characters at a time. But when it comes to the Mendel, he studied limited, or I would say one character at a time, right? Right? So he studied the inheritance of only one or two characters at a time, unlike earlier workers or the predecessors who studied many characters simultaneously. So this reduced the confusion and allowed him to have a correct, clear pattern recognition. Then, as I said, he quantitatively analyzed the inheritance. He quantitatively, you'll study this line in the NCERT. He quantitatively, right? He quantitatively studied the inheritance of the qualitative characters, right? Mendel quantitatively analyzed the inheritance of qualitative character, right? Right, these characters he picked up, he selected them, and quantitatively he analyzed them, right? The statistical records he maintained, okay, and after that he maintained that ratios and all, and moreover, large sampling size, right? Large sampling size. So that was something very, very, very important. Is that clear? Is that clear? Now, selection of material. Garden pea, that is your pea plant, that is your Pisum sativum, right? Having 2n is equals to 14, or I would say 7 pairs of chromosome it contains. See, some people say that Mendel got lucky because even in pea plant there are some other phenomena. We also discuss about the incomplete dominance there, yeah, Pleiotropy there, but that is not the case. See, he worked hard for 7 years. But this is the historical experiment, right? This is something that we know, but he experimented on other plants as well. He got the result there. He observed the things. Okay, so I'm not going to say that he was just lucky. He worked very hard, right? And that's why he was able to get the results. So when you work very, very hard, then this kismet thing, it also favors. So remember this, okay? So why? So he selected the pea plant. It was amazing. Why? Because it is an annual plant. Its life cycle is short. It is around 2 to 3 months, right? Plus it produces large number of offspring, large number of seeds. So you are getting large number of seeds, that too in less time. Obviously it's a win-win situation, number one. Number two, it is having many contrasting traits, right? Like flower color, violet color is there, white color is there, flower position, axile is there, terminal is there, pod color, pod shape, seed color, seed shape, height, alternating characters are there. Then because this pea plant is what? Bisexual. So naturally self-pollination is there, but if required, can also go for the artificial or the cross-pollination, right? And it is very easy to cultivate. So that is why we say the selection of material is also good. So yes, we can even get the question from this part, and it is even a PYQ that out of the following, what are the reasons of the, what are the reasons or why pea plant was better? So the question like that can also come. Is that clear? Okay. Is that clear? Okay. Done? So up to this part all clear? Beta, my energy is quite high, but I think today you are not keeping well. Okay. So if you have joined late, then please rewind the video and then you can just check it out and then you can just understand. Fine? Fine? Okay. So this is for the selection of material. Okay. So now let's talk about his experiment. Let's talk about the characters he selected. Okay. So I don't remember the year right now, but yeah, they have even asked this, that which characters he studied, how many characters were color based, that type of questions were there. So Mendel, see, Mendel studied 7 characters. How many characters, students? 7 characters. So if I am saying 7 characters, right? If I am saying 7 characters, can I say that 14 traits? Like I told you now, pea plant is having the contrasting, right? Contrasting these traits. Okay, so if 7 characters, so that characters will have the contrasting forms as well, right? The variant forms as well. So for 7 characters, can I say that 14 traits? 7 characters, 14 traits? Yes or no? Or we can say 7 pairs of contrasting, 7 pairs of contrasting traits. So either you can write 7 characters, 14 traits. So 7 pairs of, right? 7 pairs of contrasting traits, right? And he selected 14 true breeding pea plant varieties. He selected 14 true breeding pea plant varieties. Why 14? Because 7 characters means 14 traits means 14 plants. But what exactly is the meaning of true breeding? What exactly is the meaning of true breeding? Yes, everybody, quick. What exactly is the meaning of true breeding? When I'm saying true breeding, you can also call them as pure lines. So it simply means that they are homozygous for a trait. Okay. Homozygous for a trait, right? And they are formed by continuous self-pollination. Right? They are homozygous for a trait and they are formed by continuous self-pollination. What does it mean? What does it mean? What does it mean? Yes, everybody. We are saying that pure lines. We are using the term or the word here, that is the pure lines. So what is the meaning of pure lines? Yes, please. What is the meaning of pure lines? So pure lines means, true breeding means they are homozygous for a trait. Like if it is tall, so it has to be like this. If it is dwarf, so dwarf to indeed. So they are the pure lines. Are you getting my point? So how can we get it? By continuous self-pollination. Like you have a tall plant, you are continuously self-pollinating it for many generations. So then this particular character, right, this particular trait, I would say, it will become stable. Is that clear? Right? So pure lines or true breedings, they show the stable trait in inheritance and the expression for several generations. So they show stable trait inheritance for several generations. Are you getting my point? Right? Stable trait inheritance and expression for several generations. Right? Understood? So that is the meaning of pure lines. Okay. So the characters that we discuss here, we even need to know the chromosome number, that is 1457. So characters that he selected, see, stem height, or I can say that stem length, flower color, flower position, seed color, seed shape, pod color, pod shape. These are the 7 characters. Do we need to know about that? Yes. Yes. These are the 7 characters that he studied. So which one is dominant? Which one is recessive? We'll discuss that, right? But before that, see, you should remember that these are the 7 characters, right? These are the 7 characters that he studied. 1, 2, 3, 4, 5, 6, 7. Okay? Right? Like tallness, height. Tallness is the dominant trait. Dwarfness is the recessive. Okay? Got that? Yes. So any doubt? Any doubt? So are you clear? Any doubt? Sure? Are you sure? Done? So flower color, violet, white, exile, terminal. Yes, we need to remember this as well. Yellow, green, round, wrinkled, green, inflated, yellow or constricted. That's what you have. Understood? That's what you have. So dominant and recessive, right? It is the dominant and recessive. So students, they get confused in pod color and seed color part. Seed is yellow, which is dominant, green, which is recessive. Pod color is green, which is dominant, yellow is recessive. Is that clear? Yellow is recessive. Is that clear? Sure? So write down. Quick. Quick, everybody. So that's a question actually. In Mendel's selected characters, in pea plant, how many are color based? So how many characters are color based? How many characters are color based? Yes, people. How many characters are color based, everybody? 3. Seed color, flower color, pod color. So the answer is going to be 3. Okay. So again I'm telling you, pod color, green is dominant and yellow is recessive. Yellow is dominant, green is recessive. Am I clear? Sure? Yes, please. Quick. Okay. So yes, you have to remember the chromosome number as well. So stem height, chromosome number 4, flower color, chromosome number 1. So overall, the chromosomes are 1, 4, 5 and 7. So 4 is for stem height, plus for flower position, for pod shape. Then 1 is for the flower color and the seed color. So color 1. Then this 5 will be for the pod color and for 7 it is the seed shape. So you can just write it down. You can just write it down quick. Done? So that is it. So this is the diagram, right? So here itself you guys can mention the chromosome number as well, okay? Here itself you can mention the chromosome number as well, okay? So stem height, yes please, which chromosome? Stem height, chromosome number 4, chromosome number 4. So then comes the flower color. So for color I told you it will go for 1, and even for seed color it will go for 1. But what for pod color? So pod color will be 5. And what about pod shape? So pod shape will go for again 4. Flower position, so flower position will be 4, and then seed shape is 7. This is the chromosome number, okay? So here you have the dominant and recessive trait that we have already completed. Now we'll talk about the technique, right? The technique that Mendel has used. See students, look at that diagram and then let's see this part. So he applied the artificial hybridization technique here. How come it is possible? It is just like, I always give this example, it is just like the forced arranged marriage, right? Like let's suppose a girl likes a boy, the family is like eliminate that boy, then they just lock the girl, right? And then they find a suitable groom as per them, and then they are like, you just got married. It is like that. Ma'am, what are you eating? Matlab, you have problem with what I'm eating? I'm just eating fruits here. Even I am hungry. Wow. So rather than focusing there, na, you better focus here. Okay? So artificial hybridization. So how are we going to start? So pea plant. So in the case of pea plant, the flower. What's that flower? That is bisexual. I'll use the symbol here. So because we want to go for the cross pollination, so here there will be the removal of anther in immature stage, removal of anther in immature stage to avoid self-pollination. So firstly, what will be done? The plants will be selected, they are bisexual. So in the immature stage itself, what will be done? The removal of anther will be there. And what do we call it, students? We call it as emasculation. See, a repeated previous year question. So that's the beauty of NCERT. You are not allowed to leave even a diagram. Okay, then emasculation. Then after that, and only in the case of bisexual flower only, you will go for the emasculation. Otherwise, there is no need, right? So then what will happen? The emasculated flowers, they will be covered with a bag to avoid paper bag, to avoid any undesired cross pollination. So it's bagging. So this is basically the bagging. Okay, what's that? This is basically bagging. And after this bagging, students, tagging will be done. You know, all that number and all, like this plant, this is how it will be placed. Then desired pollen grains, they'll be collected and they'll be spread over this emasculated flower. And you know that later on what's going to happen. Further, the seed formation will occur further. This is it. So this is the technique that he followed. Am I clear? This is the technique that he followed. Understood? Sure? Sure? Okay, so that's it. So now let's discuss about the monohybrid cross. So I told you now that he was good in this sense that he studied the inheritance of one or the two characters at a time. He was not somebody ki chalo theek hai, let's study all the characters at the same time. No, it was not like that. And that was the reason for his success as well. Okay? Okay? Okay. Done? So monohybrid cross. So see, in monohybrid cross, what is the point or what is the thing here? That here you will see the inheritance of one character at a time, right? Like let's suppose you have you have two plants. All the characters, they are all same. That they just vary. Like one is tall, one is dwarf. It's like that. Okay. One is tall, one is dwarf. Is that clear? Is that clear? Are you getting my point? Is that clear? So what is this monohybrid cross? So it is the inheritance of one character at a time. It is the inheritance of one character at a time. Okay, so let's say firstly we'll take the parent generation. It's a very simple cross. See, we are talking about the Mendel's cross. So he took pure lines. Like see, here you have homozygous dominant, homozygous recessive, right? And always remember, so this is homozygous dominant, homozygous recessive. So F1, what is this F1? It is the first filial generation. It is the first filial generation. So here you will see that there will be heterozygous dominant. Okay, there will be heterozygous dominant. Like these are the crosses that he made, he studied, he draw some conclusion, and on the basis of that conclusion, he provided us the basic laws of inheritance. Is that clear? Is that clear? Yes. Right? Is that clear? Done? So, inheritance of one character at a time, right? Inheritance of one character at a time in a cross is called the monohybrid cross. Then parent generation, we are saying we'll go for the homozygous dominant, homozygous tall, right? Homozygous dominant, homozygous tall. This one is homozygous recessive, homozygous dwarf, and here you have the heterozygous dominant, right? Here you have the heterozygous dominant. So this is the F1. This is the F1, that is the first filial generation. So this is the heterozygous dominant. So before going forward, right, we'll write about it as well. Before going forward, I'll tell you about one more thing, that is the Punnett square. There is one thing, this is a Punnett square. It's a kind of checkerboard, right? It's a what? It's a kind of checkerboard. What exactly is this Punnett square? Students, this Punnett square is given by Reginald Crundle Punnett. Who gave it? Reginald Crundle Punnett. That's a PYQ, that who exactly gave the Punnett square. So it's a kind of checkerboard. Here what will you do? Just let's suppose there will be a square. Okay. And it will tell us, right? There will be a square. It is going to tell us about the, like the zygotes, right? The probability of that part there. Okay. So, like for an example, if I have to define it. So for an example, so it is basically, it tells us about the generations, right? In the form of symbols or squares, like we can just check that generations. For an example, we have one parent as capital T, another parent as small t. First of all, this parent, we'll talk about the gamete formation here. Because it's a pair of alleles, both are dominant, both are same rather. So same type of gametes will form. So this parent is going to form gametes, capital T. This parent will form small t. So in a checkerboard, what will you do? One side you will write the male gametes, another side you will write the female gametes. Male gametes horizontally, female gametes vertically. Male gametes horizontally, female gametes vertically, right? So capital T, let's suppose this side, small t, this side, just a minute. Okay, small t, this side. So capital T, small t, capital T, small t, capital T, small t, capital T, small t. So here you have the gametes from one parent, gametes from another parent. You are crossing it, you are getting it. So you can tell that next generation will be heterozygous dominant. For F1, we generally do, like it's not like that you cannot draw it, we can. So that is how we can check, right? It is basically the representation of generations, right? It is basically the representation of that generation that we can analyze in the form of these symbols or the symbols of squares. Is that clear? So it is a checkerboard. But why are we not making it for the F1 here? Because it is obvious. See, it is obvious. If It is not the incomplete dominance or codominance, which we will discuss. See, when homozygous dominant parent cross with homozygous recessive parent, homozygous recessive parent, the F1 will be heterozygous. Remember this, right? Because see, this parent is having dominant allele, right? Both are dominant, both are recessive. Obviously, it will cross capital T and small t. And whenever there is the presence of single dominant allele, it is going to express itself here. So, homozygous dominant parent cross with homozygous recessive parent, the F1 will be heterozygous. So, this is what Mendel did, but he checked the F2 generation. So, for F2, what you have to do? For checking out the F2, you have to do the selfing of F1 individuals. Means from this parent generation, you got the F1 individual, first filial generation. Now you are picking up the individuals here. You are going to do their selfing, right? You are going to do their selfing. Is that clear? You are going to do that selfing, right, students? So you are going to do that selfing. So selfing mein what will we have? Capital T, small t, you will cross it with capital T and small t. Now what's the point? See, this, just look at this part. Here you have two different gametes. Here also you have two different gametes. So this side you will write capital T, small t. This side you will write capital T, small t, right? And here you can see capital T, capital T, capital T, small t, capital T, small t, small t, small t. So capital T means tall. Matlab phenotype will be tall, here also tall, here also tall. It doesn't matter. Like I'm talking about the Mendel, I'm talking about the Mendelian inheritance, right? So this is what we are discussing. So just focus here, right? See, even if there are two dominant alleles, yeah, one dominant allele, the expression is going to be same, tallness, right? Tallness. And here it will be dwarf. So when you look at it, you'll see the phenotypic ratio, phenotype like physical appearance, 3:1. So 3 tall, 1 dwarf. Then genotypic ratio. Here you are talking about the genotype. So genotypic ratio is 1:2:1. So 1 is homozygous tall, 2 are heterozygous tall, and 1 is homozygous dwarf. So that's what he got in F2. So in F1, only the expression of dominant trait was there, but in F2, along with dominant, recessive was also there, right? Right, and why he conducted the F2? Because if there will be any recessive allele, it is going to express, recessive trait, it is going to express itself in the F2. Got it? Got it? Yes? Yes? Just allow me 1 minute. Done? Okay. Mr. Gaming with Time, stay here for some time, you'll get to know about it. So this is the monohybrid cross, okay? And I told you about this Punnett square as well. So it was developed by Reginald C. Crundall Punnett. So British geneticist. So this is a previous year question. This is a previous year question. Got that? This is a previous year question. So as I said, it's a checkerboard method. It's a graphical representation to calculate the probability of all the possible genotypes of offsprings in a genetic cross. Okay? So it is the graphical presentation, and moreover, as I said, it is a graphical presentation, and moreover, as I said, right? So here the squares, the symbols are used. So the possible gametes are written on two sides, usually the top row and the left column. All possible combinations are represented in boxes below in the square, which generates a square output form. So write down, so male gamete horizontally, female gamete vertically. That's what you have to remember. Do it quick. Write down quick. Okay. So, should I move to the next slide? Because on the basis of his monohybrid cross, he drew some, observed many things. He gave the postulates, and that postulate helps to give the law, the first law of inheritance, the law of dominance. Just see from the square what he observed, right? He said that factors they occur in pair, right? Factors they occur in pair. Okay, and now when you look at the F1, the first filial generation, when you look at the F1, the first filial generation, you can see the two alleles are different. One is capital, one is small. One is capital, one is small. Can you see that? There are two different alleles, right? These are two different alleles. This is what you people can see. So when two different alleles are there, can you see this tallness, this expression is there? है ना? That tallness part is expressing itself. So Mendel was like that. Firstly, factors occur in pairs. Factors are stable, and in the presence of two dissimilar factors or dissimilar unit factors, one will express, the one that express is the dominant, and the one which is not able to express is the recessive. This is what he concluded. So first thing is, so on the basis of his conclusion, he gave the laws. So let's see the first conclusion, which is also known as the postulate of paired factors. So as per that, characters are controlled by discrete units, the separate units called factors. These points are important, right? This is all NCERT students, right? So characters are controlled by discrete unit called factors. Factors occur in pairs, right? So that is why it is the postulate of paired factors. Okay? Next, this one is the postulate of dominance. So as per that, when two different unit factors are present in a single individual, one will express itself and called as dominant factor. The another one which is not expressing itself is the recessive factor. So this is the law of dominance, and you can also call it as first law of inheritance, right? First law of inheritance. So actually what Mendel did, right? So law of dominance, how can we explain this law of dominance? So this law of dominance can be explained in this way. So you can just add this first plus second conclusion. So both the conclusions, right, they gave this law of dominance. So if I have to explain this law of dominance, so I will say that characters are controlled by discrete unit called factors. Factors occur in pairs, and in dissimilar pair of factors, one factor will express itself and the another will be the one which is expressing is dominant, and the one which is not expressing is the recessive. So this is the law of dominance. Anybody having any doubt here? Yes, please. Anybody having any doubt here? Anybody having any doubt here? Quick. Okay, so this is about the law of dominance. So now again this is your NCERT, right students? This is again your NCERT. Just have a look. This is very important. So the law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross, right? See, you just check, you open the NCERT, you check the title law of dominance, 3 points are given. Below that 3 points, you are going to find a paragraph. This is that paragraph that we are discussing. Below that, you will see the another heading where law of segregation is written. So we are right now talking about the law of dominance, right? You can even see there the test cross part is given, right? That the diagram. So law of dominance, the title is law of dominance. 3 points are given that characters are controlled by discrete unit called factors. Factors occur in pair, and in a dissimilar pair of unit factors, the one that expresses itself is dominant and the another one is recessive. Got that? And below that, what will you see? The law of dominance, it is used to explain the expression of only one of the parental character in a monohybrid cross in F1. Because in F1, this is what we are getting. And because law of dominance is saying whenever there are two different alleles, so the one that will express itself is called the dominant. So obviously dominant is going to express itself. So that is what this law of dominance is explaining, and expression of both in the F2. Later on what happened? Both the traits, they expressed in the F2. So that is also explained by this law of dominance, right? In F2 you know that small t, because factors, they are the discrete units. So small t is together, so we were able to see the recessive trait. So this law of dominance explains the proportion of 3:1 obtained at F2. So question can be, which law explains the ratio 3:1 in F2? What should be the answer? What should be the answer? Law of dominance. Law of dominance. Write down. Law of dominance. Write down. Guys, do it. Done? Noted? So these are the details you need to know from the NCERT, okay? Okay? Chaliye. So third conclusion is the law of segregation. So, see, what is the meaning of law of segregation? It is the second law of inheritance. It is also the law of purity of gametes. But yeah, before going forward, let me tell you that incomplete dominance, these are two phenomena that we will study under the gene interactions. So incomplete dominance, number 2, codominance. These are two exceptions for the law of dominance. Like if there is incomplete dominance, if there is codominance, so it means there will be no law of dominance, means law of dominance is not fulfilled. Got that? Got that? Sure? Okay. So which law explains the ratio 3:1 in F2? Law of dominance. And whenever there is incomplete dominance and codominance, then the law of dominance is not followed. Now, law of segregation or the law of purity of gametes. So students, at the time of gamete formation, you know that even in our case also, right? So at the time of gamete formation, I hope you know that in our case also. So you know that our diploid state is having 2n is equals to 46 and our gametes are having n is equals to 23. So the number of chromosome will be reduced to half. We know it very well. So even so here, of course, later on we'll join this gamete part, the chromosome and the gene will relate it. But now at the time of gamete formation also, at the time of gamete formation, each allele at the time of gamete formation from that pair of alleles, that the pair of allele gets separated, and then each allele pass to each gamete. This is in a very simple language I'm writing. Then we'll discuss the technical language as well, the NCERT one, of course. So law of segregation is saying that at the time of gamete formation, from that pair of alleles, right? At the time of gamete formation, from that pair of alleles, both that alleles will get separated and each allele will pass to each different gamete. Like, what am I trying to tell you? That as per the law of segregation, same alleles, right? Yes, alleles for a same character, right? I'm just giving you the bullet points here. You understand it first. Alleles for a same character will not pass to same gamete, they'll get separated. Are you getting my point? Are you getting my point? Like, for an example, you have capital T, small t. So capital T will go to a different gamete. Small t will go to a different gamete. Like, let's name this gamete as A. Let's name that gamete as B. So if the individual is having capital T or small t, then the gamete A will either get capital T or small t, but it will get only one allele. So let's suppose it will get capital T. This one will get small t, right? Or, so it will never be that you have gamete A and that gamete A is having both capital T, capital T, small t. This is not going to happen. That's what I'm trying to tell you. Are you getting my point? That's what I'm trying to tell you. Okay, guys, see, technical glitch was there. My class started around 6. So now you are expecting a break at 7:30 only. Not possible. Let's finish these laws, right? Let's finish this Mendelian inheritance at least. Then we can have the break. Fair enough? Then we can have the break. Okay? Sure? Then definitely we will have the break. Okay, the chapter is lengthy. I don't want to compromise. I literally don't want to compromise the content. Okay, so that's why I'm telling you. Done? So at the time of gamete formation, the pair of allele get, right, from that pair of allele, both the alleles will get separated and each gamete will receive only one allele for a particular character. So alleles for a same character will not pass to the same gamete. Okay, now let's suppose here itself, you can take the example of capital R or capital R. So if it is having capital T, it can have capital R. Here also capital R will pass, but it will never be that this is a gamete. No, this is not going to happen. So this is the law of segregation, right? This is the law of segregation. Now when you look at this law of segregation, right? Now when you look at this law of segregation, so like let's suppose you are talking about the gametes, right? So let's suppose you have gene A. Let's consider it in this way only. Let's suppose we are talking about these 4 genes. So what is the possibility? A, B, C, D can be together. B, capital C, small d can be together. Like, this is the possibility. But you cannot say that, right? You cannot say that ki capital A, small a, capital B, then small d, capital D, or capital A, right? Capital A, capital B, small b, a capital C, small c, they'll stay together in a gamete. No, not at all. Right? No, not at all. This is what I'm trying to specify. So gamete, right? So gametes are pure for a trait, right? Gametes are pure for a trait. gametes are pure for a trait, right? They will not have two alleles for a single trait. No, they are not going to have. They'll get only one allele for a particular character, right? They will get only one allele of that particular character. So they are considered as the pure for that character. Is that clear? So this is the law of segregation, that in the case of hybrids, when you see that hybrids, right, you will see that in the hybrid varieties, let's forget about the hybrid. In simple words, you will see that when there is the gamete formation, right, each gamete will receive only one allele for a character. That's why this law is also known as the law of purity of gametes. So if you ask me about the exception here, so basically if you ask me about the exception here, this law is the universal law, okay? But if there is non-disjunction means if there is failure of separation of chromosomes or chromatid at the time of division, then definitely there is the possibility that two same alleles can pass to the same gamete. But that is a very, 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 very rare situation. Is that clear? Is that clear? Is that clear? So we have the third conclusion here, right? And this third conclusion will give you law of segregation. So from the monohybrid cross, right, the law of dominance, it was explained. The law of segregation was also explained, right? The law of dominance was also explained. The law of segregation was also explained. So you people can see this, this law is based on the fact that alleles, again, this is the NCERT, check the title, law of segregation, under that you will find it. So this law is based on the fact that alleles do not show any blending. Alleles do not show any blending, and that both the characters are recovered as such in F2. That's what we have studied in the monohybrid cross, 3:1. One is showing me the dwarfness. So this law is based on the fact that alleles, they do not show any blending, right? They do not show any blending, and that both the characters are recovered, right? And that both the characters are recovered as such in F2 generation, though one of these is not seen in the F1. So what is this law saying? It is saying that, okay, if there are like alleles, they are not going to show the blending, and if there is a character which was not seen in F1, we can still see it in the F2. That is explained here. Are you getting my point? Are you getting my point? So alleles do not show any blending, and that both the characters are recovered as such in the F2, though one of these was not seen at the F1 stage. So the parents contain two alleles during gamete formation. The factors, you can call them as unit factors as well, or the alleles as well. You can even use the term unit factors or alleles, right? Of a pair, they segregate. So what is the meaning of segregate, students? It means separate. They separate from each other such that a gamete receives only one of these two factors, right? Gamete will receive only one. So see, in simple language, at the time of gamete formation, the pair of allele gets separated, and then each allele passes to each gamete. Okay, so alleles for a same character will not pass to the same gamete, and gametes are pure for a trait. Okay, right? So here we can see gametes are pure for a character or a trait, right? We can see the character here, right? So that's why it is also the law of purity of gametes. It is also the law of purity of gametes. So I believe this part is clear, right? It is also the law of purity of gametes. Am I clear? Right? So if a parent is homozygous parent means same alleles for a character, right? Like same allele for a character. Let's suppose capital T or capital T. So that parent is going to form the same type of gamete. But if there is a, right? So gametes are going to be similar there. If there is a heterozygous parent, so heterozygous parent, you can take this example, capital T or small t. So you will get different types of gametes. Am I right? You will get two kinds of gametes here, each having one allele with equal proportion, right? Right? So let's suppose if you are getting from one cell, you are getting 4 gametes, right? 4 haploid gametes. So 2 will have capital T, 2 will have small t in heterozygous individual. Here all will have, right? Here all will have the same gametes. Understood? This part is clear. So if there is any doubt, please feel free to ask. Yes, please. After that, we will discuss the dihybrid cross. So if there is any doubt, please feel free to ask. Done? Because if you don't have doubt, then I'll pick up that NCERT portion and we are going to discuss that. Do it quickly. You can write down, you can even underline it in your NCERT. Quick. Right? So just take 2 minutes, not more than that. Just take 2 minutes and recall everything. Okay? And you are forgetting one thing. You people are not subscribing our channel and you people are not hitting the like button, people. What's going on? Hit the like button and subscribe to our channel if you are new to our channel. So you take 2 minutes, and just revise it quickly. Okay? So I believe it's done, right? Okay, so next is this. See, in the NCERT, this expression part is given that from the Punnett square, it is clearly easily seen that 1/4 of the random fertilization lead to TT, 1/2 lead to capital T small t and this. And then they are saying about the phenotype, 3:4 will give you the 3/2, 4 will give you the tall and the dwarf. So this ratio part is given. This is a binomial expression that they have given. So see, the 1/4, see, we have the ratio 1:2:1. So how many total number of zygotes are there? 4 na? So 1/4, that is how you can check this probability. 2/4, which will be 1/2 only, and 1/4. So they are saying 1/4, 1/2, 1/4. The ratio of capital T, capital T, capital T, small t, small t, small t is mathematically condensable in the form of this ratio actually. Like this, from this part the question can come in paper. So this ratio is actually condensed in the form of this binomial expression. Just with normally we write now, students, we know it very well. A plus b square is equals to a square plus b square plus 2ab. So here they are showing ax plus by square. So obviously it will be, right? So you can just expand it like that. So it is the condensable form, it is that expanded one. So 1 by 2 plus 1 by 2 whole square, right? So 1 by 2 plus 1 by 2 whole square, see 1 by 2 plus 1 by 2 whole square, 1 by 2 whole square plus 2 into 1 by 2 or 1 by 2. So you will see 1 by 4 plus 1 by 2. So this is your capital T, small t, capital T, small t. So here you have done this. Half of t, half of small, 1 by 2 t plus 1 by 2 small t, then square. And then that is how you are getting the value. So any doubt here? Any doubt here? Sure? Any doubt here? Sure? Okay, so law of segregation, right? I told you about the conclusion. So here you can see the points, beta. So this law is based on the fact that alleles do not show any blending and that both the characters are recovered as such in F2, though they were in F1. I think we have discussed this part already. Okay. So now then, now let's go for the test cross. Dihybrid cross. So beta, we will discuss the dihybrid cross first, the law of independent assortment, then the test cross. Got that? Then the test cross. That is the complete flow. Okay? That is going to be the complete flow. Understood? Understood? Okay, so let's talk about the dihybrid cross. So now the word is dihybrid. So when I'm talking about the dihybrid, so what are we doing? We are here discussing the inheritance of two characters or two genes at a time. Right? Inheritance of two characters at a time. So what are the characters that we are going to pick here, students? So the characters are round. The characters are number 1, seed shape, number 2, seed color. So this is what we are going to pick, right? Seed shape, seed color. Is that clear? So seed shape, one is round, which is dominant. So I'm denoting it with capital R. Another is wrinkled seed shape, which is basically small r, recessive. Seed color, it is going to be yellow. So yellow is going to be dominant. And there is going to be green, and green is going to be recessive. Is that clear? Is that clear? So these are the two characters. Like I am going to discuss this inheritance of two characters at a time. So if the character is this seed shape and seed color, this is what we are going to select. So when you are talking about the seed shape, right, you know the rules here. Firstly, what Mendel did? Parent. So capital R, capital R, capital Y. So it means homozygous dominant, right? That is round seed shape or yellow seed color. Round seed shape or yellow seed color. So this is homozygous recessive. Are you getting my point? Homozygous recessive. So this is wrinkled green. Got that? So we are going to cross them. So now when you cross a homozygous dominant with homozygous recessive, what do we get? When you cross homozygous dominant with homozygous recessive, what do you get? What do you get? Homozygous dominant with homozygous recessive. Guys, show some energy. Right? Show some energy. Be the part of the discussion here. Right? This chapter is important, and trust me, we are learning it in a very simple and a very amazing way. Right? After this class, do read the NCERT. You are going to find it very easy, because wherever you are going to find the complexity in the NCERT, I'll discuss it in the live class. Okay? We'll have the MCQ practice as well, but I want the session to be interactive. Good. So keep on reverting. So F1 will definitely be RRYY, that is heterozygous dominant, Am I right? It is going to be RRYY. So that simply means heterozygous dominant. And heterozygous dominant means, again, here it is going to be round and yellow. Round seed shape and yellow seed color. Am I right? Round seed shape and yellow seed color. Am I right? Now what will we do? There will be, we'll go for the F2. So for F2, the selfing has to be done. The selfing has to be done. Now students, here you have two characters at a time. Heterozygous they are. So obviously you will get the different kinds of gamete. So how can we make the gametes? Right? So for this gamete formation, I'll discuss one thing with you. That is the Falkline method. This is what we are going to discuss here. What are we going to discuss here? Falkline method. Like see, here you have capital R and small r. You are clear from the law of segregation, right? See, you are clear from this law of segregation. Here we have discussed this law of segregation now. I'm writing this again, right? See, for the law of segregation, I told you that law of segregation is universal. Do you remember that? That each gamete will receive only one allele for a character. So law of segregation is what? It is universal law, right? Right? And I told you, if it will not be followed if there is, otherwise it will always be. If there is non-disjunction. So what's that non-disjunction? Yes, please. What is that non-disjunction? So it is the failure of separation of chromatids or chromosomes during division. So in that case it will not be followed, and that case is due to sudden changes, of course, right? So otherwise it will be followed. Like you remember anaphase? You remember anaphase? Let's suppose if it is the anaphase 1, right? Even if it is of mitosis, let's suppose here you have capital T, obviously. So if it will not get separated, like two different cells will not get capital T, one single cell will get 2 times of this. Are you getting my point? So that is what I'm trying to tell you. So from this law, one thing that we can understand is, right, one thing that we can clearly understand is that each gamete will receive only one allele for a character. So that is why here we are writing this capital R and small r separately. This is what we have here. So now capital Y and small y, it can also go with capital Y or small y. So you see the gametes, capital R, capital Y, capital R, small r, capital Y, small r, small y. So some students, they know it, right, in a very easy way, like they practice the question out, their genetics part is good, so they know it. Otherwise, those who are not clear about it, for them, right, I'm just making it. So one parent is making how many gametes? One heterozygous individual is making how many gametes here, students? One heterozygous individual is making total, yes please, one heterozygous individual is making total 4 gametes here. RY, RY, RY or RY. And here RY, RY, sorry, RY, RY or RY. So here you can simply see capital R, what will be the genotype? This, yes. Here capital R, capital Y, small y. So tell me about the genotype, everybody. Yes. Yes, tell me about the genotype here. So here it will be capital R, capital Y, small y. And what about this part? Yes, everybody, what about this what? Capital R, small r, capital Y, capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y. So that's how you can simply write the genotype here, isn't it? You can simply write the genotype here, isn't it? Uh-oh. This is the genotype, isn't it? So look at this part. So this is round yellow. Okay, round yellow. So round yellow. Here also round yellow. Here also round yellow. Can you see that? If you are not able to understand it, just ask me. See this. Can you see that? Yes. And what about this capital R, but there is small y. So what should be the genotype? Yes, Naveen. What should be the genotype? This is capital R and small y. What should be the genotype? Yes? Yes? Exactly. So it will be round wrinkle. It will be round wrinkle. Okay? It will be round and wrinkle. Done. So this one again round. Oh, round wrinkle nahi, round green, my bad. Yes, round green. Round green. Wrinkled yellow. Round green. Again wrinkled yellow. Isn't it? Wrinkled yellow. This one is also wrinkled yellow, and here I have wrinkled green. Yes or no? Yes or no? So students, just let me know the phenotypic ratio here. Just let me know the phenotypic ratio here. Yes, please. It is 9:3:3:1. Am I right? It is 9:3:3:1. Am I right? So 9 here are round yellow. 1 here is wrinkled green. Yes, please. 3 here is round green. Round green. And this one is wrinkled yellow. So yes, that's the possible phenotype here. That's the possible phenotype here. Yes, please. That is the possible phenotype here. So this round yellow, it is parental type. It is one of the parents. This one is also parental type. Right? These two are non-parental type. Like it's... So this phenotype is not like any of the parent, because it is not round yellow or wrinkled green, it is round green, it is wrinkled yellow. So it is the non-parental type, or can we say that it is the recombinant? Yes, please. Can we say that it is the recombinant? Yes, please. Yes, please. So this is the genotypic ratio, right? This is the genotypic ratio, but I will discuss it later. Why? Because I'll teach you a method by which you will be able to get it by yourself. So firstly, tell me this phenotypic and genotypic ratio here. See students, we are about to finish the Mendelian inheritance, right? The incomplete dominance, codominance part we can start after the break. So you guys will get the break, but at 8:30, okay? right? Because of the studio issue, I was not able to start the class at 5. I started the class at 6 or 6:15, I guess. So now, okay, let's have a break at 8:30. I'm telling you clearly. Okay, let's have a break at 8:30. We'll finish this portion first. So round yellow, parental type. These two non-parental type, recombinants they are. That is parental type. This is the genotypic ratio that we have. Now, this is the dihybrid cross, and it helped to explain what? It helped to explain the third law of dominance, law of independent assortment. What exactly is this law? See, in the law of segregation, simply we talked about the pair of alleles, and we said that pair of alleles during gamete formation, from that pair of alleles, each allele will separate and will pass to the two different gametes, right, randomly. Now, when it is the law of independent assortment, we are explaining it with the help of dihybrid cross. So, whenever we see any cross which is having, more than, I would say, which is having more than one character, it is dihybrid or trihybrid, whatever it is. So let's stick to the dihybrid. So in that case, what is happening? You have two different genes, right? Two different genes. This gene is also having its allele. This gene is also having its allele. So this is having two different genes having their alleles. So they have two different genes. Again, I'm repeating. So the segregation, the separation, right here, the segregation of these two different genes, they are not affecting each other's segregation, each other's separation. Like the alleles of that genes, they are randomly segregating. Independently they are segregating. Independently they are meeting in the next generation. Means it is not mandatory that if, just a minute. So it is not mandatory, right, that parent generation, if parent generation is having capital R, capital Y, like both are dominant, so it is not mandatory that next generation will get this only. Capital R can go with small y also, right? And small r can go with capital Y also. So that is what I'm trying to tell you. Are you getting my point? Are you getting my point? That is what I'm trying to tell you. Yes or no? Yes or no? Done? Is it clear? Yes or no? Got that? So that's something you have to keep in your mind. Okay, that is something that you have to keep in your mind, that if you are considering two different characters, if in a hybrid, if you are considering two different characters at a time, so the segregation of one gene or a pair of alleles will not affect the segregation of another. This is the meaning of law of independent assortment, that genes, their alleles are independently assorting. Doesn't matter one parent is having capital R, capital Y, it will stay like that only. Capital R can go with small y also. Small r can go with capital Y also. Small r can go with small y also. So law of segregation tells us that whenever you are talking about a gene, both its alleles will get separated. They'll pass to two different gametes randomly. This is what we discussed. But in this case, what am I trying to specify? I am saying that it doesn't matter that if in parent generation both homozygous dominants were together, so it doesn't matter, they'll stay like that. Capital R can go with if both the alleles were dominant, it doesn't matter, they'll always be like that. Capital R can go with capital Y, but it can also go with small y. Small r can go with small y, but it can also go with the capital Y. So this is the independent assortment. Are you getting my point? This is the independent assortment. Are you getting my point? Yes. So you can see this dihybrid cross even in the NCERT, round yellow, right? F1 generation, then the selfing, and this is that checkerboard here. So from this cross, we explains the law of independent assortment, which states when two pairs of trait, if I'm saying two pairs of a trait, can I say two characters? Definitely I can. So when I'm talking about two characters, when two characters are combined in a hybrid, segregation of one pair of character, right? Like segregation of this RR, yeah, RR, whatever it is, so is independent of the other pair of character. Like capital R cannot influence capital Y cannot influence capital R, so their segregation is random. So this non-parental combination that we have seen now, these non-parental combination, जैसे 9:3:3:1, right? this 9 is to 3 is to 3 is to 1. So this combination that we have studied now, see, these two were the non-parental one. I told you, these two were the non-parental one, right? Just now I told you they are the non-parental one. This one is parental, this one is parental. Hena, this is non-parental one. So why this non-parental one is there? Yeah, how this non-parental one can form? Obviously, this is due to the independent assortment, right? It is because of the independent assortment. It is because these alleles are not affecting each other's segregation, right? It is because these alleles are not affecting each other's segregation. Is that clear? Is that clear? Right? So, for the law of independent assortment also, we have the exception, and that is the linkage. So, if linkage is there, the independent assortment will not be there. But if independent assortment is there, linkage is not there. So, we'll discuss the linkage as well. So, you tell me this part clear? Yes? You tell me this part clear? Are you sure, everybody? Are you sure, everybody? Done? Okay, thank you so much. So let's move to the next part. So this is how, so these are three laws that we have discussed, and I hope it is clear to everybody. So what's the next point? Yes, what's the next point? So now I'll tell you some formulas, okay? I'll tell you some formulas, I'll tell you the test cross, we'll practice few questions, then we'll go ahead. Done? Then we'll go ahead. At 8:30, I'll definitely give you break. Now, I'll teach you something known as product rule. What am I going to teach you? I will teach you something known as product rule. Okay? What exactly is this product rule? See, let's talk about the monohybrid cross. Which cross? Monohybrid cross. So, monohybrid cross, like type of cross. Here, I'm writing its phenotypic ratio. Okay, here I'm writing its genotypic ratio, and here I'll write some other formulas. So you people can see this very clearly. See, monohybrid cross, phenotypic ratio and genotypic ratio. See, in the case of monohybrid cross, you are talking about the inheritance of one character, na? So, phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. Anybody having any doubt? Anybody having any doubt? Yes? Clear? Now, I'll go for the dihybrid cross. Obviously, here, this ratio you are getting when you are crossing to heterozygous individual, right? You got this ratio when you cross to heterozygous individual, right? Now, in the dihybrid cross, it is the inheritance of two characters at a time, and you are following the same case, right? Here also, let's suppose I'm talking about the crossing of two heterozygous individuals only. So, guys, see this part, 2 times I'll multiply this, right? I got the phenotypic ratio this, 2 times I'm multiplying this. So, how can I do that? So, 3 3s are 9, 3 1s are 3, 1 3s are 3, 9:3:1. So right now you counted this, right? We draw that, we discuss that, but this is how you can, by simple multiplication, you can got it. Same here for the genotypic ratio. Just multiply 1:2:1 with 1:2:1. You have to just multiply this. Just a minute. Okay. So let me write it properly. 1:2:1. 1:2:1. So 1 1s are 1, 2 1s are 2, 1 1s are 1, 2 1s are 2, 2 2s are 4, 2 1s are 2, 1 1s are 2 1s are 1. This is what you got. That's how you can check it. You can do the same for the tri-hybrid cross as well, right? Tri-hybrid cross as well. Like here, you'll multiply 3:1 with 3:1 with 3:1. So, already we know this ratio, 9:3:3:1. Am I right? You cross it with 3:1. So that's how you can get it. So this is the simple thing, product rule, right? So no need to cram it. If by any chance if you got such type of question in the paper, you can just simply solve it, you can get it, and then now you can multiply this. So this is the product rule. This will tell you about the ratios. I hope this part is clear, right? I hope this part is clear. Now there are some other formulas. See here, how can you check the number of phenotypic classes? How can you check number of phenotypic classes? Like, see, there are some other formulas as well. So tell me, up to this part all clear? So how many of you are knowing this for the first time? How many of you are knowing this for the first time? So product rule clear? Now, if you are my student, then you definitely know this. So next is number of phenotypic classes. How can you check? The formula is 2 raised to power n. Means, see, n. If there is monohybrid cross, n will be 1. If there is dihybrid cross, n will be 2. So, see, 2 raised to power 1, 2. 2 raised to power 2, 4. So, here also, in the monohybrid cross, we just check. The ratio is 3:1. How many phenotypes were there? 2. In dihybrid cross, we just check the ratios are 9:3:1. So how many phenotypes are there? 4. So that's how we can check it, guys. That's how we can check it. So number of phenotypic classes, this is the formula, 2 raised to power n. And what about number of genotypic classes? Yeah, number of genotype, 3 raised to power n. So for monohybrid cross, it will be 3. For dihybrid cross, it is going to be 9. Right, so see, so this is for monohybrid, this is for dihybrid, right? So you remember in the monohybrid cross what we did, 1:2:1. So 3 genotypes are there. In the dihybrid, the ratio was 1:2:1. So you people can see how many ratios, 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's how you can just apply the formula and you can get it, students. So for number of zygotes or the offsprings, there are two formulas. 4 raised to power n or gametes square. That's the formula for number of zygotes. So number of zygote means the boxes that you make. Right, जैसे monohybrid cross में we make 4 ना. So this is telling us about the probability of the possible genotype of the next generation. And in the genotype, in the dihybrid cross, what was that? It was 4. So total 16. So how can you check it? So see, for monohybrid cross, 4. For dihybrid cross, 4 into 4, 16. If there will be trihybrid cross, it is going to be 64 like this. Like this. Got that? And now how come gamete square? Sorry. So simple. It's in monohybrid cross, right, guys? One parent is making 2 gametes. So 2 square, 4. Here one parent is making 4 gametes. 4 square, 16. One parent will make 8 gametes. So 8 into 8 will be 64. So these are the formulas. Right? These are the formulas. So try to process it, and if there is any doubt, then tell me. Try to process it, and if there is any doubt, then tell me. Done? Quick. Done? Sure? So see, I have added this here, number of phenotype 2. So n is 1. So formula is here. I have even mentioned the formula 2 raised to power n. So number of genotypic class 3 raised to power n. So here you can see the genotypic ratio, right? And then this is the phenotypic category. So the formulas are mentioned here. Done. So next point that we have to discuss is back cross. Okay, next thing that we have to discuss is back cross. So what is this back cross? We have to discuss out cross and test cross, but it is a type of back cross only. So what exactly is the back cross? See, back cross, in back cross here, F1 individual, what is going to happen here, students? F1 individual is crossed with one of its parent. This is the meaning of back cross. What is the meaning of back cross? Yes, please. First time heard these formulas. So it means you have attended my class for the first time, because I have already discussed, I have discussed it many times. Right? I'll discuss this test cross part, then we will have some questions that we have to practice. So tell me, up to this part all clear? So we are talking about the back cross, in which we are saying the F1 individual is crossed with one of its parent. Yeah, any of its parent, that is what I'm saying. Okay. Now, the another part is, so this is the back cross. So F1 individual is crossed with one of its or any of its parent. Now, back cross is having two types. In our syllabus, test cross is given. Test cross is very important, but even you should know about the out cross. I'll tell you why. Now, when you talk about the out cross, here what is happening? In the out cross, F1 individual is crossed with dominant parent. Dominant parent, you know that either will be homozygous or heterozygous, right? So majorly it is dominant parent, or I can say that homozygous dominant parent. Okay? So out crossing mein what will be done? F1 individual is crossed with the dominant parent. Is that clear? So what are we talking? Back cross. We are saying in the back cross, the F1 individual or the hybrid is crossed with one of its parent, any of its parent. We are not specifying here when we are simply saying back cross. Now back cross is having two types, right? Now back cross is having two types, in which number one is out cross, in which number one is what? In which number one is out cross. Number one is what? Number one is out cross. In out cross, F1 individual or the hybrid is crossed with dominant parent, right? Is crossed with dominant parent. Now see, what happens is that when there is the continuous inbreeding or continuous self-pollination, yeah, inbreeding basically, like there used to be a chapter strategies for enhancement in food production. We used to discuss inbreeding. So when continuously, you know, in the same generation, breeding is there for 6 to 8 generations, 4 to 6 generations, or 6 to 8 generation, when continuously there is the inbreeding, right? So after certain time, what is going to happen? The inbreeding depression will be there, means the fertility and the productivity will be reduced. You can even see the same thing in the plants. If there is continuous self-pollination, after few generations, what will happen? After few generations, what will happen? That will come down. The productivity and the fertility will come down. So in that case, right, just to overcome that inbreeding depression, we call it as inbreeding depression, which occurs due to the continuous self-pollination or the inbreeding, then that results in the decreased fertility and the productivity of the progeny. So how can we overcome inbreeding depression? By using outcross, right? Used to overcome inbreeding depression as well. Okay, so it means that the one whose productivity is low, right, what will happen? That will be crossed with the, like obviously not from the same generation, from another one, right? Species will be same, breed can vary. So that will be crossed, okay? That individual will be crossed. This is basically the outcrossing, right? That is something that we used to discuss there. But here what are we simply saying? That it is the F1 individual which is crossed with the dominant parent. So this part is clear. Now the topic here is the test cross. So what is test cross? Test cross is a type of back cross in which F1 individual is crossed with its homozygous recessive parent. Is crossed with homozygous recessive parent. It is crossed with homozygous recessive parent. For the test cross, students, this condition is fixed. You are going to cross F1 individual or the hybrid with the homozygous recessive parent. So this test cross is used to determine unknown genotype. Now if you will ask me that ma'am, how can we determine unknown genotype with the help of test cross? See, in the test cross we have specified that we are going for the homozygous recessive parent, right? Homozygous recessive parent. So if you are crossing something with a homozygous recessive parent, if that something is dominant, then it will express, because another party, the another parent is in the recessive state only. So what can we do? Like, how can we understand that? I'll explain you by taking two cases, right? I'll explain you this test cross by taking two cases. Just let's suppose, not exactly the cases, but yeah, in two ways, in two examples I'll give you by which you'll be able to understand. Just let's suppose you're looking at a plant, okay? When you are looking at a plant in a population, you can simply say, though when we talk about the tallness, we see the intermediates there, but in general, let's consider the example. Just say, let's suppose you're looking at a plant, right? You are looking at a plant. When you are looking at a plant, by looking at it, you can say its phenotype, it's tall. Or I'll give you another example. Let's suppose if you're looking at a seed shape, can you say that whether it is round or wrinkled? Yeah, you can. By looking at it, you can say its phenotype, but not its genotype. Yes or no? By looking at it, you can simply say its phenotype, but not its genotype. So if we have to figure out its genotype, we'll go for the test cross. Just let's suppose here, if it is dominant, yeah, if it is tall, so how many possibilities are there? Two. Either it can be homozygous tall or it can be heterozygous tall. Either it can be homozygous dominant or the heterozygous dominant. So in the case one, what do we have? Let's suppose, because you know the phenotype but not the genotype, let's suppose, I'm just assuming it ki unknown genotype, so that you can understand ki unknown genotype is homozygous dominant. So you draw it, you'll get to know. Now you know that in test cross this condition is fixed. It's a test cross. So this condition, this particular condition is fixed, right? This particular condition is fixed in the case of test cross, that other parent will be homozygous recessive parent. Now what will you do? Right? Now what will you do? You'll make the Punnett square here. Right, so this side gamete will have capital T, capital T. This side it will have small t, small t. So now see. So in this cross, what you get? The next generation, all are dominant. So if in a test cross, my dear students, if in a test cross, you are conducting a test cross, you have the F1 individual, you are crossing it with homozygous recessive parent. So if you are, after crossing it with them, then you saw the next progeny, if you find that all are dominant, so what can you conclude? So what will be our conclusion? So it means, right, it means unknown genotype is homozygous dominant, right? It simply means unknown genotype is homozygous dominant. That's the meaning here, right? That is what this ratio is going to tell us. Got that? Then here, let's assume you don't know the genotype, na? You know the phenotype, but we are assuming that here unknown genotype is, capital T, small t, that is heterozygous dominant. Now let's take the cross. Because see, one parent we are assuming, but the another parent, for the another parent we are clear, right? We are simply clear because it is a test cross. So another parent will be homozygous recessive. Another parent has to be homozygous recessive if it is a test cross. Are you getting my point? Are you getting my point? So capital T, small t, small t, small t. So capital T, small t, capital T, small t, small t, small t, capital T, small t, small t, small t. This is the result that you are going to get. Okay, so here it is tall. The phenotype is tall. The phenotype is dwarf. The phenotype is tall. The phenotype is dwarf. So can I not say that 50% tall means dominant and 50% here are recessive, or I would say dwarf, isn't it? 50-50, isn't it? It's 50-50. It's 50-50. Yes or no? So can I not write this ratio? So the ratio here is 1:1. So if test cross ratio, so if test cross ratio is 1:1, so if test cross ratio is 1:1, it indicates that the unknown genotype is heterozygous dominant. Am I clear? Unknown genotype is heterozygous dominant. Am I clear? Done? So this is about the test cross. Okay? This is about the test cross. Understood? Yes? Yes? So this is the test cross. That is how it is helping us. Both are test cross because one parent, this is the F1, this is the parent, and we are saying that should be homozygous recessive. Here also both are test cross. What are you talking? What have you learned so far then? Right? So this is the test cross. That is how you can check the ratio. So whenever you see that the test cross ratio is 1:1, you should be clear that the unknown genotype is. So see, all flowers are violet means all flowers are dominant. So unknown flower is the, so interpretation will be what? That unknown flower is the homozygous dominant. Now see here, half of the flowers are violet, half of the flowers are white. So unknown flower is the, this is the interpretation, heterozygous. So this is from the NCERT, homozygous recessive parent. One side you will see dominant phenotype. So one side is homozygous dominant and this is something that we have to figure out. So that's why in the form of these cases I have explained you this. So this is all about the test cross, guys. This is all about the test cross. Got that? Now, if let's suppose, if you are talking about the dihybrid test cross. So again the story is same, students. If it is a test cross, let's suppose if one parent is this, another parent is this. So this is again a test cross. Why? Because this parent is homozygous recessive. So you know that next generation all dominant. But that's not the case. Let's suppose if this is the example. If somebody is giving you this particular cross, so there is no need to sit to draw, right? See, look at this. This one is heterozygous dominant. This one is heterozygous dominant. And you know that this one is homozygous recessive. So you should be clear about it that here we are talking about a test cross only. Even let's suppose if it is written like this. It is a dihybrid test cross, right? Just giving you example. If it is written like this, it is a trihybrid test cross. So in test cross, one of the parent, one of the individual that is crossing should be homozygous recessive, right? So here in this, you'll get the ratio 1:1:1:1, 25% each, right? 1/25% each. How come this is possible? Let me show you. So you know that what will be the possible gametes here? Capital R, capital Y, capital R, small r, capital Y, small r, small y. And here, what are the possible gametes? They are all going to be same, that is small r, small y. So here, you will get capital R, capital Y, small I. See this. This is going to happen. This is the test cross. So this parent will make 4 different types of gamete. It is the heterozygous dominant, but this parent is making only one type of gamete because it is the homozygous recessive. So that is going to be the progeny, right, after crossing here. RRYY, right? RRYY, right? So this is going to be this. So of course, that's why we are saying in dihybrid test cross ratio 1:1:1:1:1. 1:1:1:1. Got that? Yes, got that? 1:1:1:1. So heterozygous dominant, homozygous recessive, you got the ratio. So this is all about the test cross. Okay. So now you obviously you'll get the break, but I have a question for you, that is possible gametes for following genotypes. Number 1, chaliye. So you have to tell me the possible gametes, possible gametes for following genotype, but after a break. So let's meet at 9 p.m. Enjoy your break. So see you all then. Okay? So as promised. Hi everyone. So I hope I'm perfectly audible. Am I audible? Sure. Okay, so now here you can see, let's start with this part. Okay, so it's a tetrahybrid cross. So if you have to make the gametes here, how can you do that? Again, see, write it like this, capital A here, small a here. Then you have capital B, small b, capital B, small b, capital C, small c, capital C, small c, then capital D, small d, capital D, small d, capital D, small d, capital D, small d. Same as here. See this. Okay, so A, B, C and D, 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 A, B, capital C, capital D, small d, A, B, small c, capital D, B, small c, small d. That is what we have. Can you see that? Right? And same thing you have to do here. So what you guys can do, it is small a, capital B, capital C, capital D, small a, capital B, capital C, small d. So you can see this here. So that is how. So one parent is giving how many gametes? Yes, please. One parent is giving how many gametes? Because it is a tetrahybrid cross. So one parent is giving, right? One parent is given 16 gametes. Now when you look at this, see, AA will give you same type of gamete. This will give you same type of gamete. So here you will get, because it's a kind of trihybrid, so here you will get 8 different types of gametes. Here you will get 4 different types of gametes. So you people can solve it. You can just let me know the answer in the comment section and please make sure you solve it, okay? This is important. Chaliye, so let's discuss the next part here. Let's discuss the next part here. So the next thing that we have to discuss is gene interaction, right? What we have to discuss? We have to discuss the gene interaction. Now when you talk about the gene interaction, so you have 2 things, my dear students. One is the allelic interaction, right? One is the allelic interaction and the another one is the non-allelic interaction. One is allelic interaction and the another one is the non-allelic interaction. Now students, when you are talking about the, yes, when you are talking about the allelic interaction, you are talking about the alleles here, right? So can I not say that it is also the intragenic interaction? Can I not say that it is also the intragenic interaction? And here this one is what? Intergenic interaction. So this is the, right, so one type, and this is actually the post-Mendelian inheritance. Here you will see the deviations from the Mendel's law. Okay, so one thing is the allelic interaction, which is also known as intragenic. So what is the meaning of intra? Intra means within a genes. So within a gene we have the allele, right? And that is the intergenic interaction, which is the non-allelic interaction. So here what will you study? We are going to talk about the incomplete dominance. Yes, students, here we will discuss the codominance. We will also talk about the multiple allelism. We'll talk about the Pleiotropy here and the lethal genes there. So they all come under this allelic interaction. And here there are many, but we will majorly focus on complementary genes and the epistatic genes. If you will solve, if you pick up the AIMS PYQ, you will get a question on the epistasis as well. So that's what we are going to discuss, okay? That's what we are going to discuss. So in the intra, that is within. Inter means two different genes. So in the intragenic interaction, we will see that how within a gene, two alleles, right, they are affecting like each other's expression, or related to this we have to learn. So we will start first with, yes please, we will start first with incomplete dominance. What are we going to start? We are going to start with incomplete dominance. See students, whenever you talk about the dominance, or let's suppose we say that some genes are some alleles are dominant, some are recessive. On what basis we say so? We can explain it with the help of concept of dominance, again given in NCERT. Basically these genes, these alleles, they make the proteins. You all are aware of the central dogma of information, that DNA will form the mRNA and mRNA will form the protein. I hope you all are aware of it, that DNA will form the mRNA and mRNA will form what? Right? mRNA will form what? mRNA will form the proteins. This is what we know. That's how a gene expresses itself. So ultimately when a gene is, your DNA is making a protein, it is making an enzyme, right? That enzyme is expressing actually. Okay, that enzyme is expressing actually, yes or no? Yes or no? Yes or no? Of course. So the point is, when you talk about a dominant allele, that dominant allele will give you the functional enzyme. That will give you the functional enzyme, right? And that is why you will be able to see the expression. Now let's suppose if a gene, if it gets modified, if even due to modification, if the same enzyme form, normal enzyme form, then again that modified allele will have the same expression like the unmodified allele. Plus sometimes what happens, due to this modification, due to this mutation in the allele, what is going to happen due to this mutation in the allele, sometimes the non-functional enzymes or no enzyme at all forms, right? No functional enzymes or no enzymes at all forms. So in that case you will see the expression, either it will not be there and it will be affected, right? It will not be like the normal one. So this is the concept of dominance as well. So now when I'm saying incomplete dominance, here you will see that the dominant allele will not be able to express itself. So I'll start directly with the example here, and I'll give you the example of Mirabilis jelepa. You have seen this, and I'll show you the picture as well. So Mirabilis jelepa, which is also known as 4 o'clock plant, right? Because here in this particular plant, the blooming is at this time, 4 o'clock plant, and we also call it as marble of Peru. This is also known as marble of Peru. Now here in this particular flower, like let's talk about the parent generation, there is a red flower, let's suppose we are denoting it with RR, and there is a white flower. There is what? There is a white flower, small r, small r. So you know that as per Mendel, if red is dominant, right, we know it very well that as per Mendel, right, we know it very well that as per Mendel, if red is dominant, right, if red is dominant, then in that case you know that as per Mendel F1 should all be red. But here what happened? We know that when we cross homozygous dominant with homozygous recessive, here we get heterozygous. But this heterozygous is not red, it is pink. It was observed. It is pink. So it is the post-Mendelian inheritance where we do not obey the, here we do not see the Mendel's law. Okay. Now for the F2, what are we supposed to do? For F2, there should be the selfing of F1, right? So let's make the Punnett square here. Let's check that out, right? So let's make the Punnett square here. So what you people can check? Yes, please. So what you guys can see here in this particular part, what you people can see here, see one parent will form RR, this is the gamete, this is the gamete. So here when you look at it very carefully, you will see capital R, capital R, capital R, small r, capital R, small r, and small r and small r. This is what we see here. Is that clear? This is what we see here. So this is going to be red color, this is going to be pink color, this is again the pink color, and this one is white. This one is what? This one is white. Is that clear? So here, look at the phenotype, look at the phenotype or look at the phenotypic ratio. So when you look at it, instead of 3:1, it will become 1:2, 2:1, where 1 is red, 2 are pink and 1 is white. Am I clear? Then there is the genotypic ratio And when you people talk about the genotypic ratio, again it is going to be 1:2:1. So here 1 will be homozygous dominant, right? So ultimately you'll see the phenotypic ratio is totally modified here. This will be heterozygous dominant. This will be homozygous recessive. Done? So that's how the ratios, they will be modified. Am I clear? Yes? Is that clear? So here you can clearly see the dominant allele is not able to express itself completely, right? So there is a kind of intermediate, right? So what kind of phenotype you are going to see? The intermediate kind of phenotype is there. Okay, so intermediate type of F1 generation is there because here the dominant character is unable to express itself completely. So I'm writing this part here. So here what is happening? Dominant factor or allele is unable to express itself fully. So intermediate conditions are seen. Intermediate phenotype. Intermediate phenotype is there. So this is what the incomplete dominance is. So the question is, so they have asked in the previous year paper regarding these ratios. So these ratios are PYQ. So here phenotypic and the genotypic ratio is same. And yes, it will, right here from this part, they can even ask you what? They can even ask you the examples. Just say, I'll give you the examples here. I'll give you the examples here. I'll give you the examples here. Just have a look. So second example is flower color in Antirrhinum majus. That is your snapdragon. Okay, that is your snapdragon or dog flower. Third is the foul color in Andalusian fowl, where what we see 1:2:1 ratio. One is going to be black, one is going to be white, and this 2 is for the blue. But what is the another important example here, students? This is the size of starch grain. This is again, this is a PYQ, this is a PYQ. Okay, so size of starch grain. Size of what? Size of starch grain in pea plant. Am I clear? Size of starch grain in pea plant. Is that clear? So here I'll give you the example. Just say, let's suppose students, if you have one starch grain of 20 centimeter, another is of 10 centimeter. So now in the F1, you'll see the intermediate. Like it will be 20 plus 10 by 2. Like average you will consider. So you can simply say that it is going to be 15 centimeter. Are you getting my point? So that's what you people are going to see. So the size of starch grain in pea plant is also there. So guys, if there is one more disease, that is Tay-Sachs disease, that also shows the incomplete dominance. Do you know that? There is one more disease that also shows what? That also shows the incomplete dominance. Actually, this particular disease is because of, it is actually autosomal recessive disorder. What sort of disorder it is? It is the autosomal recessive disorder. So if it is, see, we have 23 pairs of chromosome, 22 pairs are autosomes, 1 pair is the sex chromosome or the allosome. Now, if a gene is located on these autosomes, then obviously that disorders, that character is the autosomal character. Are you getting my point? So in this particular disease, again, it also shows incomplete dominance. This disease, it also shows what? It also shows the incomplete dominance, and it is an autosomal recessive disorder. So in homozygous recessive condition, it expresses itself. But how is it possible? See, this disease occurs when there is a deficiency of enzyme. This disease occurs when there is a deficiency of an enzyme hexose aminidase A. That is very important for the lipid metabolism. Okay, this enzyme is important for what? It is important for lipid metabolism. It is important for what, beta? It is important for lipid metabolism. Now what's going to happen here? So when this enzyme, it is absent, so lipid accumulation will there, right? Lipid accumulation will be there. So in the case of lipid accumulation, what is going to happen? Nervous system can degenerate and death can occur. Death can occur. So that is how it is. Now let's suppose if an individual is dominant for this, that individual is going to be normal because it is an autosomal recessive disease. So it will be in, right, homozygous recessive individuals, they will die. Homozygous recessive, they will die, right? But when it comes to the heterozygous individual, because in heterozygous, again, incomplete dominance is there. Yes or no? Yes or no? Again, what will be there? Incomplete dominance is there. In the homozygous dominant, because the enzyme is working fine, right? So obviously there will be no issue, right? The individual will be normal. But here in the heterozygous, what will you see? Half the normal level activity, right? What you people will see? You will see half the normal activity. So individual will survive. So this is also, this Tay-Sachs disease also shows what? It also shows the incomplete dominance. So this is all about the incomplete dominance. Now please tell me if there is any doubt here. Guys, quick. Please do let me know if there is any doubt here. Yes, please. Do let me know if there is any doubt. Anybody? Hello? Anybody? So up to this portion all clear? Okay. So next is okay. So next is what? Next part that we have to discuss is codominance. And co means together. So codominance, here you will see two different alleles for a same character. The alleles are different, but they are for a same character. That's the catch here. So these are different, right? Both the alleles, they are different, right? Both the alleles they are different, but they are for a same character only. या फिर particular character only. They simultaneously express itself, right? It simultaneously express itself, right? It simultaneously expresses itself. Now I'll give you the example here, right? The best example that I can give you here is of my own blood group, that is students AB blood group, right? The best example that I can give you here is of my own blood group. That is what? That is the AB blood group. Now you know that it is the gene I that controls blood group. That controls blood group. We will discuss the same in the multiple allelism part as well, but firstly we'll discuss it here. So gene I, this gene I, it controls the blood group. So IA, this is one allele, IB, this is another allele. So these both the alleles, they simultaneously, right, both these alleles, they simultaneously expresses itself. Both the alleles simultaneously express itself, right? Both the alleles simultaneously express itself. That is, they'll give you AB blood group. So RBC, right? Here you will see RBC will have A antigen as well as B antigen. Matlab in such individuals, RBC, this is the RBC, this is the surface of RBC, this is the A antigen, this is the B antigen. So RBC surface is having both the antigens. So there is no antibody. So there is no antibody in plasma. So this blood group, AB blood group, right, the universal recipient blood group, right? So here in this particular blood group, you will see the codominance is there. This is the best example. Okay. Second example is carrier of sickle cell anemia. Okay, carrier of, it is not disease, it is carrier, HbA, HbS. This is the another example. So I'll explain you the sickle cell anemia, but do you know that this sickle cell anemia is a very important topic? Do you know this thing? Do you know that this sickle cell anemia is a very, very important topic? Right, we'll discuss it further. Yes? Yes, please. Done. So now we will discuss the sickle cell anemia, okay? Done. So now see, before starting the sickle cell anemia, I'll start with the basics here. I'll talk about the hemoglobin. I believe you all know what hemoglobin is. Hemoglobin is our respiratory pigment. It is the hemoglobin due to which our blood is red in color, isn't it? Respiratory pigment. It is the respiratory pigment. And how will it help? That help in, this is the respiratory pigment that help in transporting oxygen. It doesn't store oxygen. It is the myoglobin muscle that store oxygen. It helps in transporting oxygen. Now what you have to understand, this hemoglobin, what exactly it is? It's a quaternary protein, right? It's a quaternary protein. It is having 4 polypeptide chains. It's a protein. Ultimately, it is made up of amino acid. Amino acids, they'll join with the peptide bonds. They'll form the polypeptides. So this is a quaternary protein, and the adult hemoglobin. See, fetal hemoglobin is different. In the fetus, it is different. I'm talking about the adult hemoglobin. And let me remind you, even in the chapter human reproduction, we have discussed this, that when you talk about the fetal hemoglobin, it is having more affinity for the oxygen even under low partial pressure, right, in comparison to adult hemoglobin. So adult hemoglobin is having 2 alpha chains, alpha polypeptide chains, 2 beta polypeptide chains. Remember, 2 alpha chains and 2 beta chains. Now when you talk about this hemoglobin, students, so as you know, it's a quaternary protein. It is having the iron, right? It is having the heme is the heme part and the globin is the protein part. So heme is having this iron, Fe, right? It is having the iron that will bind with the oxygen. So I hope you know that, right? Yes, please. I hope you know that. Yes or no? Yes or no? I hope you all are aware of it. Yes, please. Yes, please. Right. So what do we see here? This is what we see, that this is the hemoglobin, and this heme part is having, we are aware of it, this heme part is having iron, and it is Fe2 positive. It is ferrous iron. Right, it is the ferrous iron. This is what we know, that will bind with the oxygen. But now here, what's our concern? We are talking about the yes, please. So yes, students, so now hemoglobin, I told you it is having the 2 alpha chains and 2 beta chains. So hemoglobin, in beta chain, where, students? In beta chain at 6th position. See, it's a beta chain. Ultimately, it is made up of what? It is made up of amino acids. Amino acids joined by a peptide bond, right? Amino acid joined by a peptide bond. they form the polypeptide. So let's suppose this is 1, 2, 3, 4, 5 and 6. So in beta chain, right, in beta chain at 6th position, glutamic acid is replaced by valine. Glutamic acid is replaced by valine. So here you can remember a trick, good villain, right? In the beta chain, see, in beta...
In your Apple Music library,
Wow.
you can ask me to play a radio station.
I'm not calling you Siri, you keep quiet. So, in hemoglobin, in beta chain at 6th position, of course, due to point mutation, glutamic acid is replaced by valine. So when you talk about the glutamic acid is GAG, right? GAG. And then it forms what? It forms valine, GUG. Are you getting my point? So GAG is replaced by GUG, like glutamic acid is replaced by valine. So it is GD, right? Good villain, right? Good villain. So glutamic acid will, at the place of that, valine will come. Why am I specifying it? Because sometimes students, instead of glutamic acid, they mention glutamin, which is incorrect. It is glutamic acid. This is valine. And these amino acids, they are synthesized by a triplet base codon of these nitrogenous bases. So for glutamic acid, here you have GAG, right? And valine, at the place of valine, see, GAG will become GUG. So I always call it like this. GAG will become GUG. Right, GAG will become GUG. So A, at the place of A, U will come, and you see the amino acid will change. And that amino acid, because of this mutation here, right, the RBC, that also changes. I'll tell you how. So basically, sickle cell anemia. So this is what sickle cell anemia is. So in sickle cell anemia, one thing that you have to remember, in pedigree analysis also we'll discuss, it is autosomal recessive, right? This disorder is autosomal recessive disorder. I'm discussing this here with the codominance only, right? I'll not discuss it later, so please pay attention. So sickle cell anemia is the autosomal recessive disorder, right? And it occurs due to the mutation in HBB gene. So this gene encodes for, this gene encodes for what? This gene encodes for the beta chain. So obviously mutation in it will result in that, and this gene is located on chromosome 11, and to be more precise, it is the short arm, right? It is the short arm. Is that clear? It is the short arm of chromosome 11. Is that clear? Is that clear? Yes, everybody? Sure? Done? Any doubt? Done? Any doubt? So now what's going to happen due to this? See, RBCs, when there is the low oxygen tension, one expression that you are going to see at the cellular level or the cell shape level, that RBC will become sickle shaped. Just a minute. That RBC will become sickle shaped, right? The RBC will become sickle shaped. This is the first thing. And you know that what, right? Beta chain has changed. That is why whenever there is the oxygen tension, then because of that, right, the RBCs, they will become sickle shaped. So this is what the sickle cell anemia is. And here I would like to add one more thing. Just a minute. Disorders, though I have already discussed, but still. Yeah, so here I have added, so because I have discussed it here, so I would like to specify that, yeah, this is even important, right? So normal A gene, what is the sequence written here? Then mRNA is having GAG. In sickle cell anemia, it will become GUG, right? So you can see the changes there. Here CT will become A. And even you have to remember what is like 6th is GLU, glutamic acid. If here GLN is written, then also it is incorrect, because GLN means, right, GLN means, right student, glutamin, but this is the glutamic acid. So you should even remember valine, histidine, leucine, threonine, proline, glutamic acid and glutamic acid. So even the diagram has to be very clear, right? So this part should, yeah, so even you have to discuss it also. So now what is going to happen? So in sickle cell anemia, no doubt we are studying it under, see, if individual is HbA, matlab both the alleles are normal, so obviously normal hemoglobin will be there. If HbS, disease, obviously disease will occur. But if it is HbS, then what's going to happen? That individual is going to be the carrier, so there will be no anemia, but here there is incomplete dominance and codominance. And codominance. See, this is at hemoglobin level. So what I just read, I'm just mentioning here that if it is HbA, matlab that individual is going to be normal, right? Kevin, what am I supposed to look here? Please tell me. Matlab what am I supposed to look here? Right? So see, guys, see, I don't have the time for the wastage. Okay? We have a chapter, and that too, right? That too lengthy and important, right? And I don't want to compromise the content. Okay? So if you are here to study, then please most welcome. If you are not, then please do not spam. Okay? So in between, I'll check the chat. I'll take your doubts, but firstly focus here. Okay? So now I am saying, right, then when you are talking about this part now, I am saying that in the case of sickle cell anemia, HbA, both are normal. Yes or no? Both are normal. Normal means disease free. This one is having the disease, right? This one is having the disease. Here you will see even that can be lethal as well. So sickle shaped RBC will be there. All the RBC will be sickle shaped. Okay? All the RBC will be sickle shaped. Okay, right? Oxygen tension is going to be there. Then it is the carrier. So anemia is not there, but here we see that this part, it is also showing us the codominance, plus it is also showing us the incomplete dominance. Now, no doubt we are studying it under codominance, but here, like, this is debatable. Do you know that? HbA, this individual is the carrier, and do you know it is debatable that whether it is the incomplete dominance or the codominance. So let's discuss it, right? So inheritance pattern here shows. So as I said, inheritance pattern here shows incomplete dominance. As I said, I mentioned it in the last slide and I am again writing it. Incomplete dominance at cell shape level or cellular level. I'll explain it. And codominance at hemoglobin level, Hb level. So anybody who can explain it to me, right? Or I would say at molecular level. Anybody who can explain it to me? Anybody? Yes, please. Anybody who can explain? Do you have any answer for this? Then explain. Quick. So because see, when you are talking about the carrier here, now why do we say it is the incomplete dominance? Because here, when you look at the RBC, they are not even the normal shape, they are not even the cecal shape. The structure is somewhere in between. Whenever there is the oxygen tension, then so it will become the cecal shape, right? But here you will see, when you talk about the heterozygotes here, what you people are going to see that here there will be a phenotype, right? The phenotype is of intermediate, right? Intermediate phenotype. Phenotype is intermediate type, right? What this phenotype is? This phenotype is intermediate type, right? Not normal, not diseased, right? Not normal, not diseased. Are you getting my point? So that's why, because here the phenotype is, because whenever there is the oxygen stress, then to RBC will become, then to for sure RBCs will become cecal shape, but otherwise this is how it is going to be. Is that clear? Is that clear? Right? So now why codominance? Because HbA will form normal hemoglobin and HbS will form cecal shape, like it will form abnormal hemoglobin, right? Or it will make cecal hemoglobin. That's what we can say. So that's how it is the codominance. Is that clear? Is that clear? Is that clear? Quick. So normal hemoglobin and cecal hemoglobin, both type of hemoglobin will form. So at the physiological level, or I would say at the phenotypical level, so this is what you are going to see. You will have normal RBCs and cecal shaped RBCs when oxygen is low. So symptoms are mild or often absent in this case. Okay? So here you will see the cecal shaped RBC only under oxygen stress. So that's how we can discuss it. That's how we can say that we can put it in both the categories. Is that clear? Right? So we have the, and you can just give it a try. See, HbA, HbS, just cross these intermediates, HbA, HbS. Okay. Let's check the ratio. So HbA, HbS, right? We are just crossing these F1 individuals. So here you can see oh my bad. See this. Now see, normal, carrier, disease. This can be lethal as well. So here 3:1 modified to 1:2:1. That's my point. Okay, 1 is normal, 2 are carrier, 1 is disease. Same for the phenotype, same for the genotype. So again, in codominance also, right? Overall, in codominance also, students, what are you going to see? The phenotypic and genotypic ratio is same. That is 1:2, 2:1, 1:2, 2:1. So that's what you people are going to see here. So the phenotypic and the genotypic ratio is going to be same. 1:2, 2:1, 1:2, 2:1. Am I clear? Am I clear? Yes? Yes? Quick. Okay. So this is about the sickle cell anemia in detail. So next part, third example, coat color in short horned cattle. जैसे see students, red colored cattle, you can also say CR cross with white that is CW, CW. So it's not like that, ki ma'am, these are the two different, right, alleles. See guys, that here you are using R or the W, like you can simply write here R or W as well. So it's not like that, right? Matlab here at the place of CR, you can also write it as R, CW, you can also write it as W. So sometimes students ask me that, ma'am, how is it possible? Why RW? See, it's very simple, right? It's very simple. No doubt they are two different letters, but point is they are controlling the same character. Like we are using R for red, W for white, but they are ultimately controlling the same character, that is the cattle coat color. So that's what I'm trying to tell you. So when you talk about the F1, so it will be CR, CW, right? So now let's take the F2. Let's take the F2. So in the F2, what you people will see? In the F2, what you people will see? See, for F2, you have to multiply this. So see this red, this is going to be Ron, and this is going to be white. So again, phenotypic and genotypic ratio is same, that is 1:2:1. Again the example. So which color is the Ron color here? This one. Red, white, Ron. Red, white, Ron. Understood? See this. So that's how we can check. So just have a look. So next part that we have to discuss is multiple allelism. Okay, multiple allelism. In the multiple allelism also we have many examples. So before starting the multiple allelism here, I'll tell you or I'll explain you the concept of dominance, okay? I'll explain you the concept of dominance. Just allow me 1 minute. Okay, I didn't add it here. Acha, theek hai, it's okay. So when you talk about the concept of dominance, just see, as I said, every gene carries the information right? Every gene carries information to express a particular character. I believe you all are aware of it, right? To express a particular character. Yes or no? Every gene is having that. So in the diploid organisms, we have the two copies. It can be homozygous, it can be heterozygous one. So the point is, right, if one is different from another, just like in the heterozygous, right? Just like in the heterozygous, right? If, let's say, you know, both the alleles are different, right? So why is it so? Why is it so? Because of some changes. We can say that because of some mutation. So what is going to happen here, students? Just a normal allele will give you normal enzyme that will act on the substrate, will convert it into the product. But when there is the modification, right, some changes can also occur. Just say, let's suppose, I'll give you the to the point just here. Just say, let's suppose if you talk about the heterozygotes, right? So here you'll see two different alleles. Here, what will you see? You'll see two different alleles. Yes or no? Here, what will you see? You will see two different alleles. So, see, normal allele will make normal enzyme. You'll get that normal enzyme will act on normal substrate, normal protein, isn't it? Isn't it? Right? Done? Isn't it? Now let's suppose if there is an allele that is modified allele. Now in the case of a modified allele, what are you going to see? Now if the allele is modified, right, theoretically, right, so it can form it can form, matlab theoretically, it can form, what can it form? Number one, normal or less efficient enzyme. Matlab even after that enzyme is forming, maybe it can be less efficient, but enzyme is there, right? Enzyme is there. It can be normal or less efficient. Secondly, it is non-functional enzyme. It is not acting on substrate. It is not giving us that product, or no enzyme at all, or no enzyme at all. These are the three chances. One allele is the normal allele that will form the normal protein, that will act on the substrate, will transform the substrate. Easy, simple, that's it. Second is what? Second is basically, like let's suppose if there is any modification, so even after the modification sometimes we get the same result. Okay, so normal or the less efficient enzyme can form. If it is normal, the normal type of phenotype will be there, right? But if there is non-functional, enzyme is not working, then obviously the results will vary, the expression will vary, isn't it? And if there is no enzyme, then to obviously, then to it is for sure na students, if there is no enzyme, the substrate will not convert into the product, you will not get the expression. So in the first case here, right, even after modification, here even after modification you will get the same, right, phenotype equivalent to the unmodified allele. Is that clear? Is that clear? Is that clear? Yes? Sure? So here the phenotype is equivalent to unmodified allele because you have enzyme here. है ना? And in this case, what you people will see? Phenotype is modified, right? So unmodified phenotype, basically it is giving me with that dominant expression, and the modified allele, modified allele is the recessive here, and the unmodified one is the dominant one. Is the dominant one. Am I clear? So this is the concept of dominance. Because of such modifications, we see the expressions, they also vary, right? Because of this, they also vary. Is that clear? Is that clear? Sure? Done? Sure? So this is about the concept of dominance that is applicable here as well. Okay, chaliye. So now let's talk about the multiple allelism. Am I clear? Now let's talk about multiple allelism. See, as per Mendel, a gene is having two alleles, isn't it? That's what we discussed. As per Mendel, a gene is having how many alleles? Two alleles. But later on, right, later on with further studies, with the post-Mendelian inheritance, we got to know that it is not just the two alleles, right, more than two alleles can also be there. Are you getting my point? What can also be there, right? More than two alleles or more than two alternative forms can also be there. So more than two alternative forms of a gene is basically what, right? This is called more than two alternative form of a gene, or I would say of same gene, right? Of same gene, they are called multiple alleles. What do we call them, students? We call them as multiple alleles. And obviously, this phenomena is multiple allelism. Are you getting my point? This phenomena is multiple allelism. Are you getting my point? This phenomena is multiple allelism. Am I clear? Sure? Yes, please. Am I clear? Yes. So Mendel was saying only two alleles are there, but here what do we observe? More than two alleles, right? More than two alleles. So how these multiple alleles formed? Multiple alleles formed due to mutations. They formed due to mutations. When there is a mutation in a gene in different direction, obviously you will get this. But what is the speciality here? That they occupy same locus. Multiple alleles occupy same locus in a chromosome or a homologous chromosome. Now what's the meaning of that? Can you explain it to me? Yes? See, let's suppose this is a chromosome, paternal chromosome, and this is a maternal chromosome. It's a homologous pair. Now it is not going to happen like you have, let's suppose you have a character A. This character, you have a character, this character is having different alleles. Just say, let's suppose you are talking about the blood group. We have IA, IB, I. Yes. Blood group is also the example of multiple allelism. It is having three different types of alleles. Now you understand. Now if let's suppose if here you have the allele for blood group, it can be anything. IA, IBI, anything. So in the homologous pair of chromosome here also, you will get the another allele. So it's not going to happen that if you have the three alleles, so one allele will be present here, yeah, another allele will be on some other locus, or it will be on some other chromosome. No, it's not like that, right? It's not like that. Are you getting my point? It's literally not like that. So whenever you talk about these points are very important for assertion reasons and for statement based questions, right? That multiple alleles occupy the same locus in a chromosome. Their locus will not change. Is that clear? They will occupy the same locus in a chromosome. Is that clear? They will occupy what? They'll occupy the same locus in a chromosome. Is that clear? Is that clear? Yes? Sure? Are you sure? So next line, that multiple alleles. See, it's not like that these multiple alleles are for different characters. No, the character is one only, like I am telling you about the blood group, but it is having the different alleles. So multiple alleles will always, right? So if it is a multiple allele of a particular character, then obviously it will influence that only. So multiple alleles always influence the same character. Always influence the, like if I told you about the blood group, right? If I told you about the blood group, so blood group is having three alleles. All the three alleles are, right? All the three alleles are basically doing what? All the three alleles are basically controlling the blood group only, and there is no crossing over in the, so there will be no crossing over in the alleles of the multiple allele series. So they will not exchange. Are you getting my point? So they will not exchange. So there will be no crossing over within the alleles of the same multiple allele series. Is that clear? Yes, please. Right, there will be no crossing over in the alleles of same multiple allele series. So these are the points that you need to know regarding the multiple allelism. And here I am going to write a very important point. That's C. If a gene is having more than two alleles, then that alleles are, then all that alleles are visible in population, but in a single individual only two alleles are present for the same character. So that's important, underline it. Quick, everybody, that's important, underline it. Yes, please. I'll repeat this again. First of all, more than 2 alternative forms of allele, because Mendel has said that 2 alternative forms will be there, but here we are saying that they are more than 2, right? So it is multiple allelism. Second, I am saying it is formed due to mutation, very easy. Third, and a very important point, that multiple alleles will not occupy the different locus in a chromosome or in a homologous pair of chromosome. They'll share the same locus. It's not like that if a gene is having 10 alleles, so that 10 will be present on the different locus in the chromosome. No, they will be present in the same locus in a chromosome. That's what you have to understand. Plus, in the case of multiple alleles, if a gene is having more than 2 alleles, it doesn't mean they'll control a different character. It will control the same character only. Like blood group having 3 alleles, so it is not controlling something else. It is just controlling the same character, that is blood group only. And in the series of the alleles of same multiple allele series, crossing over will not occur. Now guys, if see in a population, just say for an example, I'll give you this example as, in a population you'll see individuals with A blood group, with B blood group, with AB blood group and with O blood group. Am I right? But in a single individual, that is the population that I'm talking, that in the population you'll see A, B, AB, O blood group. But let's talk about the single individual. Let's take your own example. But we will have only 2 alleles for the blood group. Like my blood group is AB positive. So I got the A allele from one of my parents and B allele from another parent. One parent has given me A, another parent has given me allele for B. So overall in population you have 3 alleles. But in my case, I have only 2 alleles. Now let's take your example. Let's suppose your blood group is A, right? Homozygous A. So you will have 2 alleles only. One is obviously for A, another is also for A. One from one parent, another from another parent. So ultimately, what am I trying to say? That in multiple allelism, in the population you will see all that character, all that alleles, but in a single individual, right, because we are the diploid one, we are getting 1 chromosome from mother, 1 from father. So in our case, this is what you people are going to see, that we will have only 2 alleles. So that is why I'm saying that this line is very important, so do underline it. Okay, this line is very important, so guys, do underline it. Is that clear? Sure? Sure. So next part in the case of multiple alleles. Next part in the case of multiple alleles. So there is a formula. If you want to check the number of genotypes here, so how can you check? The formula is n plus 1 over 2. N is equals to number of alleles. Okay, like I'll give you the example here. Blood group is controlled by gene I, which is having three different alleles. Which is having what, students? 3 different alleles. Are you getting my point? Now, just for an example, IA, IB or I. This is for A blood group, right? This is for B blood group. They both are dominant. And this one is for O blood group. Do you know that? This one is for O blood group. So now if you have to check the genotype in the case of blood group, na? So 3 plus 1 over 2. It will be 6. So you are going to get 6 genotype. Let me discuss it how. Right? Let me discuss it how. So do you know that Carl Landsteiner? There was a scientist, students. There was a scientist whose name was Carl Landsteiner. What was the name? Carl Landsteiner. So in the case of Carl Landsteiner, what do you see? he discovered ABO blood group, right? Like A, B and O. So he discovered ABO blood grouping system. Do you know that? He was the one who discovered ABO blood grouping system. What? ABO blood grouping system. This part is clear? Sure? He was the one who discovered what? ABO blood grouping system. Now, D. Castello and Stirling discovered AB blood group. So Karl Landsteiner, he was not the one who discovered AB as well. D. Castello and Stirling, students of Karl Landsteiner, students of Karl Landsteiner, they discovered AB blood group. Okay, right? So you know that RBC surface here, I believe you know that RBC surface contains antigen, and in plasma, what is present in plasma? Antibody against other blood group is there. Like for an example, if a person having A antigen on RBC surface, then blood group of that person will be A blood group. Right, students? A blood group. If a person having A antigen on RBC surface, then blood group is what? A blood group, and plasma will have anti-B, like antibody against B blood group will be there. Are you getting my point? Are you getting my point? Students, I'm revising it again. What are we talking? We are talking about the multiple allelism. We are saying if a gene is having more than 2 alleles, it means it shows the multiple alleles, or the phenomena is multiple allelism. How these multiple alleles form? Due to mutation. Then, what we discussed, that in the case of multiple alleles, they occupy the same locus on the chromosome. Their locus will not change. They will occupy that same position. And other than that, we discussed that if a gene is having more than 2 alleles, all that other alleles, ultimately all that alleles are going to express a particular character only. It's not like that they'll express different characters, right? Plus, most other important point that we discussed is that if in a population, if particular character is having 3 alleles or 4 alleles or 10 alleles, doesn't matter. But in a single individual, you will always get 2 alleles, because we are diploid organisms. We'll get one allele from one parent, another allele from other. So we will always be the diploid. For that, we will be having the 2 alleles only. Is that clear? Is that clear? Now we are talking about the formula. We are saying if we have to check the genotype here, so the formula is n plus 1 over 2, where n is the number of allele. So example, blood group example we are discussing. So I told you about the history part as well, that Karl Landsteiner discovered ABO blood group, D. Castello and Stirling discovered AB blood group. So RBC surface will have antigens, and in plasma you will see the antibody. So if a person is having A antigen on RBC surface, then blood group will be A, and the plasma will have anti-B, and the plasma will have anti-B. Is that clear? Is that clear? Sure. So same way, B blood group, B antigen on RBC surface, and plasma will have anti-A. In AB blood group, both A and B antigen on RBC surface. right? So already you know that this AB blood group, it shows us what? Co-dominance. So if RBC is having both the antigens, so definitely it cannot form antibody against anybody. So plasma will have no antibodies, or in the case of antibody section, you'll write the nil. So no antibodies are there, right? No antibodies are there. Is that clear? No antibodies are there. So that's why AB blood group is the universal recipient. Now in O blood group, what will you see? No antigen. Only H antigen is there, but no this A and B. No antigen on RBC surface. Plasma will have both anti-A, anti-B. So students, O is the universal donor. Right? O is the universal donor. Now let's apply the formula. Formula says n plus 1 over 2. Right? For the number of genotypes. This is what this formula is saying. So how many alleles do we have? 3. So 3 plus 1 over 2, we will have the 6 here. So number of genotypes are 6. So how is it possible? So I'm writing it here, genotypes. Here I'm writing phenotype. So genotypes में see, homozygous A, heterozygous A, homozygous B, heterozygous B, AB and then O. That's what we have. So A, B, B, AB and O. So you people can see, right? Again, it's a previous year question. Number of genotypes 6 and number of phenotypes, they are 4 only. So that's a previous year question for the blood group. Do check it out. Quick. Right? Make the notes as well. Guys, quick. Yes, please. Is it done? Sure? So in case here you want to figure out number of heterozygous, this is extra, but yeah, this is important. You can go for n minus 1 over 2. So 3 minus 1 over 2, it will be 3 after solving it. So heterozygous 1, 2 and 3. That's how we can solve, right? So number of heterozygous will be n minus 1 over 2. And how can you check the number of homozygous here in the case of multiple alleles? Wait, I'll write it properly. I need some space here. Just a minute. Done? So that is also 3. So how come homozygous? So you can see this one is homozygous, this one is homozygous, this one is also homozygous. So that's how you can check. So these are the formulas. Understood? These are the formulas. So this is for the possible phenotype, possible genotype, right? And another example is the rabbit coat color, right? That is having the 4 alleles, right? So the next example is rabbit coat color having 4 alleles. So students, if 4 alleles are there, then please tell me the number of genotype possible. Quick. Answer this question. Number of genotype possible. Quick. I'm not writing it here which like gene is giving what, like, but still. Exactly. So it is going to be 10 here. How come 10? N plus 1 over 2. So 4 plus 1 over 2, 10. That's how you can check. Okay. So the next thing that we have to discuss here is Pleiotropy. Okay. Pleiotropy. Done? So Pleiotropy, then lethal gene, then Epistasis complementary gene, polygenic inheritance, or after that I'll give you a break, then chromosomal theory of inheritance and linkage all together. Bas, major portion done. Then the disorder and the mutation part. Okay? So any doubts till then? This class is going to be lengthy. It is to for sure, na? The chapter is lengthy. Okay. So let's discuss about the Pleiotropy, okay? So Pleiotropy means multiple phenotypic effects of a gene. Like what is, as per Mendel, there is a gene, hai na? Let's suppose there is a gene A. As per Mendel, that gene A is controlling a particular character. Let's suppose we are calling it as character number 1. That gene A is responsible for character number 1. That a single gene, that a gene controls or expresses a character. This is what we discussed. Now, as per Pleiotropy, if you talk about a gene, then that gene is not controlling just one character, or I would say character 1. That gene is also controlling character 2 also, like it is having effect on other phenotypes also. It is not affecting one phenotype only, it is having effect on other phenotypes also. So that is the meaning of Pleiotropy. Are you getting my point? That is the meaning of Pleiotropy. Right? That is the meaning of Pleiotropy. Are you getting my point? So in the case of pleiotropy, what we need to remember that here a single gene is like, as per Mendel, a single gene is controlling single character, but here what do we say, that a single gene can control more than one characters, right? So that means, what is this pleiotropy? Pleiotropy means multiple phenotypic effects of a gene. That's also you can write, or you can say multiple, or you can simply say a gene is controlling more than one phenotype. So that's what pleiotropy is. Am I clear? That's what pleiotropy is. So multiple phenotypic effects of a gene, or you can say that a gene is controlling more than one phenotype. Okay. Just say for an example, let's suppose a teacher is teaching, right? Let's suppose a teacher is teaching physics as well as chemistry as well as maths, but that teacher is a major teacher of chemistry only. Chemistry is the specialization, but yeah, teacher is also capable of teaching physics and maths as well. But what is the specialization? The specialization is chemistry. Same is the case with the phenotypic genes. Yeah, same is the case with the pleiotropic genes. Here, no doubt they can influence more than one character, right? Or they are controlling more than one character, or they are influencing more than one character, but they will have the, majorly they'll control a particular character and other, right? And they will have the secondary effect on the other characters. Okay, they will have the secondary effect on the other characters. Is that clear? Is that clear? Sure. So how this how this pleiotropy is possible? This pleiotropy is possible in such a way that a single gene and the product that it is forming is affecting many metabolic pathways. Okay, gene is going to form the protein. If that enzyme or that protein, if it is getting used in different metabolic pathways, then obviously, then without any doubt, right, then without any doubt, this is something that you are going to see, right, that this gene will affect more than one phenotype, right? Okay. So here a single gene is having, so you can just write it down here, a single gene is having effect on such metabolic pathways that influence other phenotypes. Okay. So yes, this is what you have to write, right? So single gene is having effect on such metabolic pathways that influence other phenotypes or which contribute towards them. Done? Done? So this is what pleiotropy is. Okay? This is what pleiotropy is. Understood? This is what pleiotropy is. We'll read it from the NCERT as well. So let's discuss the examples here. Here we have many examples. See, we will talk about the cystic fibrosis, sickle cell anemia, phenylketonuria here. Okay? And now the example that we have to discuss is again a PYQ. So you write down example, size of starch grain and seed and shape of seed size of starch grain and shape of seed controlled by same gene. Do you know that? Which is located on chromosome number 7. So imagine, right? So thank God Mendel, he didn't select the size of starch grain. He just selected the seed shape. So imagine the seed shape and the size of starch grain, right? The seed shape and the size of starch grain. Guys, listen to me. Seed shape and the size of starch grain. Yes, they both are controlled by same gene which is located on chromosome number 7. So here you have to understand one thing. And yes, I'm sorry, chalo, I'll complete this, then I'll go back to that topic. So size of starch grain. Just now we discussed size of starch grain. Just now we discussed, right? It was showing what? Incomplete dominance. Then seed shape here. This relationship is dominant recessive relationship. This is what? Dominant recessive relationship. Okay, dominant recessive relationship. See, firstly, this is the example of a single gene because this single gene is controlling more than one character. So it is the example of Pleiotropy, number 1. But here, when I talk about these two characters, like first is the size of starch grain, the another is the seed shape. This one is showing incomplete dominance. This one is showing dominant recessive relationship. So what does it mean? It means like when this is the genotype, now in such genotypes, what is going to be the seed shape and what is going to be the size of starch grain? That's what I'm trying to explain. Done? So seed shape or size of starch grain, BB. So here seed shape. So here you will see, because homozygous dominant, seed shape is going to be round. It is also the dominant recessive relationship round. This is going to be wrinkled. But here, because it is the incomplete dominance. So here large sized starch grain, small sized starch grain, and here you will see the size is intermediate. That is going to happen here. Here the size is intermediate. Is that clear? Sure? Sure? Understood? So one gene is controlling more than one effect. Okay? One gene is controlling more than one effect. Okay? Okay? So now second example is of sickle cell anemia. Yeah, sickle cell anemia is also there. Now you know, I told you na, sickle cell anemia, we'll discuss it again and again. So sickle cell anemia, it also shows Pleiotropy. How is it possible? See, you know that occurs due to mutation in, just now I told you, HBB gene located on chromosome 11, है ना? Which is located on chromosome number 11. That's how it is occurring. Now you know that. So HBS is having primary effect on RBC shape and hemoglobin and on hemoglobin and at hemoglobin level it is having at RBC shape at hemoglobin. This is the primary effect, okay, but it also has secondary effect, right, which are not main, which are not major. Now because RBCs are sickle shaped, so you can see that that will clog blood capillaries. It can even, students, obstruct blood flow, right? It can even obstruct blood flow, right? So now blood flow is obstructed, so the organs, it can affect the eyes because the blood flow is affected. It can even affect the other body organs as well. Is that clear? So sickle shaped RBCs can clog the blood capillaries, can obstruct the blood flow, right? And moreover, you know, the other organs, they will also get affected because of that, because the RBCs are sickle shaped. Now is that clear? Is that clear? Sure. Okay. So I am saying that, see, sickle cell anemia, which we have already discussed, right, which we have already discussed, which is an autosomal recessive disorder, right. So autosomal recessive disorder, it occurs due to the mutation in HBB gene, which is located on chromosome number 11. So I am saying that it also shows the Pleiotropy. How? See, the mutation in this gene is not just affecting the RBC shaped or not just affecting the hemoglobin, but it is also clogging the blood capillaries, obstructing the blood flow, hemolysis can occur, and obviously that body organs which are getting that like clogged blood capillaries, they will obviously, when they are obstructing the blood flow, then again it will be a problematic condition for the other body organs as well, isn't it? Isn't it? So that is what I'm trying to, that is what I'm telling you here. So that's it. Okay. So that's it. So this is about the sickle cell anemia, that how is it acting like a just a minute. So how is it acting like, how is it showing the multiple phenotypic effects? Okay. Done? So, third example here is phenylketonuria. So, see, I'll discuss the disorders here only, then we'll be left with the chromosomal one. So, phenylketonuria is also the example of Pleiotropy, right? It is also an autosomal, it is inborn. It is an inborn error in metabolism. It is an inborn error in metabolism, and it is also what? It is the autosomal recessive disorder, right? It is also an autosomal recessive disorder. Is that clear? Is that clear? So it is an inborn error in metabolism, right? And it is what? It is an autosomal recessive disorder. Again, this is also located on the recessive gene. So on chromosome number 12, you will see a gene, right? On chromosome number 12, there is a gene, right? There is a gene that makes an enzyme called phenylalanine hydroxylase. Called phenylalanine hydroxylase, that is PAH. This is the name of the enzyme, and a gene that is going to make it. Okay, so a gene, this is your PAH gene. So PAH gene means phenylalanine hydroxylase, right? So this particular gene forms an enzyme which is the phenylalanine hydroxylase. Is that clear? Is that clear? Right? There is a gene that makes an enzyme called phenylalanine hydroxylase that converts, this enzyme converts phenylalanine to tyrosine. So I believe you know that phenylalanine is also an amino acid, tyrosine is also an amino acid used in different purposes. I'll mention the one here. Tyrosine is important for the melanin production. Melanin is our students, melanin is our skin pigmentation. Do you know that? Melanin is our skin pigmentation and the tyrosine is very important for that. Okay. So what am I saying? That on chromosome number 12, there is a gene that makes an enzyme called phenylalanine hydroxylase, right? So this gene is PAH gene, the enzyme is PAH. So this enzyme is going to convert the phenylalanine into the tyrosine, right? Right? Now, due to this single gene mutation, so single gene mutation, what exactly is going to happen in this mutation? No PAH enzyme, right? No PAH enzyme. So because of that, what will happen? This phenylalanine, it will not be converted to tyrosine, right? It is not going to form the tyrosine. So phenylalanine gets accumulated as, gets converted or accumulated as phenylpyruvic and other derivatives. So basically, firstly, see, it is not going to form tyrosine. So if it is not going to form tyrosine, it means the skin related issues will be there, skin pigmentation issues will be there because tyrosine is not available, number one. Number two, here, because of the accumulation of this phenylpyruvic, mental retardation can occur. So there is a mutation in single gene, but it is also affecting the skin pigmentation. It is also because tyrosine is also required for other hormones as well. So it is also affecting the brain. It is also affecting the hair. It is also affecting the skin pigmentation, right? So accumulation of these in brain, accumulation of these in brain results in, number one, mental retardation, है ना? This is तो for sure this effect is there. So other than that, so skin pigmentation is affected. So we can say that reduction in hair and skin pigmentation, right students? Reduction in hair and skin pigmentation, comma, short stature, excessive sweating, etc. Right? Excessive sweating, etc. Done? Okay? Understood? So write down, please. Write down. Okay? Quick. Done, students? Sure. So one more example of pleiotropy is cystus fibrosis. Right, cystus fibrosis, in which CFTR gene is involved and that gene is present on chromosome number 7. Cystus fibrosis is also given in NCERT. So no doubt these are the disorders also, right? But here, see, it's easy. So they also show the multiple phenotypic effects. So we are completing it here, right? So we will not revise it then. Done? Done. So let's write the cystus fibrosis. So cystus fibrosis may gene involved. Which gene is involved here? Gene involved is CFTR gene, right? Obviously this gene will get mutated and the problem will start. So what is the CFTR gene? So it stands for cystic fibrosis, right? So what is this gene? So this gene is cystic fibrosis. Right, students? Transmembrane conductance regulator, and it forms chloride channels. Conductance regulator. It forms chloride channels which are present in different organs, right? So obviously if chloride channels will not work, the sodium and water absorption increase, chloride secretions will get affected, mucus will become thick. So it will not affect just one particular, right, organ. It will start affecting many. So that is why it is also the example of, right, that is why it is also the example of what? It is also the example of your this pleiotropy. Okay? Okay? Right? Done? Okay, so gene is located on chromosome 7. It forms chloride channels in epithelial cells of lungs, pancreas, sweat glands, etc. Like many, right? In many, not in just one. In many, this is going to form what? This is going to form the chloride channels. Am I clear? This is going to form the chloride channels. Is that clear? Right? Lungs, pancreas, even the sweat glands, etc. Okay? Okay? So it keeps, what will it do to the mucus? So it keeps mucus hydrated and thin by controlling chloride secretions, sodium absorption and water movement. So obviously if there will be any change in this gene, so chloride secretions will not be there, mucus will become thick. So all the body organs then will be affected. Is that clear? So if there will be mutation, so chloride secretion will decrease, sodium and water absorption will increase. Right? Sodium and water absorption will increase. So the mucus will become thick, viscous and basically sticky. Okay? Okay, so a lung infection can be there, right? Even in the reproductive tract, even in the sweat glands, pancreas, they all will be affected. So due to mutation in CFTR gene, and due to which mucus becomes So it affecting many body organs, so that is why it is the Pleiotropy. That is also I have written, right? So just do it quickly. Go for it. Okay, so lungs, pancreas, they'll be blocked, infections will be there, cough can be there. Done? Done? So that is how it acts. So this is the cystis fibrosis, right? This is the cystis fibrosis. Yes, students, this is the cystis fibrosis. Okay, so then let's go. So this is Pleiotropy. See, even in NCERT it is given. So it also comes under the allelic interactions. So we have so far seen the effect of a gene, right, on a single phenotype or trait. There are, however, instances, means example, where a single gene can exhibit multiple phenotypic expression, where a single gene can affect many phenotypes. Such a gene is called pleiotropic gene, and the mechanism of mechanism is called pleiotropy. Okay, so why this pleiotropy is occurring? Because here you can see in most cases it is the effect of a gene on metabolic pathways which contribute towards different phenotypes. Like one gene, it is making something that is not affecting one metabolic pathway, that is affecting many, right? That is affecting many. Is that clear? Is that clear? So here clearly written the disease phenylketonuria, which occurs in human, right, where the phenylalanine hydroxylase is getting mutated, right? So it is also the example of pleiotropy. But I told you, right, not just this, your sickle cell anemia, cystis fibrosis, they all are example of pleiotropy. And moreover, we have discussed one more thing, and what's that? That is the size of starch grain and the seed shape. Okay, so let's come back to this portion. So see what we have covered so far. See this. We are done with incomplete dominance, codominance, multiple allelism, pleiotropy. We'll discuss a bit about the lethal gene. Then I'll tell you these two. And finally, we will talk about the polygenic inheritance. Right, then I'll give you a short break. Then after that, chromosomal theory of inheritance, which will not take much time, not the chromosomal theory of inheritance. Then after chromosomal theory of inheritance, chromosomal theory of linkage. And that's it. Hai na? Then the sex determination, mutation and the disorders, and we have completed many things from the disorder. Pedigree analysis part also we'll discuss today. Water absorption also increases, beta. Water absorption also increases. Okay. So let's discuss the epistasis, lethal genes. So done. So what do you understand with the lethal word? See, there are some genotypes in the nature that can cause the effect severe to the death, means that can cause the death. So in that case also the phenotypic ratio get modified. Instead of 3:1, it becomes 2:1. So lethal means all that changes that can cause the death. So there are many genotypes, like in the plants, even in the animals. I'll give you the example of hemophilia. We know that hemophilia, which is the bleeder's disease, it affects the male, है ना? And in the females are the carrier. Females cannot, in the case of females, we don't see hemophilia. Why? Because both the X chromosomes, if they get the defected allele, then it is a lethal combination, right? Then it is also a lethal combination. Are you getting my point? Are you getting my point? Right? So that is also the lethal combination. Is that clear? Is that clear? Okay? Done? So I'll give you the example as well, so you can write down that lethal genes. So what are the lethal genes? Just write down. So term lethal is applied to all such changes that can cause effects right, that can cause effects, right, severe enough to cause death, severe enough to cause death, right. So this is the important line here, the ratio 3:1 get modified to 2:1. Okay. Next, if a dominant allele is lethal, then it is lethal in its both homozygous dominant and heterozygous dominant. Exactly. Are you getting my point? So this is the meaning of lethal. So this line is important. Remember this. So the phenotypic ratio, which is 3:1, it will become 2:1. Is that clear? It will become 2:1. Okay. Okay. Done. So example, if I have to give you any example here, so you can consider the example of in maize, the amount of chlorophyll is controlled by so in maize the amount of chlorophyll is controlled by a recessive allele G, which is lethal in its homozygous recessive form. That's it. So if it is GG, then normal. If it is GG, then normal. But if it is GG, then it is going to be lethal. That's all. Okay? That's all. Okay? So this is the meaning of lethal gene. So that is it. So next part that we have to discuss is, let's discuss a question. But yes, before discussing the question, I just forgot this. See, students, when you talk about the blood group now, in the case of blood group C, AB blood group shows codominance. Overall blood group shows multiple allelism. Here you see dominant recessive relationship. है ना? So in blood group, multiple allelism, codominance, dominant recessive relationship, both are there. Okay? All three are there. Okay? So that's what you have to remember. Understood? That is what you have to remember. Done? Okay. So next point, so let's solve the questions. So see, F2 generation in a Mendelian cross showed that 1:, both genotypic and phenotypic ratios are same. So what does it represents? Yes, please. What? Yes, please. What should be the answer here? Very simple question. Yes, very simple question. What should be the answer? So here you are talking about the genotypic and phenotypic ratio is same. So obviously even it is in the codominance. Codominance maybe you will see F2 generation is having monohybrid cross with incomplete dominance also shows that. Isn't it? Isn't it? Even monohybrid cross with incomplete dominance, it also shows the genotypic ratio and phenotypic ratio as same. Chaliye, if two persons with AB blood group marry and have sufficiently large number of children, okay, two persons with AB blood group, these children could be classified as A blood group, AB group and B blood group in 1:2:1 ratio. Modern technique of protein electrophoresis reveals presence of both A and B type protein. So see, initially it feels like that question is difficult, but actually it is not. It is just the example of codominance. Now the next question. Yes, please give it a try. So see, blood group of father is A, blood group of mother is B, then predict the progeny's blood group. Obviously it's simple. See, father either can be this or mother can be this. So what are the chances here? So even without drawing this now, students, you can just simply answer this. AB blood group, right? B blood group is also there. A blood group is also there. O is also there. So all are possible actually. All are possible, right? All blood groups are possible. Then in a marriage between male with blood group A and female with blood group B, the progeny had either AB or B. See, now one partner is having A blood group, another is having B. See, they are saying they have either AB or B. So in that case we can simply say that this can be the possibility. Matlab, this can be the possibility. You can draw the Punnett square. So because here AB, here also B, here AB, here B. So even if you are confused in such question, you just pick up the options. Just say let's pick up this option, right? Just pick up this particular option. So if you pick this option, right, you can just check it. You'll get your answer. Then go for the next one, because if you will go for the next one, it will always be AB, right? Then if you go for the next one, you can get AB or A. So as per the question, this one is right. So you can just pick up the answer and then you can try to answer it. Okay? And see, the blood group part, I already told you among the blood type, how many different genotypes and phenotypes are possible, right? So this is again, you can check, very simple question. Okay, I want you people to answer this particular question. Question number 60. Take your time and answer this. Quick. Take your time and answer this. It is easy, trust me. You can just look at the option and you can just tell me. It's easy, try to answer it. Everybody quick. Students, see, what are they saying? They are saying that in a cross between pure breeding pea plants, right? They say RRYY. You can write it in this way also, right? And even this is fine, RR. You are crossing it with this. You know that F1 is going to be this. And if you have to go for the F2, you'll get the ratio 9:3:3:1. And for that 9:3:1, this is going to be your round yellow. This is going to be your round green. This is going to be your wrinkled yellow, and this is going to be your wrinkled green. Can you see that? So just look at it. What are they asking? They are just asking for a very simple thing, that in F2 how many will have yellow color? 9 here, 3 here. 9 plus 3, 12. Sorted. Okay. Question number 78. That's it. Rest we have to solve it. Question number 78. Question number 78. In a test cross involving F1 dihybrid flies, okay, more parental type offsprings were produced than the recombinant type. It is a test cross, more parental type than recombinant. This indicates, yes. Give it a try. It's easy. How come D? Guys, they are saying they are more parental type. More parental type is a sign of linkage. Chalo, let's solve it later. So, let's talk about the non-allelic genes, and we are going to start with the complementary genes first. Okay? So, there are many non-allelic interactions, right? But we will discuss these two, right? Here we will see in non-allelic interactions, which are also known as intergenic interactions, right? Non-allelic interactions, which are also known as intergenic interactions. What will you see? Here we are going to see that 9:3:3:1 ratio is modified. Like here, this ratio, we are not going to get this ratio. This ratio is modified. Are you getting my point? This ratio is modified. So we will start first with the complementary genes, right? We will start first with the complementary genes. See, the word itself is explaining, you are talking about the complementary genes, right? We are talking about the complementary genes. So here now, first of all, here the character that you are discussing is not like, here you are talking about more than one, like more than one gene, right? That is why it is the intergenic interaction, like two different genes are there, okay? Two different genes are there. So no doubt they are the two different genes, but we will see that how are they going to affect a character. So basically what is the point here? In the complementary genes, for the expression of a particular character, you need two different genes in their dominant form. If they are not in their dominant form, you are not going to see, right? You are not going to see the phenotype. So here, two pair of non-allelic genes, two pairs of non-allelic genes in their dominant form are essential in their dominant form to form a particular character, to express a particular character. Are you getting my point? So what's the point here? Two pairs of non-allelic genes are essential in their dominant form to form a particular character. Is that clear? Is that clear? Right? Both genes in their dominant form can produce that effect and cannot produce it separately. Like if these dominant genes, if they are separately present, they will not produce that effect. They can only do it together, right? So both type of genes should be there in the, yes please. So both, remember, this is the keyword here, both the genes should be in dominant form. Should be in dominant form. Okay, so I'll give you the example here. Okay, so both the genes should be in the dominant form. Okay, so let's suppose example, flower color in sweet pea. So it's not garden pea, it's sweet pea, so it is Lathyrus odoratus. So let's suppose, students, if capital C and capital B are present, flower is going to be colored, okay? But if there is capital C and small p, flower colorless. If there is small c and capital P, flower colorless. Capital C and small p, flower colorless. So flower will be colored only when they both are in their dominant form. Why is it so? Because the gene C, right, like it will act on a substrate, it will form a color producing substance, and then the product of gene P will act on that color producing substance, and finally the color will form. Are you getting my point? And then finally the color will form. So that is how it works. That's why both the alleles should be, genes should be in the dominant form. Is that clear? Right, both of them should be in the dominant form. Just for an example, we have the raw material. On that raw material, the product of gene C will act, and then it will form a chromogen. Chroma means color, gen to generate. Then gene P, then anthocyanin, right? That purple kind of color will be there. And anthocyanin is a water soluble pigment, remember. Okay, so this is how it is. You have a raw material, right? What do you have? You have a raw material, a gene C, the product of gene C will act on it, will convert it into a color producing substance. So now let's suppose if there is a color producing substance, but this is still not the color, right? So let's say if there is only capital G, like capital C, then chromogen will form, but further color will not form. And if there is only capital P, no capital C, then this chromogen will not form. So that's why we need both in the dominant form, right? Both in the dominant form. So when you are going to make the cross, you people can see how can you start. See, you'll start the, like usually how do we start? Just say here you have capital C, small p, small p, small c, small p, small p. Or I'll suggest you this. Let's suppose colored, because both are capital, cross it with this colorless. Then F1 colored. Then for F2 we have to do the selfing. है ना? So So capital C, capital P, capital C, small p, small c, capital P, small c, small p, and here also capital C, capital P, capital C, small p, small c, capital P, small c, small p. Now what's going to happen? See. See this, look at this, right? Now this one is going to be colored because both are dominant, right? Now tell me this one, colored or colorless? Yes please. This one, colored or colorless? Yes everybody. Both dominant are present. This is again colored, colored, colored. But here one dominant, other is not. Colorless, colored, colorless, colored, 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 colorless, 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 colored. So that's what we have, right? That is what we have. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 are colored. And rest 7, because total is 16, are colorless. So here the ratio is modified from 9:3:3:1. It will become 9:7. Is that clear? So that is the meaning of complementary genes. Okay, that is the meaning of complementary genes. One more is there, that is epistasis. Fine. One more is there, that is epistasis. So again it is also the example of non-allelic interaction, intergenic interaction. So epistasis, it's a very simple phenomena, students. Epistasis is actually, so here what is happening, one gene is suppressing the expression of another. Means there is one gene which is not able to express itself when the another gene is present. Like for an example, let's suppose A, there is a gene A, this gene is epistatic gene. Epistatic gene means it suppresses the expression of another gene, and that another gene, this is will be hypostatic gene. Okay, so B, which gene it is, whose expression is getting suppressed. Okay, this is the gene whose expression is getting suppressed. A is the one who's suppressing the expression. Okay, so this is going to be your hypostatic gene. So what am I, this is, sometimes it happens in the families na, like in front of your father or the mother you are not able to express yourself, but when they are absent then you are like pak pak pak. Same is the case here. Okay, so A is the epistatic gene which can suppress, right? It suppresses the expression of another gene. Okay, so that gene whose expression is getting suppressed, or I would say you can also use the word masked, right? That is the hypostatic gene. So let's suppose, so this is what the epistasis is, in which one epistatic gene is suppressing the expression of, right? One epistatic gene is suppressing the expression of another hypostatic gene, right? So during epistasis, epistasis. One epistatic gene masks the expression of another hypostatic gene. Got that? Yes, please. Got that? I'll give you the example. So there are two types of epistasis. One is the dominant epistasis. And accordingly the another is the recessive epistasis. So in the case of dominant epistasis, right, in the case of dominant epistasis, the epistatic gene should be in its dominant form. That is, so here epistatic gene should be in its hypostatic form, sorry, in its recessive form. That is A. So that is the meaning of dominant and recessive epistasis. Okay, that is the meaning of dominant and recessive epistasis. So epistasis where one gene is masking the expression of another, the gene whose expression is getting masked is called the hypostatic gene, and the one who's masking its expression is called epistatic gene. So it is of two types, dominant and recessive. So you people can see when it is the dominant, the epistatic gene should be in its dominant form and otherwise in recessive. Okay. Done. Okay. Done. So if I have to give you the example, I'll give you the example of summer squash. I'll show you the picture as well. So example mein fruit color of summer squash. That is your cucurbita, peppo. So here you have to know that capital Y, yellow color, yellow fruit color. Small y, green fruit color. And W, it is epistatic gene over capital Y and small y. It gives white fruit color. Am I clear? So what will it give? It gives the white fruit color. Is that clear? So how can you start it? Again, you'll start with the same story. See, the epistatic gene is in the dominant form. So it will not be yellow, it will be white. Then you are going to cross it with this homozygous recessive. So here, because it is dominant epistasis, this is not epistatic here, so it is going to be green. Then you talk about the F1, WWYY, again capital W, epistatic. So again here also fruit color will be white. Now just make the Punnett square. See, write down the gametes, capital Y, see this. So students, just look at it very carefully. When, see, capital W is present, then the fruit color is going to be white. Here also it is going to be white. Here also it is going to be white. I am not writing the genotype, that you write later. Here also, because wherever capital W is there, this is going to be white. this is going to be white and this is going to be white. Understood? But when you see this part, this is also white, this is also white, but here, look at this. This is going to be the genotype. Now, because this epistatic gene, it should be in its dominant form. It is not dominant. So, here you will see yellow color. Here also you will see yellow color. Then white, white. Then again here you will see yellow color, but here you will see green. So 3 yellow, 1 green, we are left with 12. So this is the ratio. So this is the example of dominant epistasis. Just have a look. And if there is any doubt, please ask me. So for dominant epistasis, the ratio is going to be 12:3:1. Quick. 12:3:1. So it is going to be 12:3:1. Do it. Okay, so here you can see the graph as well. See white, green, and this is 12:3:1. So this is the dominant epistasis. Okay, this is the dominant epistasis. 12:3:1. Done. So next part is polygenic inheritance or the quantitative inheritance that we have to discuss. So let's discuss this. Okay, polygenic inheritance. Students, I have seen students getting confused, and I always repeat this part, right? I have seen students getting confused in these 3 terms, though they should not, but I have seen this. See, when it is the multiple allele, the multiple allele to still easy. Here you are saying single gene having more than 2 alleles. Are you getting my point? A single gene is having more than 2 alleles. Is it clear? Meaning of multiple allelism. Then there is Pleiotropy. A single gene is having more than many phenotypic effects. Is having many phenotypic effects, or in another word, a single gene controls the expression of more than one trait. Is that clear? So it's a gene is single, but it can control more than one effect. Now the topic that we are going to discuss is polygenic inheritance. Right, this is the polygenic inheritance. So poly means many, genic, genes. So here simply you are saying many genes, like more than one gene is controlling, or even 2 genes is controlling the expression of a single trait. Here genes are many of a single trait. Now why do we say it is the post-Mendelian inheritance? Because as per Mendel, one gene will control one character, but here we can see one gene is controlling more than one character. Here we are saying that as per Mendel, one gene is controlling one character, right? But here we are saying many genes, more than one genes are controlling the expression of a single character, right? More than one gene is controlling the expression of a single character. So this is how I can just illustrate it. Just a minute. This is what you people can see. See beta, here gene, right? This is the gene. There is gene A. It is controlling character 1, character number 2, character number 3. Here, how can I show this? There are many genes. Genes A, gene B, gene C, but they all are influencing one character. That's how these two, they vary. Can you see that? That is how these two, they vary. So here you have one gene, but many characters are there. Just a minute. Here you have one gene, but many characters. Single gene is affecting many phenotypes or more than one character or one trait, right? A single gene controls the expression of more than one trait. So here, character, here gene A, gene B, gene C, so many genes, they are controlling one character. That is how these two topics, they differ. Are you getting my point? That is how these two topics, they differ. Is that clear? That is how these two topics they differ. So here traits are controlled by 3 or more genes, right? Trait is controlled by, like more than one gene means 2 genes are controlling one character. So more than one gene, or I would say traits are controlled by more than 2 or 3 genes. Are you getting my point? So this is the polygenic inheritance. So polygenic inheritance and pleiotropic clear? Polygenic inheritance and pleiotropic clear? That what exactly it is? So here single gene but more than one phenotypic effects that they are controlling. Many genes but they are controlling just one character. So here if I have to give you the example, the best example I can give you is human skin color. Right, human skin color, it is controlled by 3 genes. Human height, right, human height, it is also a polygenic inheritance, right, it is also a polygenic inheritance. So in polygenic inheritance, number of dominant alleles, they'll decide the phenotypic expression, okay. This is C. Mendel studies describe that mainly those traits have distinct alternate forms such as flower color, either. But if you look around, you'll see there are some traits which are not distinct in their occurrence. They are in the gradients. What is the meaning of this line? That they are spread across a gradient. What is the meaning of this line? Like, I'll give you the example of human height. You cannot say that the person who's having 6 feet is tall or 3 feet is short. No, you'll see some people, they are 5.5, 5.4, 5.6, 5.7, 5.9, right? 5.9 is also tall, 6 is also tall, 6.1 is also tall, right? 6.5 is also tall. So there are gradients. Let's talk about the skin color. You cannot say that this individual is fair, this one is dark. If there are fair individuals, you'll see that their skin tone, it changes. Like you can call 2 people that they have the fair skin, but still you'll see the differences in that. So why gradients are there? And why these gradients are there? Right, it is decided by the number of dominant alleles. Just for an example, we do not just have tall or short people as 2 alternatives, but a whole range of possible heights, right? So such genes or such traits are generally controlled by 3 or more genes, and that is why, students, we are calling them as the polygenic traits. So poly means many genes. So here, instead of more than one gene, let's specify more than 2 or 3 genes. I would say 3 or more. Three or more genes are controlling the expression of a single character. Understood? Right? So it is in the form of gradient. So again, I can frame a question from this part as well. Right? I can simply frame a question from this part as well. Right? In polygenic inheritance, many genes control one character, true or false? Yes, this is true. Right? Second, in Pleiotropy, we see gradients, right, in traits. No, this is false because here we'll see in polygenic inheritance mainly. Right? Polygenic inheritance mainly. So yes, you can frame the question from this part. Okay? Done? So, besides means in addition to the involvement of multiple genes, here we also account the influence of environment. So yes, in the polygenic inheritance, environment plays a very important role. Right? I'm not writing it separately. You can simply write it from the NCERT. You can just write down. So human color is also, skin color is also the example. And in the polygenic trait, the phenotype reflects the very important point, contribution of each allele. That is, the effect of each allele is additive. So more number of dominant allele means the more will be that expression, less number of dominant allele will, less will be the expression. Like if you talk about the human skin color, right? I'll take the example AABBCC. So that person is going to be Negro, that is the complexion is going to be very dark. Now, let's say AABB and CC. Here you are going to be fair, that is going to be, like I'll say, white. Are you getting my point? So, white. So now when you take F1, so F1 will be this na, heterozygous individual. So it is intermediate. This is what you have to understand. It is intermediate. You will call it as mulatto. So here F1 individuals, they are not like any of the right, their phenotype is not similar to either parent. So how can you get F2? You know that, right? The selfing here. And students, I believe now you know how to make the gametes here. So that is how you can simply make the gametes for this tri-hybrid cross. You can make it very, very easily, right? You can simply make it very easily. ABC, AB small c, AB capital C, sorry, small c, so I have already discussed this Falkline method with all of you. So one parent will form 8 gametes. So 64 zygotes, right? 64 zygotes can form. How many zygotes can form? 64 zygotes. So what are the important points? That here the environment plays the role, and here you will see the additive effect. Each allele is going to contribute, isn't it? Isn't it? So in simple words, in polygenic inheritance, right? In simple words, when I'm writing polygenic inheritance, so I can say three genes or more than 3, right? 3 or more. Genes control a trait or a character. Then environment also influence the phenotype. right? Environment also influence the phenotype here, right? The phenotype reflects the contribution of each allele, I would say dominant allele. So that is additive effect is there, addition, add-on is there, right? There will be the additive effect, right? Add-on will be there, isn't it? Isn't it? And so traits are spread across gradient, right? In polygenic inheritance, we see traits are spread across gradient. So that is how it is, right? So each number of dominant alleles, basically I can say, though we have covered this, number of dominant alleles decides intensity of the character. Okay, number of dominant alleles decide the intensity of the character. Am I clear? They will decide the intensity of the character. Am I clear? Yes, please. So how can you check the, here also we have a formula. So you can check number of phenotypes here. It's not the genotype. Do not confuse it with the multiple alleles. So it's going to be 2n plus 1. N is equals to number of polygene, right? N is equals to number of polygene. So I'll give you the example here. So if let's say, if n is equals to 3, then it will be 7. So 7 phenotypes. So there will be 7 phenotypes. So n is equals to 3, matlab number of polygene in human skin color. So in human skin color, how many phenotypes you people are going to see? You people will see 7 phenotypes. Understood? 7 phenotypes you guys are going to see. Okay? Right? Okay? So we can also check Chalo, I'll write it here only. Done? So I hope it's done. Done, students? So that is how you can also make the flow charts. You can write the important points. So here to check the contribution of here now we can also check the contribution of a single gene, right? So what you have to do to check the contribution of a, just a minute, single gene. Maximum value, you can take the maximum value minus minimum value to the number of contributing alleles. That's how we can check, right? Total number of contributing alleles, or I would say the genes. Is that clear? So we can see the effect of the single gene as well. So that will come as total number of contributing genes rather, not the alleles, because here we are checking the effect of single gene, contributing genes. So that's how you can check it. That's how you can calculate it. So that is it. So now I'll show you the expression and even the MCQs here. See, here the F1, it doesn't resemble any of the parent. Okay. Now when you will take the F2, right? Now when you will take the F2, you can just simply see that. See this. Just say one side 8 gametes here, 8 gametes here. Total 64 possibilities are there. Okay, total 64 possibilities are there. Can you see this type of distribution here? It is, see, you have to understand one thing, because each allele is contributing, right? So obviously there are the gradients here in the phenotype, plus number of dominant allele will decide. Like for an example, if I talk about the skin color, right? Let's suppose if a person is having AA, B, B, C, C, like dominant alleles are 1, 2, 3, 4, 5. 5 dominant alleles. Then there is another person which is having 3 dominant alleles. There is another person which is having 2 dominant alleles. Obviously, the one having 2 dominant alleles will be fair in comparison to the one having 5 or the 3. So, each allele is contributing. Is that clear? Is that clear? Is that clear? Okay? Sure? So, one minute. So that's what we have in the F2, right? You can make the cross. In the F2, how many? We just checked it. 2n plus 1 is the formula. 7 phenotypes are there. Negro black, very dark brown, mulatto, light brown, very light brown and white. So you can also check it by, right? So this is what we will get. So it is 1, 6, 15, 20, right? Then 15, 6, 1 again. This is how you can write 1, 6, 15, 20, then again 15, 6, 1. So this is going to be the ratio. So obviously Negro black 1 by 64 because total number of 64 possibilities are there. Very dark brown 6 by 64. So that's how you can check the probability. Okay, that is how you can check this part. Okay, so this is all about the polygenic inheritance, right? Right, this is all about the polygenic inheritance. Now we'll start the chromosomal theory of inheritance, but before that, let's practice few questions, okay? Let's practice few questions. Yes, so you can see question number 82. That's easy, trust me, that's easy. Just solve question number 82, right? It's easy, just read it once and just solve question number 82, then I'll give you one more example of polygenic inheritance. Yes, please, give it a try. Give it a try. Quick. See, human skin color is controlled by several gene pairs. So let's suppose there are 3 gene pairs on different chromosomes and obviously that they are present on different chromosomes. So there are 2 alleles, an incompletely dominant that codes for melanin deposition. So see, if a very dark skinned person marries a very light skinned woman, what will be the chance that their offspring will have very dark skin? I told you here, right, very dark skinned men with very light skinned women. F1 will be mulatto, intermediate, right? So there are no chances that F1 will be either of the parent. So the answer should be 0 here. Okay, 0 here. Okay, so that's how we can check it, right? So this is all about the, yes, so this is all about the polygenic inheritance. One more example other than human skin color or human height you can write. So I'll write the examples here. So So examples, human skin color, and the kernel color is also there, right? Kernel color in the case of, kernel color is also the example here, that is in the case of wheat, that kernel color in wheat is controlled by 2 polygenes. Okay, 2 polygenes, understood? That's what you have to keep in your mind. So this is all about the polygenic inheritance. So we will start chromosomal theory of inheritance after a short break. So let's start again at 12:10 a.m. So very good morning after 8 minutes. Okay, done? So rest we'll discuss later. Hi guys, welcome back. Welcome back. Good morning, good morning. How are you all? What's up? Okay, so let's start the next part. So next part that we have to discuss is the chromosomal theory of inheritance. Okay, chromosomal theory of inheritance. So before starting the chromosomal theory of inheritance, let's have a quick revision of the cell cycle and cell division part, like cell cycle and cell division, a portion of that we are going to revise. Okay, so see, overall this is what we have learned with time. Just wait a minute. Wait a second. Okay. So see, this is the part that we have already covered. Ki in 1900, we know that the three scientists, Carl Correns, Eric von Schemack, Hugo de Vries, they rediscovered the Mendel's work, right? Independently. They were not working together. Independently, they were experimenting. They got the Mendel's papers and they basically rediscovered the Mendel's work. So also by the time, due to the advancement in microscopy, of course, you know that if we are if we are able to see the internal structure of cell, that is also because of the microscope only. So because of the advancements in the microscopy, right, students, scientists were able to carefully observe the cell cycle and cell division, and due to which what happened, due to which there was the discovery of structure in the nucleus, right? There was the discovery of structure in the nucleus that appeared to be double and divide just before each cell division, right? This appeared. So these were called the chromosome. Are you getting my point? So basically, right, at the time of Mendel it was not there, but later on, due to the advancements in microscopy, what happened, students, right, the scientists, they have observed that nucleus, they also divide before the division, and in nucleus something is dividing. What exactly is that thing? That is the chromosome. Chroma means color, so these are the colored bodies, right, students? So by 1902, okay, the year is important here. By 1902, the chromosome movement during meiosis. What is meiosis? Meiosis is the reductional division. Meiosis is the reductional division that will reduce the chromosome number to half. So by 1902, the chromosome movement during meiosis had been worked out, right? So scientist name, the another important question. So Walter Sutton, Theodore Boveri. Walter Sutton, Theodore Boveri. They noted that the behavior of chromosome was parallel to the behavior of genes and used chromosome movement to explain the Mendel's law. Important. This line is important. So by 1902, the chromosome movement, right, the chromosome movement during meiosis, right, by 1902, chromosome movement during meiosis. Mr. Venkatesh, my studio is still not working. I'm taking the class from somebody else's studio, right? And once my studio will start working, I'll record the human health and disease. It is also my job to fulfill this, okay? And I know that and I'll do it, right? So, like, I don't know what is the issue with the students, like whatever chapters we discuss, they don't focus on that chapter. They keep on asking the other chapter. Like some students in the chat, they were like, ma'am, start biotech. Ma'am, start this. Arey yaar, I've already started something, you at least focus on that part, na? Okay, so this is important. So as I said, by 1902, the chromosome movement during meiosis, it had been worked out, and Walter Sutton and Theodore Boveri, they noted that the behavior of a chromosome was parallel to the behavior of genes and used chromosome movement to explain the Mendel's law. Is that clear? Is that clear? So before going forward, right, so in the short notes, do include that. So the year as well as the scientist name. So scientist name, what we discussed there for the scientist name. So it is Theodore, it is Walter Sutton and Theodore Boveri. Okay. And actually they are the one who gave the chromosomal theory of inheritance. And later on, Morgan provided the experimental proof for this chromosomal theory. So let me discuss that with you all, and even I have started the class with this part only. Just so you know that we have we have 23 pairs of chromosome. Firstly, we know that students that in the interphase, what do we have? We have G1, S and G2, isn't it? We know that it is the S phase in which DNA replication occurs. In which DNA replication occurs. We know that, right, in which DNA replication occurs, right, that is DNA amount. If initially it is 2C, it will become 4C. And number of chromosomes remains same. Number of chromosomes will remain same. Are you getting my point? Number of chromosomes will remain same. And what happened in the G1 and the G2 phase? Here the RNA synthesis, protein synthesis, all that things will occur. So basically we know the process, right? Just say let's suppose if it is the interphase, the genetic material, the nucleus is in the resting state. So actually it is not resting, metabolically it is quite active. It's just that you will see that chromosomes are in this form, right? They are basically decondensed. They are not even condensed, right? So if I have to show you this, so you can take it in this way, that these are the chromosomes, right? These are the chromosomes here. So they are in monad form, students. They are in monad form, means single arm is there, right? Means single arm is there. So you know that this is the position of gene on the chromosome. It is a locus. Let's take the example of A. Let's take the example of B here, right? I'm randomly taking one example. Let's suppose this is chromosome number 1, chromosome number 2. Now in the S phase, what's going to happen? DNA will get duplicated. DNA will get duplicated, yes or no? So that's how these two arms are going to be, right? Right. So this is A, it is getting duplicated. See, this is A, right? Here, this is B, this is B. Let's suppose here you have, this is another chromosome D and this is E. So here also you will have D or D or E or E. Up to this part all clear, guys? Up to this part all clear? So how we started? We are talking about the nucleus. We are saying in that nucleus, obviously you have the genetic material. So no doubt in the interphase, the genetic material is not condensed. You don't see such chromosomes there or such chromosomes there. But because I have to explain you this, so I'm saying just imagine it in this way. Okay. So let's suppose you have two different chromosomes, chromosome 1 and 2. They are having, I'm talking about these 4 genes. Just giving you one example, just talking about these 4 genes. So in the S phase, the DNA content will get doubled. So obviously if this arm is getting duplicated, if it is having A, this will also have A. If this is having B, this will also have B. If this is having D, this will also have D. And if this is having E, this will also have the E. Are you getting my point? So that is what we have. That is what we have. So that is what we have. Now we know the division part, isn't it? Now we know the division part. So if you people remember this, right? I'm giving you a quick recap also. You know that meiosis, it occurs in 2 rounds, right? So in the meiosis, that is how we discuss, right, students? There is meiosis 1, okay, which is having prophase 1, which is having PMAT, right? PMAT is the trick here. Prophase 1, metaphase 1, anaphase PMAT, anaphase 1 and telophase 1. Prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay. So, and we know that there is meiosis 2 as well. So that meiosis 2 will have prophase 2, metaphase 2, prophase 2, metaphase 2, anaphase 2, telophase 2. Right? This is the division that we have. And students, just for your quick revision, the prophase 1 is having 5 substages, that is LZPDD, right? That is LZPDD, that is leptotene, zygotene, pachytene, diplotene and diakinesis. This is what we have. Now, I'll take the example of 2 chromosomes only. Okay? Just even let's suppose I have shown you this. So I'll take the example of few chromosomes so that I can explain you. Just say these are the phases that we have. In the anaphase 1, I'm writing here, there is the separation of bivalent. And in the anaphase 2, there is the separation of sister chromatids. There is a separation of what? there is the separation of sister chromatids. So I'll give you the example here. Let's take the example of a cell having nucleus, having, let's suppose, 4 chromosome, right? Let's suppose 4 chromosome. So you know that if there will be, if this is the meiosis one, so in the prophase one, in the zygote, the pairing will be there, crossing over will be there, all the things are there. So just imagine, I'll start with the metaphase 1 directly. I'll start with the metaphase 1. That's going to happen. That's going to happen. This is, let's suppose this is the spindle fiber. This is the spindle fiber. So that's how it is going to get arranged. Are you getting my point? That's how it is going to get arranged. Is that clear? Is that clear? That is how it is going to get arranged. So now let's suppose here you have A, this side is A. Let's suppose here small a is given. Just giving you one example. Let's imagine capital B, here small b, small b. Let's imagine here small d, here also small d and small d is here. Giving you just one example. Okay. This is the metaphase 1. After metaphase 1, what do you have? You have anaphase 1. So as I said in anaphase, there is the separation of this bivalent. Okay. So here, the chromosome that we have, can you just see this? So here you have A, B, then C, A, my bad, B and B. This is getting separated. It is anaphase 1. And there is another chromosome also. It was having DD. Let's suppose this is also having DD. And let's say here you are considering G or G and small g or small g. Just giving you one randomly examples, right? Because I'm just trying to show you something. So when there is the anaphase, can you just see that? When there is the anaphase 1, this is the metaphase 1, right? It is in pair. Like here, this individual is having capital A, small a, this capital B, small b. Now, small b. Now in anaphase 1, this is getting separated. Like 2 cells, like 2 daughter cells will form now after meiosis 1. 2 cells will have. This cell is having capital A, capital B, small d, capital G, small a, small b, small d or G, small g. This is what we have. Now we'll proceed from this part. Now, let's suppose this is the metaphase 2. Let's suppose this is what? This is the metaphase 2 for the same. This is the metaphase 2. Now, in the metaphase 2, what you people will see? We had, I'll pick up this example for metaphase, okay? Just here we have A, B, let's suppose D and G. I think this was the thing na? This is what we have here. Are you getting my point? Now in the anaphase 2, what is going to happen? In the anaphase 2, what is going to happen? They'll split the sister chromatids. They'll split. So here you will have capital A. Here you will have capital A, capital B, small d. This is capital G, small d or capital G. So now, what am I trying to tell you here? We started with the division part. See, let's suppose you are talking about the chromosome. You started that chromosome 2n is equals to 4. So obviously in metaphase 1, 2 pairs will be there. They will have the genes. So in anaphase 1, they see this bivalent is getting separated. Like if I had A or A, this A or A is getting separated. A is moving to a different cell. Another A is moving to a different cell. So remember law of segregation, right? Right? So from this pair, one gamete is receiving capital A, another is capital small b. One is receiving capital B, another is small b. So small d, both are same. So they are passed to the different gametes, and GG also. They are passed to the different gametes. Are you getting my point? They are passed to the different gametes. Are you getting my point? Then we are moved to what? Then we are moved to the meiosis 2. So in meiosis 2, let's say you have picked up this cell. This is having A or A. Now you are splitting it in this way. You are splitting it in this way. So basically what have we learned? We have, even from the anaphase 1, we are clear that A or A, like from the pair of allele, right, each gamete will receive only one allele for a character. That is what we have seen there. Here though it is basically the DNA content that will get divided. Now the another thing is, okay, fine. Here we started like this, that here one cell is having A, B or D or G. So of course that can also happen, that if, let's suppose, here you have A or B, right? Here you have A or B. There are high chances that this chromosome, capital D or small g, can go there. Wait, let me show you the picture. Let me show you the diagram. Right? Let me show you the diagram, then it will be very clear to you. So see this. When you look at this part, right, G1, and then you know that in S phase it got doubled. So that is why in G2, because here in G1 they are simply showing monad, and here in G2 they are showing dyad. Okay, so in anaphase 1 we are separating this yellow and the orange one, this red and green one. We have separated that. In meiosis 2, in anaphase 1, we have even separated the sister chromatid, which results in having these 4 germ cells. Now, this is one thing. So this is explaining us the law of segregation. So it is telling us, right, it is telling us, just see, A or A, let's suppose B or B. So obviously this A, let's suppose this is B, this is B. Now here you have separated this A and A. Okay, right? I can use capital or small also, you can use this one also. Okay, so it is having A, B, A, B, A, B. So you cannot say that ki each cell is having more than one, like these two are not moving together. That's what I'm trying to tell you. So this is showing us law of segregation. So chromosomal segregation is telling us about the law of segregation as well. Now come to this part. This is also the diagram now. This shows us the independent assortment. How come independent assortment? Just see, orange, yellow, green or red. So orange, see, orange, yellow, they to definitely get separate. Orange is going with the yellow one here. This yellow is going, orange is going with the green one here. Yellow is going with the red one. But when you look at this orange can also go with this green, this yellow can also go with this red also, right? Just a minute. One second. Meiosis 1. So meiosis 1, meiosis 2. So exactly. So matlab this is what we are saying. I was right. See, here this side. Matlab here we are showing that on one side we are showing, as per the separation, this orange or green, right? But here orange and red can be there, yellow, red or yellow and green can be there. So in simple words, this orange can go with the green, it can go with the red also, this can go with this and this also. So this is the independent assortment, because as per independent assortment, what we have discussed, that the segregation of, right, one pair of alleles, just let's suppose here it was capital R, or let's suppose small r, here it was capital Y, let's suppose small y. So capital R will not affect the segregation of capital Y only. Capital R can go with capital Y, as you can see, but capital R can also go with small y. So this is showing us the independent assortment. Is that clear? Yes, it is showing us the independent assortment. What is it showing? It is showing us the independent assortment. That's what I'm trying to tell you. So this part is showing us the law of segregation. This part is telling us about the two possibility, one long orange short green chromosome, long yellow short red at the same pole. Here one long yellow short red and long yellow and short green chromosome at the same pole. So it is telling us about the independent assortment. So let's discuss about this theory in detail, right? Let's discuss about this theory in detail. So it is the chromosomal theory of inheritance given by Sutton and Voveri. So they basically compared, they basically showed the parallel behavior in between the chromosomes and genes. As we see that after the anaphase 1, right, during anaphase 1, the bivalent chromosome is getting separated. So it is clearly showing that these chromosomes, they are in pair, genes are also present on them in pair, they are getting separated. Law of segregation. Plus we have seen that colored part as well. So let's suppose if there is one chromosome, it is paired with another, right? So obviously paired one to getting separated, but let's suppose let's suppose maternal, paternal, like there is the independent assortment. When we say na capital R can go with capital Y or small y as well, so obviously capital Y is coming from one parent, small y from another. Like let's suppose one parent was having capital R and capital Y, but it is also going with small y, that is coming from another parent. So it shows the segregation. Okay, it shows us what? It shows us the segregation. So by 1902, the chromosome movement during meiosis had been worked out. So Walter Sutton, Theodore Vaubery, they showed this parallelism, right students? Right students? Okay. So here you can see that I'll discuss the postulate separately, that Sutton and Vaubery, right? But Sutton and Vaubery argued that the pairing and the separation of a pair of chromosome would lead to the separation of pair of factors as well. We have just discussed it now. How? So in anaphase 1 it is getting separated. So Sutton united this knowledge and he gave the chromosomal theory of inheritance. But beta, the experimental proof of chromosomal theory of inheritance is provided by the father of experimental genetics, Thomas Hunt Morgan. Okay, so here you can see. So Sutton and Vaubery, what they argued, that pairing and separation of chromosome would lead to the segregation of pair of factors that they carry. Okay, so they united this knowledge and gave the chromosomal theory of inheritance. So this is another important question, that who provided the experimental verification of proof of this theory? That is Thomas Hunt Morgan. So he applied the Mendel's law on the Drosophila, he saw the variation, then he gave chromosomal theory of linkage. Okay, he gave the chromosomal theory of linkage. Is that clear? So let's discuss the Sutton and Vaubery, let's discuss their points. So what they said, that chromosomes like alleles, they also occur in homologous pair. We know that, right? 2n is equals to 46 means 23 pairs are there, right? 23 pairs are there. So 2n is equals to 46, so it means 23 pairs are there. How many pairs are there? 23 pairs are there. So what am I trying to tell you here, right? What am I trying to tell you? All I want to say is that, see, chromosomes are in pair, means they have the genes, they are also in pair. So that is what we are saying. Now paired homologous chromosomes, they separate during meiosis 1. So if I have to be very specific, I'll say anaphase 1. So each gamete will receive only one chromosome or allele of each pair. So I hope that a diagram is clear in your mind. Okay. Next, this separation forms the Mendel's law of segregation of paired factor, already discussed. Then chromosomes of each pair, right? Jaise, right? So see this. So this one can go with this one can go with this one as well. So this random segregation is also there, right? This random segregation is also there. Is that clear? Is that clear? So chromosomes of each pair, right? Each pair segregate independently of other pairs. Like it is not mandatory. Just say, let's suppose this blue one will always be like, yeah, I'll stick with the green only. Just say, let's suppose this blue one is having the capital R and this one is having the capital Y. This one is having the small r, this one is having the small y. So it is not mandatory. Capital R will be like, yeah, I'll stay with the capital Y gene. So you understand it in the form of a gene, but gene is present on what? Chromosome. So capital R is like, I can go with the small y as well, right? So how is it possible? So it is possible because you are talking about, like we discussed it in law of independent assortment. So genes, so law of independent assortment, in law of independent assortment you talked about the genes, and here you are talking about the chromosomes, but chromosomes are ultimately located on genes only. So that is why the chromosomes of each pair, they segregate independently of other pairs during anaphase 1. So during anaphase 1, so it is saying that during anaphase 1 when it is segregating, right? So chromosome of each pair is segregating independently of the another pair. Is that clear? Any doubt here? Any doubt here? Done? Then Mendel proposed that factors of each pair are segregated independently of other pair in their distribution into gametes. Then homologous pair form from two parents come together, right, in the zygote, and that is formed by the male and female gamete fusion. So each contributing one homologous chromosome. So Mendel held that paternal maternal factor come together in the zygote by fertilization. So see, these 2 points are also relatable. So here they are saying that homologous chromosome at the time of fertilization they will come together, and Mendel also stated that these factors from the mother and the father will also come together during the fertilization. So chromosome retain their structure and individuality throughout the life, same as for the factors. So this is how we have compared. Right. So let's start. So we have discussed two pictures. One was showing the law of segregation, in which we see that in the anaphase 1 that how the bivalent gets separated. So bivalent is separating in anaphase 1 is clearly indicating that basically what is happening, the alleles from a pair are getting separated. Plus there are other chromosomes also. So it is not mandatory that if in parent 1 this and this chromosome was together, then this cannot go with the maternal one. This can go. This can go. It doesn't matter that they'll go to the same gamete only. They can go with the other one also. Is that clear? So that is what they have explained. Ki paired homologous chromosomes separate during meiosis 1, to be more precise, anaphase 1. Each gamete will receive only one chromosome or allele of each pair. Then this separate form the basis of, this separation forms the basis of law of segregation. Then chromosome of each pair segregate independently of the other pairs during anaphase 1, right? Chromosomes of each pair segregate independently of other pairs during anaphase 1. Is that clear? Chromosomes of each pair segregate independently of the other pairs during anaphase 1. So this, and Mendel also proposed that factors of each pairs are segregated independently. Right, जैसे capital R can go with capital Y and small y, same as for the chromosome, both are compared. Then next line, कि homologous chromosomes, right, they'll come closer during fertilization, same way, factors also come closer. So chromosome also retain their structure and individuality, and Mendelian factors also. So that is how this theory is related. So students, here there is a comparison, right, there is a comparison table also given. So जैसे A, see, it occurs in pairs. Segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. Okay. Independent pairs segregate independently of each other. You are talking about the genes here, and here occurs in pairs. See, you can compare. Segregate at gamete formation and only one of each pair is transmitted to a gamete. So one pair of chromosome segregates independently of another pair of chromosome. Like this can go, doesn't matter that if it was with in parental generation together, so they can go with this and this also. That is what I'm trying to tell you. So this is your gene, that is your chromosome, this is what you need to know, and this is the diagram, right? So this one, law of segregation, and this one, law of independent assortment. Any doubt? Any doubt? Yes, please. Any doubt? Guys, quick. Those who are actually here to study something, so they can ask me the question. Is that clear? Sure? Okay. So next thing that we have to discuss is linkage. But before linkage, let me tell you, let me remind you that who provided the experimental proof of the, right, who provided the experimental proof for the, this theory, right, chromosomal theory of inheritance. It is the Thomas Hunt Morgan. Okay? Okay? Sure? Chaliye, let me write it. So chromosomal theory of inheritance. So I'll share the notes with you all. Okay, I'll share the notes with you all. Okay, I'll share the notes with you all. So this is what you people can see. See, experimental verification of chromosomal theory of inheritance is provided by Thomas Hunt Morgan, number 1, and his colleague, right? So they worked with the tiny fruit flies Drosophila melanogaster having 2n is equals to 8. Yes, please. Having 2n is equals to 8. You can even write it down in your book as well. So why Drosophila melanogaster? They could be grown on simple synthetic medium. Students, whenever we conduct any experiment on a plant or an animal, we see the feasibility. Like we said, pea plant is easy to cultivate. So when you experiment on the animals, so then we have to see which one is easy to deal with. So this one can be easily grown on simple synthetic medium in lab. This is the number 1 feature. Then they complete their life cycle in about 2 weeks. Second important point. And a single mating could produce large number of progeny. Number 3, here the sexual dimorphism was there in the male and female, which we can easily distinguish, and types of heredity variation, right, that we can see with the low microscope was also there. So 2n is equals to 8 here, and remember in Drosophila, right, just like humans, this is the type of sex determination. In Drosophila, the sex determination is just like the humans, XX and XY. So XX is female, XY is male, right? So this question is important. So chromosomal theory of inheritance done. This part is done. So A is your gene, B is your chromosome. So again I'm saying this. So this is your law of segregation. This is your law of independent assortment, where orange can go with green, orange can also go with red, yellow can go with red, and yellow can also go with green, right? So do write this in your notes. And I told you why Drosophila, okay? So do write it in your notes. Who provided the experimental proof of chromosomal theory of inheritance? So the answer is Thomas Hunt Morgan. What's the answer that we have here? That's Thomas Hunt Morgan, and he is also the father of experimental genetics, right? He's also the father of experimental genetics. And as I said, what you people need to remember here, right? This is the Thomas Hunt Morgan, father of experimental genetics, and he experimented on Drosophila melanogaster. Okay, do not forget this. So easy to grow, more number of progeny, melanogaster, right? Sexual dimorphism, where the variations are easily seen, right? And here I'm writing it again. This Th Morgan, this Thomas Hunt Morgan, right, student? This Thomas Hunt Morgan, when you will see that Thomas Hunt Morgan gave the chromosomal theory of linkage. He was the one who gave the chromosomal theory of linkage. Am I clear? Right? Am I clear? Done? Chromosomal theory of linkage. Okay. So firstly, let me tell you about the linkage. No doubt when Morgan, when he experimented, guys, listen to me. So when he experimented, on the Drosophila melanogaster, when he applied the Mendel's law, he was expecting the ratio, but he didn't get the same ratio. Okay, he literally didn't get the same ratio. So after that, when he observed, when he analyzed, then he came to this thing that there is one more, like, then he came to this chromosomal theory of linkage. But Morgan was not the first one who gave the concept of linkage. When we talk about the linkage, what exactly is the linkage? Like, this is something, this Morgan has explained, but what exactly is the linkage? Linkage means something is linked, something is attached. So here, what are you talking? You are talking about the genes, right? Here, what are you talking? You are talking about the genes, right? you're talking about the genes which are present on the same chromosome, that are the linked genes, right students? And that linked genes, are you getting my point? That linked genes, they'll stay together during the inheritance process as well. That's also we have to discuss. Is that clear? Right? So now we are starting the experimental genetics. We have already discussed who has provided the this what do we say, the experimental proof. Now we will write with the definition and we will discuss it in detail. Okay? So the tendency of the genes present on the same chromosome, the tendency of the gene, right, present on the same chromosome to inherit together during inheritance or the transmission process, right, to inherit together, that is also done. Process is called linkage. So the tendency of the genes, right students, to stay together, right, to inherit together, or I would say to stay together in transmission process is called the linkage. Is that clear? So I'll give you the example. Just say, let's suppose this is a chromosome, right? This chromosome is having the genes. They are present in a linear order, of course. Let's suppose A, B, C, D, E, right, F. This is what I'm doing. So these genes present on same chromosomes are called linked genes. Is that clear? Genes present on same chromosomes are called linked genes. Am I clear, students? Am I clear? Genes present on same chromosomes, what do we call them? We call them as linked genes. That's what you have to remember, right? Are called linked genes. अब जैसे let's see. Let's suppose this. These are two different chromosomes. So जैसे E and 4, just giving you example, are non-linked genes, are not linked as they are present on different chromosomes. So the genes which are present on same chromosome are called linked genes, and the one which are not present on same chromosome, they are not linked, non-linked genes. Okay? So this is what you have to remember. So I have just given you one example, that let's suppose you are taking the example of E and 4 because they are present on two different chromosomes, so they are not linked. Okay? So that is what linkage is, the tendency of the gene to stay together during the transmission process or during the gamete formation or during this inheritance process is called the linkage. Is that clear? Is that clear? So this is what linkage is. So yes, we will also discuss the types of linkage, in which we are going to talk about the complete linkage, which is very rare in nature, and we will also talk about the incomplete linkage, right? So I'll tell you the concept of linkage and incomplete linkage as well. So complete linkage, as I said, rare. You'll see it in male Drosophila or female silk moth. That's it. That's it. Very rare. Right? And then comes the incomplete linkage. So we will discuss its types. But before that, I'll give you a kind of history of this linkage part. So linkage was not something that was first reported by Morgan only. Watson and Punnett, right? Two scientists were there. Watson and Punnett. Students, they experimented on sweet pea. They experimented on what? On sweet pea. That is your Lathyrus odoratus. Okay? So two scientists, the name Watson and Punnett, they experimented on sweet pea. What they did, they experimented on sweet pea, Lathyrus odoratus, right? Lathyrus odoratus. Is that clear? Is that clear? This name I have already discussed, odoratus. Okay? So somewhere around 1905 to 1908, so they experimented. What they selected the pollen, right? They selected the two characters they selected the pollen, basically the pollen length and the flower color, right? This is what they selected, okay? So this is the experiment that I have mentioned here. Here you guys can see simply, right? Who experimented first, students? Who experimented first? The way slides are moving, they are just telling me, Ambika, you need a coffee. Okay, just a minute. So Watson and Punnett, right, they experimented on sweet pea, right? I'll tell you here. They experimented on sweet pea, which we are calling as Lathyrus odoratus. Okay? Lathyrus odoratus. They experimented on this. So they did an experiment, right? They like flower color and the pollen length. Like here, they were like blue flower color with long pollen grains, and the another was red flower color with round pollen grains. Yes, है ना, red flower color with round pollen grains. This is what they experimented. Now they experimented to get the, so Watson and Punnett, they experimented. So they selected the characters, two characters. So they were trying to get dihybrid test cross ratio, right? They tried to get what? Dihybrid test cross ratio. So when they tried to get the dihybrid test cross ratio. ठीक है बच्चे? Okay. Third point, tried to get dihybrid test cross ratio. So expected ratio, you tell me, what should be the expected ratio? Today we have discussed the test cross, na? So expected ratio was what? Expected ratio was 1:1:1:1:1, right? This was the expected ratio, 1:1:1:1:1. But the ratio obtained, it was 7:1:1:7. Can you believe that? Expected ratio was 1:1:1:1, but the ratio obtained was 7:1:1:7. Like more parental types, less recombinants. So it was a question mark to them. Look at this cross here. Blue flower, long pollen grains, BBLL, right? Red flower, round pollen grains. This is the F1. Now dihybrid test cross ratio. So you know that in the test cross ratio, they'll hybridize it with the homozygous, they will cross it with the homozygous recessive parent. So they got blue long, red round more, like 7 and 7, and blue round and red long less. Matlab it was not 1:1:1:1, which was expected ratio, but the observed ratio, the ratio obtained was this, right? This is the observed percentage frequency. More parental type, less recombinants. So this was a question mark for them. They were like, how is it possible? Why is it possible? Why is it happening? Then they gave the concept of this, coupling and repulsion. Like they were not able to explain that coupling and repulsion are basically the two phenomena of linkage only. They put forward, right, Watson and Punnett, Watson and Punnett, they put forward the theory of, coupling repulsion theory. Are you getting my point? they put forward the coupling repulsion theory. So what is this coupling? What is this repulsion? Coupling, like something is coupled or staying together. Repulsion, something is repelling. So what is it? So coupling means when two dominant genes or two recessive genes, just say let's suppose here this capital A capital B, capital A capital B, matlab when this dominant one is coming from one parent and this recessive one is coming from another parent, like parent 1 is having capital A capital B, parent 2 is having small a small b, right? So basically what am I trying to tell you here? When two dominant genes or two recessive genes are inherited together more frequently than expected. When two dominant genes or recessive genes are inherited together more frequently than accepted, this is the coupling. So dominant pair, just say AB, right, tends to pass together, it is moving together to the next generation. When there are gamete formation, AB is passing together. So parent produce more AB and AB gametes. Like parents are having, one parent is having A or B, both dominant, one is having both recessive. So this one is making, rather than capital A or small b, this one is making more A and B and A and B. This is what they said. In the repulsion, when a dominant gene and a recessive gene come from different parent, right? Just say let's suppose you are, take the example of parent like this, right? Take the example of parent like this. Just say, see guys, see that though I am just in general showing you. If I have to show you the homologous pair also, let's suppose one parent is having capital A capital B, another parent is having small a small b. That's how I can show now, because if it is a homologous pair, then what is going to be same? What is going to be same? The locus, when the gene is, right? This gene will occupy the same locus there. And the gene will occupy the same locus there. Okay, so this is what we have. Or I can write it in this way also, right? So this is still kind of F1. So let's suppose or you can say this also. In any way you can define it. This is what I am trying to tell you. But in simple language I am saying that A and B, both the dominant alleles are on same parent, recessive are on same parent. This is what I am trying to tell you. That's it. Right? That's it. Okay. So we can also mention it in this way. Just say, ye dekho. One parent is having this, another parent is having this. So this one is making AB only, this one is giving us AB only. More these gametes are there. So this is coupling, right? This is coupling. But when they are coming from different parents, just say let's suppose AA, BB and AA, BB. Or I would say, if I have to show you here, this, if I have to show you it in the form of pair, right? So this parent will give which gamete, A or B. This parent will give small a or small b. So this is showing the repulsion here. So when a dominant gene and a recessive gene, a dominant and a recessive, right? Dominant and a recessive, right here. So when a dominant gene and a recessive gene come from different parents, right? Dominant is coming from different parent, recessive is coming from different parent, right? So they tend to stay apart, okay? Thus combinations like AB, but here this is the combination actually better that we have to consider, right? Because one chromosome is having dominant form of one gene and another is having and of another gene it is the recessive form. So one will form AB or AB appear more frequently with this. So overall, what this Wetson and Punnett said, they have put forward the coupling repulsion theory. They observed that if capital A, capital B, dominant, if they are more, or recessive, they are like capital A, capital B, or small a, small b, if they are inheriting together, if they are like, if you are able to see such gametes more, then it is the coupling. But when let's suppose combination is like this, capital A is going with capital A, right? Capital A is going with small b or small a is going with capital B, this is the inheritance, right? This is the inheritance, right? So you can show it in this way, and this one, so you can show it in this way. That one, the one that I mentioned, that is basically for the hybrid, right? So they will tend to enter the different gametes. Is that clear? Is that clear? So they provided this, but they were not able to explain the reason behind that why it happened. Then in 1910, right? Then in 1910, so here you can see Betson and Punnett did not know about chromosomes and crossing over much. Like they were not that much aware. Later, Thomas Hunt Morgan, right? They couldn't explain, I would say, they couldn't explain the exact reason behind this. So later, Thomas Hunt Morgan in 1910 explained that genes which are located on same chromosome, that shows the linkage. So coupling means you are talking about the linked genes in the cis configuration, right? And repulsion means linked genes in the trans configuration, right? Right? And even Morgan explained, even one more thing that you can write here is that when you talk about the Morgan, so Morgan was like, he explained that coupling and repulsion are two aspects of that coupling and repulsion are two aspects of linkage only. They are two aspects of linkage only. Am I clear? Right? They are the two aspects of linkage only. Am I clear? Quick. Just a minute. I am looking for my charge. Okay? Okay? So this is what they explained. So Watson and Punnett, you know, they were the one who first time mentioned that, and then this is the thing. Okay? I hope I'm perfectly audible now. Hello. Okay, so this is the coupling. Done. So Morgan later explained that these are the two aspects of, right? Morgan later explained that these are the, this coupling and repulsion, they are the two aspects of the linkage only. Okay? Okay? Done? Sure. So now here what are we saying, that see in the coupling you'll see linked genes in the cis configuration. So let me show you the cis configuration, that what exactly is the cis configuration. Just a minute. Cis configuration. Okay. So cis configuration means, students, that A and B, both the dominant are like this, recessive are like this. Okay. And when there is the trans configuration, then in that case what you people will see, this will be A and B, this will be small a and capital B. Am I clear? Am I clear? Sure. Okay, so this is the cis configuration where both the dominant and recessive are together in the trans. Dominant, recessive, recessive, dominant. That's how they are. Is that clear? That is how they are. Is that clear? So this is the cis and the trans configuration. Okay, this is the cis and the trans configuration. Am I clear? Sure. Done. So Watson and Pinnett, they were not aware about this thing, right? They were not aware about this thing. I would say they were not aware about how they were not able to explain this, basically the linkage and repulsion in a proper way. Okay, they were not able to explain it, right? They don't know the reason behind. Okay, they were not aware of the reason behind. They have just put forward the theory of coupling and repulsion, but in that coupling and repulsion, as I said, so this is how they have explained. When two dominant genes, right, they are on same chromosome and the recessive on same, they will inherit more frequently. It's a kind of, what is it? It is a cis configuration, right? So AB will go together, right? This AB, yes, this AB will go together, the small a and small b. And here, right, you can say capital A, small b, or small a and capital B. This is the trans configuration, okay? This is the trans configuration. So that is what this, yes, that is what we discussed here. So this is the Watson and Pinnett, and so Thomas Hunt in 1910. So the year is important again, right? Year is important. This is important. So Morgan explained that coupling and repulsions are two aspects of linkage, cis configuration and the trans. Now, okay, so Morgan carried out several dihybrid crosses in Drosophila. Okay, he experimentally applied all the Mendel's law on the Drosophila, right? So crosses were similar to the dihybrid crosses carried out by Mendel in peas. So, just here, he hybridized yellow body with white-eyed females to brown body, red-eyed males and intercrossed their F1 progeny. So, instead of getting 9:3:1 ratio, he got the different ratios. So, that is why he came to this thing that there is some other phenomena which we are calling as linkage, okay? So now I'll explain you this linkage and the independent assortment thing. First of all, guys, I'll tell you what complete linkage is. What is the complete linkage? So complete linkage means you will see 100% parental combination. What is the meaning of complete linkage? That you will see 100% parental combination. Like let's suppose if your parent one is having capital A, capital V, or another parent, so you will see the ditto, the same thing. Like next generation will 100% like the, right, the next generation will be 100% like the parent generation, right? This is the complete linkage. What is it? It is the complete linkage. Yes, everybody? What's that? It is the complete linkage, and as I said, it's rare, right? You cannot say that complete linkage is something very common. No, not at all, right? Not at all. So here in complete linkage, you will see the Yes, please. What will you see? The 100% parental combination. And what do you think? Is it possible? Is it possible? Is it possible? Is it possible? Done? Beta, complete linkage, I have repeated it 2 or 3 times that complete linkage is rare, right? You cannot see normally this complete linkage thing. You are not going to see normally. Whenever you talk about the complete linkage, it is like 100% parental combination, which is very rare, right? So 100% parental combination, so this is rare. So you will discuss the male Drosophila and female silk moth. So where will you see this? Male Drosophila and in the female silk moth, this is where you are going to see the 100%, right? 100% parental combination. And whenever you see the linkage partner, just say let's suppose this, you are crossing it with this. Whenever you see a sign like this, sign like this, it is showing the linkage. So this parent will form this gamete only, this parent will form this gamete only. Fine, you got this as you got this as F1, right? Now if you are even trying to do the selfing here, okay, if you are trying to do the selfing here also you will get more parental types only, right? Just say let's suppose AB or AB will stay together, yeah, AB or AB will stay together. So rather than, so here one parent is not giving you 4 gametes, rather 2 gametes here, 2 gametes here, right? 2 gametes here, 2 gametes here. So then you can just simply cross them and then again you'll see that what are you going to get. Again the parental types will be obtained. That's what I'm trying to tell you, okay? Okay. So incomplete linkage. When I'm talking about the incomplete linkage, so more parental type, less recombinant. This is going to be the incomplete linkage. More parental type and less recombinant. Now, before going forward, I'll discuss 2 aspects with you. Just say, let's suppose there are 2 things. One is the independent assortment, right? And you know that wherever there is independent assortment, linkage will not be there. And there is another thing, linkage. Students, linkage will occur when your genes that you are studying, when they are present on the same chromosome. Are you getting my point? When this linkage will occur when the gene present on the same chromosome. When the gene that you are studying is present on what? When it is present on the same chromosome. Is that clear? When it is present on what? When it is present on the same chromosome. So then you will talk about the linkage. But when there is the independent assortment, let's suppose now you are taking the A, this A is here and this B is here. When this is the case, right, this is the independent assortment. Do not confuse it with the cis and trans. In the case of cis form, right, in the case of cis form, what I just discussed, I just told you that this is the arrangement, the dominant together from one parent, recessive together on another chromosome means from another parent. Trans may one dominant, one recessive, one dominant, it was like that. Now I'm saying, right, I discussed the incomplete linkage with you all and I told you that there are more parental type and the less recombinant type. Now see, in independent assortment, right, genes taken are like whatever genes you have selected or the we are studying are on different chromosome. So because they are on different chromosome, that is why independent assortment is taking place. If they will be on same chromosome, then it will not be independent assortment, it will be linkage. Here genes studied are on same chromosome, are on same chromosome. Now because it is on different chromosome, so here you people will see, right, capital A can go with capital B, capital A can go with small b, small a can go with capital B, small a can go with small b. So you are getting 4 different type of gametes. 4 different type of gametes. Are you getting my point? But here, because A and B together, so one gamete will be this, another will be this. So here you will see 2 types of gamete. Are you getting my point? Are you getting my point? Because here you see the linkage. So in the complete linkage, say AB, this will give you this only, this will give you this only. And now in the next generation also, because we have defined there is a complete linkage. So these are the gametes that you are going to get if this is a complete linkage. Then of course, when you will cross them, you will again get the parental genotype or parental phenotype only. Right? So that is why I am saying that it is going to be the complete linkage. Is that clear? Right? It is going to be the complete linkage. Is that clear? Okay, done. So independent assortment, 4 different types of gamete, 2 different types of gamete, incomplete or complete linkage. Is that clear? Chaliye. Now let's give it a try. Just for an example, I'll tell you. I am saying you have gene AB, AB. I'm telling you this gene is showing you the linkage. So tell me the gametes here. What are going to be the gametes? Gametes are going to be AB and AB only. Right, now let's suppose I am telling you just say AA, BB or simply, chalo, even if I'm not writing it in this way, but if there is the linkage na, I'm saying this, I'm specifying this, कि complete linkage is there. So then you will be like AB and AB. Okay, AB and AB. And I told you how to write it. Right, so for the linkage, that's how we write. Okay, that's the gamete. Now let's say, this is the case that I'm giving you. I am telling you that these 2 genes, they show independent assortment, and this one is showing linkage with them. Then tell me about the gametes. What will be the gametes here? Then tell me about the gametes. What will be the gametes here? Wait. I'll set you solve quick. Quick. Yes? Guys, quick. Guys, quick. Is it done? See, if they are showing independent assortment, it means capital A can go with capital B, right? Capital A can go with small b, small a can go with capital B, small a can go with small b as well. Plus, here, because this is showing the linkage, so it will not leave, na? Matlab ABC can be there, ABC, okay. Got that? So two are showing the independent assortment. So if two are showing the independent assortment and I am saying that this is showing the linkage, and let's suppose, firstly let's keep it simple, and then let's suppose these are both CC and I'm saying linkage. So A, B, A, B, A, B or A, B. So you have to consider the C always, because we are clear that it is showing the linkage. Now, one more thing. I am writing AA, BB. I am saying these two are showing linkage. Okay. This one is showing independent assortment. Then how will we go for it? How are we going to solve it? Yes, please. Then how are we going to solve it? Do it quickly, everybody. Do it quickly. Is it clear? Now see, if I'm saying this is showing independent assortment, right? But this one is showing linkage. So B will go with C, small b will go with small c, right? Right? So this is independent assortment. So obviously capital A can go with this also. So capital A, capital B, capital C. Capital A can go with small b and small c. Small a can also go with capital B, capital C. Small a can also go with small b and small c. So this is what you got. Now the same case that we have discussed here, let's change it again. AA, BB and CC. Then these two independent assortment, right? These two independent assortment, and I am saying that this one is showing the linkage. Now again, right? If it is showing the linkage, right? This one is showing the linkage, but with what? With this part, right? So A, B, and A, B or A or B, right? So this will go with capital C, and then same you have to write for the small c as well. Okay? Small c as well. Okay? Okay? Done? So in this case, let's try one more. So let's try one more part, and in that part, let's change the game a bit. Let's suppose this one is showing the linkage. I think this one we didn't discuss, and this one is showing the independent assortment. So A, B, A, B. So C, add capital C, A, B, small c, A, B, small c as well. Isn't it? Done? So that's how you can check it, guys. That's how you can check it. And this is about the linkage. So complete linkage, as I said, is rare. And then we talked about the incomplete linkage, more parental type, less recombinant. So you can add one more thing here, right? That recombination frequency, that recombinant will always be less than or equals to 50%. Now, this is what I'm going to simplify. Equals to 50%. So, gamete part done. Okay, gamete part done. So, now this is how we are going to proceed. We'll pick up the Morgan's experiment, but here I would like to specify a few things. See, I told you that whenever you consider the genes which are located on different chromosome, then we are saying that there will be the independent assortment. I believe this much part is clear to everybody, right? This much is clear to everybody, I believe so. And that's a good thing. Now, we are saying complete linkage. Now see, let's suppose you are talking about the inheritance of two genes and they are very close to each other. And here also you are taking the inheritance of genes which are little away from each other. You know that here linkage strength will be more. Is more, right? Here linkage strength is less. Why is less? Why? Because here you see more distance, right? The distance is more. Here the distance is less distance. So here more distance means chances of crossing over are also there. Are you getting my point? Chances of crossing over are also there. Is that clear? Is that clear? Sure. So independent assortment no doubt occurs when the genes are present on different chromosomes. But if by any chance if they have too much of distance in them, let's A and B, then obviously there are high chances that crossing over will occur and there are high chances that they will get separated. Okay, so it is the distance. Are you getting my point? It is the distance which plays a very important role here, right? It is the distance which plays what? Which plays a very important role here. Is that clear? So more will be the distance, less will be the linkage, less will be the distance, more will be the linkage. So if two genes, they are very close to each other, they will show the complete linkage, they'll inherit together, right? If two genes, they have a distance in them, then obviously there are the chances of crossing over as well. Is that clear? Is that clear? Chaliye, one more example here. Now let's suppose you are taking the example of complete linkage. Incomplete linkage, A and B, है ना? They are very close to each other. Then obviously they'll inherit together. They'll show complete linkage, these two genes, right? Here, this gene A and B show complete linkage, है ना? So if you talk about the inheritance of AB or AB, right, it will not be like a dihybrid cross. जैसे dihybrid cross is because of the ratio 9:3:3:1 is because of the independent assortment. But here, because you will see the complete linkage, here you will see the complete linkage. So dihybrid cross ratio will become monohybrid ratio, right? Monohybrid cross ratio, right? This will become monohybrid cross ratio, right? Because they are moving together like a unit. We are not separating them. We are considering them that they will go together only, right? They will go together only. So gene A and B shows, right, linkage here. So I am saying, and yeah, they'll show complete linkage in a sense because they are so close, so they'll move together only. So in that sense, they have the complete. So dihybrid cross ratio will become monohybrid. So in the case of linkage, what you people will see, dihybrid test cross ratio one is to 1 becomes 1:1 because both the genes are moving together only. Is that clear? both the genes are moving together only, right? So now let's discuss the Morgan's experiment. So what Morgan experimented on what? Morgan experimented on Drosophila. So these are the characters that he selected. He experimented on Drosophila. So these are the characters that he selected. So see, one is wild type. For wild type you use the symbol plus. For mutant type minus. Matlab wild type, nature has formed it. It was formed as such. And with time when there are the mutations, then obviously the phenotype changes. So there you can use the negative. So Morgan selected body color of Drosophila, eye color, wing size. Matlab these are the three things that we have to discuss. Body color, eye color, wing size. Which three characters are we studying right now? Body color, wing size, and the eye color. Now wild type body color is brown and the mutant type is yellow. You can even put negative, or eye color is red here, mutant type is white. Okay, so wing size is large here, this is miniature. So we are specifying that body color and eye colors are linked. These two genes are linked, and eye color and wing size, this is non-linked. So if it is linked, there are high chances it will inherit together to the next generation and they will comparatively less. So how can we check it? You come to the cross that we see in our NCERT, right? You come to the cross that you see in your NCERT. So see, there are two crosses. Here they are talking yellow and white. So what exactly are they talking? What are these two characters that they have taken here in, right, that they have taken into consideration here? That they have taken into consideration here? Tell me. This is the yellow, right? So yellow and white. So yellow is what? Yellow is the mutant type. This is also the mutant type. So what is it? So this yellow here, you are talking about the body color. What are you talking? Body color. And this is basically what? This is basically the eye color, right? This is basically the eye color. So this is the cross number 1. Okay, this is the cross number 1. Am I clear? This is the cross number 1. Am I clear? Yellow and white. So you are talking about the body color and the eye color. Okay? Okay? And here you are talking about the eye color and wing size. Eye color and wing size. So basically whatever information I have shared with you initially, like I am talking about the Morgan's experiment, I have clearly specified that Morgan experimented on Drosophila. These are the crosses that I am discussing right now. I am saying I have selected body color, eye color and wing size, right? So body color, eye color and wing size, right? I am saying body color and eye color, they are showing linkage, and eye color and wing size is not linked. So how can I check that? You just look at it very carefully. This is the yellow, this is basically the body color, this is basically what? This is basically the eye color. Can you see the distance in these genes? Even the mutant, wild type and the mutant type, the gene distance is less, right? Here if you observe this, you'll see distance is, what will you see? You'll see the distance is less here. Now here, you are talking about the eye color and the wing size. Distance is more. Distance is what? Distance is more. So obviously if distance is less, so we'll see more linkage. More linkage means, what does it mean? It means, more linkage means more parental type. More parental type. Here comparatively less parental type. Here comparatively less parental type. So now I'll move to this part. So you can see in the cross A, parental type is 98.7%, recombinant type is 1.3%. Cross B, eye color and wing size, parental type is 62.8%, recombinant type is 37.2%. So because here the distance between two genes, it was more, here it was less, here it was more, here it was less. So basically you will see more parental type but less recombinant type, less parental type in comparison to this, and more recombinant type in comparison to this. Is that clear? Is that clear, everybody? Done? So that is, from this we concluded that eye color and the body color is showing the linkage, matlab more close to each other, and eye color and the wing size is little far. So accordingly these ratios they vary. Is that clear? Sure? Any doubt? So this is the chromosomal theory of linkage by Morgan. So Morgan has given that. Okay. He also provided the experimental proof for the chromosomal theory of inheritance, repeated it multiple times because I know majority of students make mistake. So genes that show linkage are situated on the same chromosome and are called the linked genes. Done. They remain bounded together and can't be separated during inheritance except by crossing over, because they are present on same chromosome. Linked genes, only crossing over can separate them, and only when the distance is more. So distance between linked genes determines the strength of linkage. So genes are arranged in a linear fashion on chromosome. So strength of linkage is inversely proportional to the distance. Important. So in crossing over, the strength of linkage is directly proportional, but in linkage, the strength of linkage and the distance is inversely proportional, but in crossing over, distance and the strength of crossing over is directly proportional. So if distance between two genes increases, strength of linkage decreases, and if they are moving apart, crossing over will occur more frequently. Then arrangement part, though I have already told you. Coupling and repulsion, I don't think that I need to repeat this. Done, students? So these are the gametes here. So here AB, here this. So if on a chromosome, if there are linked genes and if the distance is too much, then yes, they can assort independently. But these are the few things that you have to remember. So even in the NCERT, guys, if you will check the ratios, so yes, the question can come from this ratio part, right? So here you can see they have mentioned that genes white and yellow were tightly linked. So 1.3% recombinant. So body color and eye color it is, right? So that's what, and that's how they have discussed. Okay. So now see, Morgan's student, this is the next topic that we are going to discuss. Alfred Sturtevant used the frequency of this recombination between gene pairs on same chromosome, right? Just I told you, incomplete linkage means recombination, but it will never be more than 50%. It will be less than or equals to 50%. So Morgan has used this frequency of recombination by Morgan to check the distance between gene pairs on same chromosome as to measure the distance between genes and to map their position. Okay. So today these genetic maps, do underline this in NCERT, these genetic maps, physical maps or chromosome maps, they are extensively used as a starting point in the sequencing of whole genomes as was done in the case of HGP sequencing, right? So cross is done. Now we will discuss about this chromosomal mapping, okay? But before chromosomal mapping, we'll also discuss the linkage maps, linkage groups. That's the next topic. And then I will discuss, right? This is the last topic, this is the linkage group, last topic of linkage, then chromosomal mapping, sex determination, mutation, right? Disorders may, you after mutations, you'll get to know about Down syndrome and all, Klinefelter, this is what we have discussed because, and hemophilia of course, that inheritance patterns, right? We'll discuss that, right? We will discuss that. So, ma'am, please remove water bottle from there. What is the issue with the water bottle? Chalo, so I'll be back in a minute. Till then you just, I'll be back in a minute. So Okay. Class is still going on. We have the topics. So next is the linkage group. So what do you know about the linkage group? What do you know about the linkage group? Anybody having any idea? Yes, please. So students, a linkage group, basically a linkage group is the group of genes which are located on the same chromosome. Obviously they'll inherit together as well, right? So a linkage group is the group of genes. A linkage group is what? They are the group of genes that are present on same chromosome, right? That are present on same chromosome. They are present on what? They are present on, yes please. They are present on what, beta? They are present on same chromosome and tend to inherit together, right? That tend to inherit together as they are physically linked and do not assort independently. Now what's the meaning of that? I am saying it's a group of genes. It's a group of genes that are present on the same chromosome. It is the group of genes that are present on the same chromosomes that tend to inherit together as they are physically linked and do not assort independently. I'll give you the example. Just say we humans, we'll take the humans females. How many chromosomes do we have? We have 2n is equals to 46, right? So basically we have 23 pairs. How many pairs do we have? 23 pairs. So 22 pairs are autosomes. One pair is what? Is of sex chromosomes, which are also known as allosomes. Yes or no? Which are also known as allosomes. Are you getting my point? So autosomes and allosomes. Now what I want to tell you, see, these are the homologous pair of chromosomes. Just say, let's suppose if this is one homologous pair of chromosome, definitely they will have the same gene. If here you have the gene for height, here also the gene for height will be there. If here you will have the gene for eye color, here also you will have the gene for eye color only. So the genes which are present on one homologous pair, they are basically making one linkage group because they are the same genes, right? They will inherit together as well, right? So if in human female, if you ask me that in human female, because we have XX chromosome also, they are also the homologous pair. So in human female, 23 linkage groups are there. How many linkage groups are there? 23 linkage groups. And when you talk about the human male, because in male, you know it very well, XY chromosome is there. They are different. They don't have the same genes. They are not making the one linkage group. So they will have 24 linkage groups. So 22 autosomes are seen. So 22 autosomes from autosomes plus X plus Y. So males may there will be 24 linkage groups, right? Yes, please, 24 linkage groups, but in the female there will be, yes, so there will be 23. So usually the number of linkage group in an organism is equals to its haploid number of chromosome, right? Because obviously two homologous pair of chromosomes are there. They are making one linkage group only, right? But in the case of human males, because X and Y, they both are different chromosomes. They both have different genes. So they are two different linkage group. Is that clear? So they are the genes located on the same chromosome that tend to, genes located on the linkage group is the group of genes that are present on the located on the same chromosome that tend to inherit together and are physically, right, linked and do not assort independently. Is that clear? So this is about the linkage groups. Now the next thing that we have to discuss is linkage map or the chromosomal maps. Then the next thing that we have to discuss is linkage map or the chromosome map or the genetic map. Okay, so we will discuss this topic in detail as well. So okay, let's discuss this. Just a minute. Okay, so let's go for this. So what are we discussing? We will discuss the linkage map or the genetic map, or sometimes even referred to as the crossover map. So it will tell us the distance between the two genes. And who gave this? Alfred Sturtevant. He was the student of Morgan. So Morgan, he provided this recombination frequency. So on the basis of that, right, on the basis of that recombination frequency, he proposed this thing. So what exactly this linkage map or the genetic map is? So it is the diagrammatic or the graphical, the diagrammatic, comma, graphical representation the diagrammatic, comma, graphical representation of relative distance between linked genes, of relative distance between linked genes, right, between linked genes of a chromosome. If linked genes are there, so it means it belongs to a same chromosome, of course. So if chromosome is called linkage map. Is that clear? Is called linkage map or the crossover, linkage map or the genetic map. Okay, okay. So such linkage maps are the outcomes of crossing over, so also called crossover map. Is that clear? So also called crossover map. Understood? Also called crossover map. Understood? Yes, please. Right? Yes, please. Clear? So any doubt? Yes, any doubt? So Morgan proposed, I'll write it too. So Morgan proposed that the percentage so here we have number of recombinants to the total, number of recombinants to the total individuals into the 100, right? Number of recombinants or total. So he proposed, actually what he did, he proposed the recombination frequency. So that is how percentage of recombination frequency. So the percentage of recombinant in the progeny, percentage of recombinants in the progeny depends on what, students? Depends on the distance, obviously. That depends on the crossing over, obviously, right? So linkage map, the diagrammatic or graphical representation, right? So it is the result of crossing over only. So percentage of recombination frequency we have obtained. So it depends on what? It depends upon the frequency of crossing over between linked genes. Depends on the frequency of crossing over between linked genes. On the frequency of crossing over between linked genes. Between linked genes. Is that clear? Frequency of crossing over between linked genes, right? So as per Morgan, जैसे if let's suppose the recombination frequency is, right? If recombination frequency is is 1%, then distance between two genes is 1 centimorgan or 1 map units. Is that clear? This is what he said. Write down, quick. Done? Sure? Done, students? So 1 centimorgan or 1 map unit. So Alfred Sturtevant. Remember, Alfred Sturtevant. Who was he? He was Morgan's student. Okay? He was the Morgan's student. So he used the distance. Right? So what he did, he used the frequency of recombination so he used the frequency of recombination, right? Frequency of recombination between gene pairs as a measure of the same chromosome as a measure of distance between two genes. Okay, so that is what he got. Understood? That is what he got. Is that clear? So distance between two genes, right? So as a distance between two genes and to map their position. That's how he used, right? So we have to remember this. But there are some facts which should be very, very, very clear. So you can write down recombination frequency is less than or equals to 50%. ठीक है? It is less than or equals to 50%. That is a fact. Okay? That is a fact. Now I'll give you a question. Just let's suppose So I'll give you, now we will solve 2 to 3 questions, right? So let's suppose AB is having a distance of 5 map units, या centimorgans. B and C is having a distance of, or A or C is having a distance of 2 map units. This one is having 3 map units. Now you know the distance. Now tell me about their position, right? How are they? Tell me about their sequence, right? Tell me about their sequence on a chromosome. What will be their sequence on chromosome? What will you see? What will be their sequence on chromosome? Quick. Quick. Yes. Exactly. So see, A and B is having a distance of 5 map units, maximum distance, and A and C is having 2 map units, B and C. So 2, 3, 3 plus 2, 5. It's very simple. 3 plus 2, 5. It's very simple. Okay, chalo. So let's solve few questions on the basis of that. So see here I have added everything. So frequency of crossing over or recombination between two linked genes is directly proportional to the fixed length. So frequency of crossing over is total number of recombinants over total number of progeny into 100. So ye dekho. So AB, if the recombinant is recombination frequency is 15%, so distance is 15 centimorgan. So here I have few questions that you people can clearly see, right? So linkage map of X chromosome, see this. The linkage map of X chromosome of fruit fly has 66 units with yellow body genes at one end and bobbed hair genes at the another. Recombination frequency. So even if the distance is 66 units, I told you recombination frequency will always be less than or equals to 50%. So we will go for the C. Chromosomal theory of inheritance was given by Sutton and Vovry. Who provided the experimental proof? Morgan. Now, so this one will come later. Then this one is done. Gene position of chromosomes were mapped by Alfred Sturtevant. So genetic maps of chromosomes are based on the frequency of, it is based on the frequency of genetic recombination. Done. Genetic recombination. So students, let's practice this question. So out of a population of 800 individuals, okay, how many individuals do we have? 800 individuals. Okay, so yellow round and green wrinkled. What will be the number of yellow wrinkled? Chalo, let's solve it. I'll teach you the crossing over questions as well. So for next 15 minutes, let's solve the question. Quick, let's solve the questions. Quick, read it first, then solve it. See students, so they are saying that out of a population of 800 individuals in F2 generation of a cross between yellow round and green wrinkled, what would be the number of yellow wrinkled? So you know that in F2 they are asking na, so F2 matlab 9:3:3:1. This is the ratio. So they are saying yellow wrinkled. So 3 are yellow wrinkled. Means, see beta, total 16 zygotes were there, right? So 9/16 for round yellow, 3 by 16 for wrinkled yellow, 3 will be for round green, and 3 will be for round green. So obviously it is 3 by 16, it is 3 by 16, it is 1 by 16. So if you have to check this, so 800 ka 3 by 16 you can check and you'll get the answer. So what will be the answer here? Right, so they are asking the yellow wrinkled. They are asking the yellow wrinkled. So solve it and tell me. They are asking the yellow wrinkled. Solve it and tell me. So what should be the answer? 150? Exactly. 150. Okay? Now round seed trait is dominant over wrinkled. Heterozygous rounded seed is cross. See, in the cross they are clearly saying this is the cross. You should be very clear it is homozygous recessive. Right? Homozygous recessive. What is the possible progeny? In such cross where one individual is heterozygous dominant, another is homozygous, it should be 1:1. Now, can we have any option which is no, nothing is, but see, these two are still almost, right? Same. So that will be the answer. Chaliye, next question. A dihybrid cross produces the progeny. See, dihybrid cross, and this is what you have. 240, 754. Beta, just now I told you that you will never get recombination frequency more than 50%. So I'll tell you a story. See, keep one thing in your mind. Just say, let's suppose if you have the 1,000 individuals in the progeny, 500 can be parental type, 500 can be new combinations, but it will never be 600 non-parental and 400 parental. Parental combination will always be more. Now when I look at this cross, what I have to see these two are more. Matlab these are parental combination, these are the recombinants. Firstly, I'll take the total. So it will be So it is 2000, right? We have 2000. So total number of individuals are what, beta? Total number of individuals here are 2000. Now these two are the recombinants. I told you always recombinants are less. So 240 plus 260. So it is going to be 500. So put the formula 500, 2000 multiply by 100. So it is going to be 25 map units. Right, so dihybrid cross is giving the following progeny. This is the progeny. So they are asking the distance between two genes on the chromosome. So that's how we have calculated total number of recombinants divided by total number of individuals into 100. So we got the answer. Chaliye. So now the next part is this. Okay, this is the next part. The distance between genes A, B, C, D is given. The sequence of this gene. Okay. See, I'll start with the maximum one. A and D. They are very far from each other. Right. Okay, then A and B. Let's try to solve it. 26. B and C is 8. If I consider this. A and C, how much A and C? No, it will not help here. Chaliye, let's solve it, guys. Let's solve it. Even I am solving it. So, A and D, they will be at the end. Okay, so this option, D or B? Okay. Done. 40. So it is ACBD, right? It is ACBD, right? ACBD, exactly. Done. So, if this question is visible to you, give it a try. Question number 121. Done? Give it a try. Question number 121. Question number 121. Is it visible? So later on you can just download the PDF and you can just solve them, and wherever there is any doubt, just let me know, okay? So next topic is of sex determination, right? So I'll start with the very basic part that you have to remember. See, in the case of sex determination, I'll start with the, as I said, very basic part that you have to remember. XX, XY, mammals. In the insects, let's be very specific, you'll talk about the Drosophila here. That's what you have to remember. XX, XO. Here XO is male, right? So cockroach, grasshopper, roundworms, they'll show you this. Cockroach, again part of syllabus, so keep it in your mind, it is important. ZZ, ZW. So here male, right? ZZ is basically your male. So here female is heterogametic. See, XX here female. Female is XX, male is XY. So female will form same type of gamete. Female is homogametic, male is heterogametic. Here also male is hetero, female is XX, male is having only one X chromosome. Here you will see the female. The female is actually the heterogametic. So see, even in the case of humans, you take the example of XX, you cross it with XY, right? So what do you see there? Female will make same type of gamete, male will make different types of gamete. So female is having X only. If male will give X, then baby girl will be there. If male will give Y, boy will be there. So it is the male gamete that is going to decide the sex of the individual. Okay, it is the male gamete that is going to decide the sex of the individual. Done? Right? So, but here in the case of birds, reptiles, the female is the one that is going to decide the sex of the baby, right? Because female is heterogametic and here also female is ZO. So these are the examples. So when it comes to haplodiplontic life cycle, you talk about the honeybee having 32 chromosomes in its diploid state, right? So in honeybee, we talk about the concept of drone. So we'll discuss it. Let's go with the NCERT highlights, and because this is the basically the gist that we have covered. Right, now let's complete it by a NCERT. So sex determination, the mechanism of sex determination has always been a puzzle before the geneticists. Right, so initial clue was about the genetic chromosomal mechanism can be traced back to some of the experiments which were carried out in insects. So initial experiments were carried out in insects, highlight that. Then when there was the cytological observations, right, like number of insects, so then it was observed that it led to the development of the concept of genetic or chromosomal basis of the sex determination, because chromosomes, the cytological information, the cell, the chromosome number, structure, they were all observed. So yes, you have to underline this, that Henkin in 1891, he could trace a specific nuclear structure through spermatogenesis in few insects, right, through spermatogenesis, and he observed that 50% of the sperm received this structure through this and 50% did not. So from that we got to know this XO, that in the case of male 50% will receive this X, 50% will receive nothing, right, they will not receive the structure. So Henkin, it was the Henkin who gave it the name X body. Is that clear? So Henkin was the one who gave the name X body. Is that clear? He was the one who gave the name X body. Is that clear? But he couldn't explain its significance. He was not able to explain what that X body is. So further investigation by other scientists led that X body of Henkin was nothing. Matlab Henkin was not aware that what exactly it is. He was not able to explain. But other scientists was like that it is the chromosome, right? It is the chromosome, and they then gave the name X chromosome. So as per Henkin, it was not X chromosome, it was X body only. Later on, it was named as X chromosome. Is that clear? So large number of X like organism, they go for the exotype sex determination, where egg will have an additional X chromosome. Matlab egg, female will have XX, male will have XO there, right? right students? So some of the sperm will have X chromosome, sperm will not have. So this is XO or XO, XX or XO type of sex determination. Is that clear students? Is that clear? Right? So why do we, so because there is the involvement of X chromosome in the determination of sex, so it was designated to be the sex chromosomes, and the rest of the chromosomes were named as autosomes. So I already told you, just in our case also 22 pairs are autosomes, 22 pairs are autosomes, and 1 pair is of the sex chromosomes, which are the allosomes. So grasshopper में there is XXXO type of sex determination. We have already discussed in that particular first slide for this topic. Now further observations was that, so see here they have mentioned mammals, they have XY or XX type of sex determination. Is that clear? Is that clear? So males in mammals will have XY and the female will have XX. Is that clear? Right? So you can also write it in the case of humans, right? Just say 22 plus XY. Sorry, my bad. 22 pairs, right? We have 22 pairs. One pair is of XY, na? So 22 pairs plus XY male, 22 pairs plus XX female. And in gametes, how can we write it? Because they are haploid, right? So 22 pairs. 22 pairs means here 44 chromosomes, na? So 22 plus X or 22 plus Y in males, right? In males. Or females में 22 plus X or 22 plus X only. So females are homogametic. Females are having the same gametes. Is that clear? Right? Is that clear? So both males and females bear the same number of chromosomes. So XX and XOY clear. Then male heterogamety. So now we will talk about the female heterogamety from here, right? So here you can see in the case of birds, right? Female birds, they are ZW basically, right? So they will make different types of gametes, okay? And male will have ZZ. Done? So next is the humans. Human part is done. That is like even in the human reproduction also we have discussed. Now in honeybee. In honeybee students we see haplodiploidy. What do we see? We see haplodiploidy. See, in the case of honeybee, you will talk about like basically parthenogenesis. I will not dive deep into that. So basically when you talk about the honeybee, it's a social insect. You know that there will be a queen which is 2n and fertile. So here 2n number is actually 32. Sterile workers are there. They are sterile. They are not going to make the gametes, but still they are also 2n. And then comes the drone. Right, then comes the drawn. So drawn is n, that is it is going to be equals to 16. So drawn grows parthenogenetically. Drawn grows parthenogenetically. Are you getting my point? How this drawn grows? It grows parthenogenetically. It means the haploid gamete grows into adult without fertilization. So gamete is haploid, that is growing into adult mitotically, of course. So fertilization is not taking place. So the adult will also have what? Adult will have the haploid number of chromosomes. So drawn is the male member here. Drawn is the male member here. So you will see, right, this table though is not proper. Just a female 32. Right, female will form by meiosis, female will form the gamete 16 and 16. Now, one gamete is growing into the male. How come? Parthenogenetically. Now from here, male is 16, male, because male is already haploid, no meiosis. So here mitosis will be there, it will form 16. So that 16 or 16 when it will fuse, these gametes, they'll form the 32, like diploid number. So this is the haplodiploidy there. Is that clear? This is the haplodiploidy there. Okay? This is the haplodiploidy there. Understood? Sure. This is the haplodiploidy. Fine. So this is all about the sex determination. So you try to answer this question and then let's discuss the concept of mutations. Quick. Yes, please. So which condition describes the correctly? Homozygous sex chromosome determine female sex in birds? No, females are heterozygotic gametic there. XO will be there in the type of sex determine male sex in grasshopper. True that. XO condition in humans as Turner. In certain Turner syndrome, there will be, okay. So they are asking for what? Correctly, XO condition in humans as found in Turner's syndrome determines female sex. Okay. Homozygous sex chromosomes produces male. So this is also incorrect, right? And XO type of sex chromosomes determine male sex in grasshopper. They are basically, so XO condition. So in Turner syndrome, we also write na 44 plus XO. है ना 44 plus XO. So we should go for the, yes please, because correct sequence they have asked. So we should go for the B, isn't it? Isn't it? Yes, please. Answer it, everybody. Yes, please be quick. So next topic that we have to discuss is of mutation. We will discuss the mutations first. Okay. Done. So B is the correct one here, guys. Right? Okay. And one more thing, Turner syndromes. So it is, you know, that this is also due to the mutation. So we will discuss the concept of mutations first, okay? So genetic disorders mein, yeah, we didn't discuss the Mendelian disorders, but yeah, we have discussed few Mendelian disorders. So we will write about the mutations first. Then I have added the flow charts that will make your, learning comparatively easy and good. Okay. So let's start. Let's talk about the mutations. So what exactly are the mutations, students? Mutations are the sudden changes, sudden inheritable changes that occur in the genetic material. right? These are the sudden heritable changes that occur in the genetic material. That's the first point. Okay, that's the first point. Secondly, who gave the concept? It was Hugo de Vries who gave this concept of mutation. How come? Because students, he was working on a plant. The name of that plant is evening primrose. What's the name of that plant? The name of that plant is evening primrose. What is the name of that plant? The name of that plant is evening primrose. Okay, so evening primrose, the scientific name is Oenothera lamarckiana. He studied that and he reported that, you know, in the next generation, lamarckiana. So he reported that immediately in the next generation, you know, the new species is forming. Later on, when he studied, he got to know that it was the chromosomal aberration, like chromosomal structural changes, I would say, that structural issues were there, right? Chromosomal aberration, chromosome structure was not proper, that is why it was happening. But Hugo de Vries said that you know, mutations are the driving force behind the evolution, and he was the one who believed in the saltation theory, right? So when it comes to the mutation, right, when it comes to the mutation, so one thing that you have to remember, that mutations result in discontinuous variation, means mutations are sudden changes, right? Mutations are sudden changes, so they are responsible for discontinuous variation. Are you getting my point? They are responsible for discontinuous variation. That's what you have to keep in your mind. Okay, they are responsible for the discontinuous variation. Are you getting my point? So the point here is कि जैसे as per the when Hugo de Vries, when he worked on evening primrose, he was very clear with this part, right? He was also very clear with this part that you know, he worked on that plant. In next generation, he saw a completely different plant. So he had that idea that it is all because of, you know, this mutations are making new species. So that is what he believed. Okay, that is what he believed. Are you getting my point? So later on, he got to know that these were the mutations, basically chromosomal aberrations, basically. Now we discuss about its types. So types may we will talk about the chromosomal mutations, right, students, and we will talk about the gene mutations here. There are two types of mutations that we are going to discuss. We will discuss the chromosomal mutations here, right, which will include the change in chromosome structure, number, change in chromosome structure or number, right? And gene mutation may the change is at the level of gene. That's what we have, right? That's what we have. So it is in the number or even in the structure. So here you will see there are two things. One will be heteroploidy. One will be heteroploidy and another is the chromosomal aberration. So one is what? One is the heteroploidy mutations and another is what? Chromosomal aberration. We'll focus on the chromosomal part first. Chromosomal aberrations. Students, when you talk about this heteroploidy mutation, you also call them as genomatic. So if you ever get the word genomatic, it doesn't mean it is a gene mutation. It's a chromosomal mutation, heteroploidy, in which there is change in chromosome number. I told you na, chromosomal mutations are of two types. So here there is a change in chromosome number, right? But here there is a change in chromosome structure, like its structure is not proper. So this is the change in chromosome structure. So that are basically the chromosomal aberrations. Am I clear? That are basically the chromosomal aberrations. Now just to save our time, जैसे heteroploidy mutations, I told you change in the number of chromosomes, and what else do we call it? We also call it as genomatic mutation. So now it is of two types. One is euploidy. Chalo, I'll draw it. Otherwise, I will not feel good. Mutation. So we just discussed about the heteroploidy. Sorry for that. Yeah. So heteroploidy, which is also known as genomatic mutation. Here you'll see one will be euploidy, another will be aneuploidy. Here we have two words. One is euploidy and another is aneuploidy. So in euploidy, there is a change in complete chromosome set. Like it can be added, it can be deleted. Like complete set. Just say I have to give you the example. Let's suppose there is a diploid individual. If one more set of chromosome will get added, individual become triploid. This is the euploidy. Let's suppose two sets of chromosome get added, 4n. So there is the point of the, like the complete set of chromosome will change. When it is the aneuploidy, so there is the change in chromosome number. Either it will be deleted or it will be added. Just say for an example, we have 46 chromosome. If by any chance if one will get subtracted, it will become 45. So that will come under the aneuploidy, right? Just say trisomy, tetrasomy, nullisomy, right? Monosomy. That is how it works. So let's talk about the polyploidy, this euploidy first. So change in the number of sets. So monoploidy, like there is loss of one set. Normally sexual individuals are normally what? Diploid. One set, if it is getting eliminated, 2n minus n, so it is n, right? Then polyploidy mein you will see triploid, 2n plus n, tetraploid, 2n plus 2n, right? So these are the points that we have here. Are you getting my point? This is what euploidy is. And when you discuss this euploidy, I'll just give you one example. The wheat that we eat, right? The wheat that we eat is hexaploid. It is hexaploid, right? It is 6x, right? And it's not mandatory, beta, this x and n. They can be or cannot be seen, right? Just say n tells me about the haploid. Haploid means half. Haploid number of chromosome. N tells me about the haploid number of chromosome, and this x tells me about the number of chromosomes in a set. Number of chromosome in a set. It tells me about the number of chromosome in a set, right? This is what I'm going to discuss now. Just let's suppose we are 2n, we have 46 chromosome, n is 23, and even in our one set, like x is also 23 here. But it is not always. Matlab our x and n is same. One set is having a 23 chromosome and even haploid number is also having 23 chromosome. Now, when you talk about 6x wheat, just say let's suppose 42. Here x will be like one set of chromosome contains 7 chromosome. Right, matlab one set contains 7 chromosome, in simple word. Contain 7 chromosome. Done? One set contains 7 chromosome. X is equals to 7. But if you see the haploid number here, that is n. Because if you have to make the gamete, haploid number n, so that is going to be 21. So that's what I'm trying to tell you here. Is that clear? That's what I'm trying to tell you here. Is that clear? So monoploidy, x and n, they are two different things, right? And the hexaploid wheat that we eat, I told you about it. So that is basically your triticum aestivum, in which 6x is equals to 40. And there is one rye grain, sea kale, right? Just say this is another cross that I'm telling you. So sea kale, that is rye, where 2x is equals to 14. So when these both were crossed, na, so first man-made cereal, triticale was formed. Triticale, right? It is the first man-made cereal. Is that clear? Is that clear? So this is regarding polyploidy a bit. Main is aneuploidy, which is very, very, very important, because here is a change in the number of chromosome in a set. What do you see here? Here you see that there is a change in the number of chromosomes in a set. Just say monosomy. It will be, so two types are there. Hypoaneuploidy. Hypo means less. The chromosome deletion is there. what is there? The chromosome will get deleted. So just say loss of chromosome. So monosomy mein you people can see loss of one chromosome, 2n minus 1. So I'll give you the example here, which is the part of your syllabus as well. Turner's syndrome, right? Turner's syndrome. So 44 plus XO, that's how we write, or you can write it 44 plus 45, 45 chromosomes are in totality, 44 plus XO. One X chromosome is missing. Nullisomy, 2n minus 2, right? 2 chromosomes are deleted, right? So this 2, it's a loss of pair. Like let's suppose one complete pair is eliminated. So let's suppose we are 46, 46 minus 2, that individual will have 46 chromosome. When it is double monosomy, you write it like this, 2n minus 1 minus 1. Matlab this chromosome is from one other set, from another set. Just say, let's suppose, just random example. Let's suppose this chromosome is deleted from the set 14 and this one from 18. So one chromosome is less from the pair 14 and another from pair 18. So it is the loss of two chromosomes. Two different chromosomes are they. They are this is the loss of chromosome from two different homologous chromosomes. But here the complete pair will be removed. Is that clear? Then hyperaneuploidy. Hyper mane extra. One chromosome is extra. Trisomy, 2n plus 1. Just you discuss about the Klinefelter's syndrome. Other than that, one extra X chromosome is there in a male. So male also show female-like character. Plus, in your syllabus, you talk about the trisomy 21. That is the Down syndrome. That is what? That is the Down syndrome. Are you getting my point? So double trisomy is also there. Like two extra chromosomes are added in two different sets. Tetrasomy mein 2n plus 2 will be there. Right? So yes, this is very, very, very important. Now students, when we are talking about the chromosomal aberrations, aberration means change in structure. So deletion, just a part of the chromosome is deleted, then obviously that gene will also be lost. Are you getting my point? Then that gene will also be lost. So one end of the chromosome break, it's lost. If it is terminal deletion, matlab ends, intercalary means in between, right? Then comes the duplication. A segment is, repeated, extra copy is added. Inversion may, let's suppose it was like 1, 2, 3, 4, 5, 6. Let's suppose if it gets like this, 1, 3, 2, 5, 4, 6, inverted. So if inversion is outside centromere, if centromere is not involved, if it is involved, then it is pericentric. If it is not involved, paracentric. That's it. Then next is translocation, which is very important, beta. So translocate. See, this is important. We will discuss CML disease here. So this is important. Just a simple translocation mein what is happening? This is chromosome 1. Let's suppose this is, I'll give you the example in this way. And I'll put one flowchart in our official Telegram group also for this. 1, 2, 3, 4, 5. This is, let's suppose, A, B, C, D. Now, a part of this 5, just go and join here. This is the translocation. So here, it is not there, it will go there. Translocation, simple translocation. Now, it's like I am giving you a gift, you are keeping it, that's it. Now, let's suppose this is another case. Reciprocal translocation, right? This is the another case, which is reciprocal translocation. In reciprocal translocation, this 5 will go here, or let's suppose E is coming here. 5 or E, sorry. So just say, let's suppose 1, 2, 3, 4, 5. This one is having A, B, C, D, E. Now in reciprocal, this E will go to this. Okay? E will go here, 5 will go here. It's like I am gifting you something. You are not giving me anything back. Simple translocation. I am gifting you something, you are returning back, right? You are giving me something again. So that will come under what? That will come under the reciprocal translocation. Right, that will come under the reciprocal translocation. So there is the exchange between, but yeah, do remember there is the exchange of segments between two non-homologous. It should not be homologous. So this is important. The very important example is CML, that is chronic myeloid leukemia right? Chronic myeloid leukemia, right? Right? So it's a blood cancer and blood cancer, and what's that? And do write it, guys. So it's a reciprocal translocation between, this is important, chromosome 22 and 9. Is that clear? Chromosome number 22 and 9. So this is about the mutations. Okay, this is about the mutation. That much will be sufficient for you. There is nothing to worry about. Okay. So yes, the pedigree analysis part we were supposed to discuss, right? But it will take another 2 hours, and we'll discuss it in detail, in depth, right? I have already discussed that with you all. But here, when it comes to the Mendelian disorders, जैसे we didn't discuss the color blindness and hemophilia yet, okay? But students, sickle cell anemia in detail is done and dusted. Phenylketonuria, done and dusted. Cystic fibrosis, done and dusted. So when you talk about this thalassemia, na, I'll discuss the thalassemia here. Thalassemia is also, see, thalassemia and sickle cell anemia. Thalassemia is quantitative, sickle cell anemia is qualitative. So you guys can see here, this is also an autosomal linked recessive blood disease, right? It is autosomal linked recessive, right? From and transmitted from parents to the offspring when both the parents are or partners are unaffected carriers. Is that clear? Both are heterozygous, they can pass it to their F1 generation, their baby. So how can this defect occur? When there is either the mutation or the deletion. Either the mutation or the deletion that can result in reduced rate of synthesis of one of the globin chain. Means the synthesis of globin chain, its rate is less. Yeah, alpha can be less or beta can be less. That's what I'm saying. So here the globin synthesis, it is not defected. It's like its secretion is less. That's why for thalassemia I clearly told you that it is quantitative, because once there was a comparison in these two. Now you know that there is a formation of abnormal hemoglobin resulting in anemia, which is characteristic of this disease. So in alpha thalassemia students, production of alpha globin chain is affected, and in beta, beta chain. So when it comes to alpha thalassemia, because alpha hemoglobin chain is affected now, so this alpha chain is affected by two genes, HbA1 or HbA2. Where is it located? Chromosome 16. And we discussed it, HbB gene, which will make beta hemoglobin chain, that is located on chromosome number 11. So when it is the alpha thalassemia, this information is important. So chromosome number 16 gene is HbA/HbA1. When it is beta thalassemia, so single gene HbB on chromosome 11, right? It is having the mutation. Is that clear? So thalassemia differs from sickle cell anemia because thalassemia is quantitative, right? Less hemoglobin molecule is there, and sickle cell is qualitative. Baki beta, this is the Down syndrome. The symptoms are important. You can directly revise it from there. Just it is trisomy 21. There you can see the karyotype. You can see one extra copy. You can see one extra copy of the chromosome 21. So broad flat face, flat back of head, many loops on fingertip, palm creases are there. Congenital. Congenital means by birth. By birth heart diseases are there. Big and wrinkled tongue, broad and flat face will also be there. Is that clear? Is that clear? Then you can see further, just here trisomia. So Down syndrome done. So I told you this disorder was first described by Langdon Down 1866, right? So individual will have short stature, small round head. Diagram is also important, furrowed tongue and partially open mouth. So from that figure also you have to pick up the symptoms. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Am I clear? It is retarded. Okay. Then Klinefelter, I told you, one extra copy of X chromosome. So male will show female like characters, right? Just say tall stature with feminized character. Here male will also show gynecomastia. So gynecomastia means the mammary glands will start developing due to the extra estrogen. So XSY here what will happen? Masculine development is there, but development of breast, that is gynecomastia, will occur in the male. So it is also expressed. So so here individuals are sterile, okay, because male will have extra estrogen. That is why gynecomastia, right? Male will be sterile here. And in Turner syndrome, where it is where it is 44 plus XO students, so short stature and underdeveloped feminine characters are seen in the Turner syndrome. Is that clear? Is that clear? Right? So this is about the chromosomal disorders, thalassemia part, sickle cell anemia part, all that is done, right? So hemophilia and color blindness, this will be discussed with the pedigree analysis. So stay tuned for this. That part will also be, because that part actually needs the separate attention. Like last time also question was there from this part. So definitely we will discuss it, but separately. Okay, but separately. So I hope guys you have liked the today's session, and trust me, in today's session I have discussed everything in detail, right? You got all the info, right? I have literally provided you everything, every information that you need, that you genuinely need to crack the NEET examination. But now all you have to do is you have to revise it, you have to practice the question, you have to consistently, right? You guys need to consistently attend the marathons and to revise it. So I'll share the PDF because so many handwritten notes are there. So Wasim sir will share the PDF, right? Yes. So you guys will get the PDF as well. So make sure you download the questions, right? Make sure you download the questions and download this PDF, solve the questions. So that is important, okay? That is important. So keep practicing it. So practice is the most important thing. So pedigree analysis, it was planned, like I will take it separately because that topic needs a lot of attention. Okay, so that you will get, there is nothing to worry about. Secondly, right, whatever is required, like we have covered the NCRT, we have revised the NCRT, right, we have practiced the questions as well. So keeping NEET 2025 level in my mind, I have discussed the questions accordingly. There are more questions also. For that you can just join our Unacademy platform. The special classes you can join, they are free of cost, but there we practice a lot of questions. So I'm still energetic, right? I'm still energetic. I can just take the class for another 2 hours also. That is not a big deal. That is not a problem at all. Trust me, that is not a problem at all. So, but all I want is that whatever we have discussed so far, please revise it. Okay, revise it in detail. So hemophilia, color blindness, in short videos you are going to see that. Pedigree analysis in detail, you people are going to see that soon. Mutation, whatever part we have completed, that is sufficient. Gene mutation is discussed in the molecular basis of inheritance. Okay, so gene mutation is a topic there. Okay, gene mutation is a topic there. So PDF will be provided to you, and if you genuinely like the session, right, then do let me know in the comment section and keep practicing, guys. Okay, keep practicing. Done? Chalo, so see ya. Bye-bye. Take care. Okay.